Okay, so now we can start. We've got the housekeeping out of the way. So first thing to say is that it's great to see so many people here. So at the moment we have over 175 participants in the meeting. We had over 700 registrants for this, so it's great to see that there's interest in this symposium. One of the things since I took over as president of the International Society of Root Research, one of the things we've been trying to do is to increase the activity of the society between the conferences. So a lot of people here will have been to the conferences that happen every 3 years or so. So the last one was in Leipzig in 2024, and the next one will be in Beijing in 2027. And it's great that we have so much engagement at those conferences, but what we're trying to do is to increase that interaction between those conferences. And that's exactly the purpose of this symposium today, is to have the opportunity for the community to come together and discuss root research on a specific day for the society, and just gives us some broader activity between the conferences. So in order to do this, what we've done is we've also developed a new set of working groups. So we have 7 different working groups within the society, and we have a range of early career researchers and younger research leaders who are leading those working groups for us, and trying to develop programs of activity that can increase the interaction within the community and on those specific areas. So we have 7 different groups. We have root anatomy and architecture, and you can see all of this on the website. We have roots and ecosystem function, rhizosphere and signaling, phenotyping method development, root modeling, roots in sub-Saharan Africa, and root systems biology. So today we've asked each of those working group leaders to develop a session for this mini symposium. So each of those working groups will have 2 talks representing their area, and these have been organized by the working group leads. Those working group leads will chair the sessions, their relevant sessions throughout the day, and so you'll get to see them. We've also put in place some breakout rooms, so that in the coffee breaks, if you want to talk specifically to that working group leader, then you can follow the breakout room to that particular working group. And if the working group leader is still in the meeting, then they'll be there to talk to you about how you can engage with that working group, for example. So we'll reserve that until the coffee breaks. But basically, we've got 7 sessions, for the whole of the day. We'll start in a couple of minutes, but then we expect to be finished by 4:30 UTC, so in about 6.5 hours or so. And like I say, we have a number of breaks during the day so that you can take a rest. And also remember that this is all being recorded, so if you want to catch up at a later date, then you can. So with that in mind, then really, as I say, the whole purpose of what we're trying to do today is to increase the engagement within the society and with the root community at large, in between those conferences. And it's really gratifying to see so many people here at the start of this. And again, like I say, over 200 people just now. And it's great to see so many people register for the event. So without further ado, I want to hand over to Root Anatomy and Architecture Working Group for their session. So I'm aware that Gwen is not available. So is it Xavier who's taking this? Yeah.
Yeah, sorry.
No worries.
Hi. Hello, everyone. My name is Jasmine Reyes. So I'll be hosting our first session today on behalf of Gwendolyn Krishna, which unfortunately couldn't join today. As Timothy said, so our working group is focused on root anatomy and architecture, and we are really happy to be the first one to kick off our meeting today. And we have two talks in this session. So as we mentioned before, so each speaker will have 20 minutes for the presentation, and it will be followed by 5 minutes of KNU and A. So please write the questions in the chat during the talk, so then I can read it afterwards. And yeah, with this said, I think I would like to start with our first speaker, who is Professor Masashi Yamada from the Biotechnology Center in southern Taiwan. So his work focuses on plant development and signaling, and it includes how rods help to shape root growth and patterning. So I think with that said, so Professor Yamada, thank you for being with us today. And please go ahead to share your screen, please.
Thank you very much for the nice introduction. I try to share for the my slides. Can you see the slides?
Yes, we can see it.
Okay, and okay, I'll start my talk. Thank you very much for inviting me such kind nice symposium. My name is Masashi Yamada. I'm from yeah, I'm from Academia in Taiwan.
Sorry, if you can mute your microphone, please. Thank you.
So can you hear me?
Yes, now we can hear you. Thank you.
Okay. So today I'm going to talk about role of root mesenchymal growth factor peptide in root mesenchymal development. So first of all, I just do the brief introduction, like Arabidopsis root development. As you can see, like Arabidopsis root is a very simple structure, like each different color is a different cell type. And here the root tip, and this blue cell is a stem cell. Like each layer have a stem cell, like producing have a different cell type. And also like easily identify for the three developmental zone. Like one is a mesmatic zone, the elongation, like a differential zone. So mesmatic zone is a very active cell division. And then after that, the cells start elongation, like we call the elongation zone. So finally, here is a differential zone. So these three developmental zones are very easily identified under microscope. And then also like this development zone is not always same size, but after coming to internal also external signal, like these three development zones change of size. So we try to understand how this regulation happen. So in the lab, we focus on a small peptide called root mesenchymal growth factor, like we call the RG1 peptide. So this peptide is originally like synthesized like for polypeptide, like over 100 amino acids. So during for processing, they produce have a small peptide like 13 amino acid. So second residue is a tyrosine, and also tyrosine always sulfation by TPST enzyme for the change for the modification. So this peptide is a secret peptide in a peptide going out cell. And then this peptide for the bind to receptor like this. And then we already understand for the downstream signal like this. So mostly have a algebra family member like a regulating for size of a root meristem in the primary root. And also we already know like other family member like inhibit for the lateral root development. And also recently find like a leaf like immune response signal. So here is the member of RG1 peptide. It's all peptide is encoding for some mature peptide is only 13 amino acid. So second residue is always tyrosine. Tyrosine sulfation is the activate of this peptide function. And the 3 gene is the Arabidopsis redundant system, and also have a 3 gene, RG1, 2, 3 is very similar. And also like expression is very specific, like only root tip have a high expression to these 3 gene, and mRNA is highly localized for the root tip around the QC cell. So this is in situ hybridization data. And also have a phenotype is a very redundant, have a single mutant is almost no phenotype. After making triple mutant, they have a very small mesosomatic zone. So this phenotype of rescue by peptide, external synthetic peptide treatment. And also like this peptide treatment, almost same effect, overexpression like a whole gene. So suggesting is that mature peptide is working at in home. And also they are regulating for the mesosomatic zone size by dose-dependent control. So here is a phenotype of only 24 hours treatment, have a white type plant for Arabidopsis root. You can see like a mesosomatic zone like a very big, and then even treat of a whole plant. So we can see the very strong effect of the only root tip. And previously, also we already know like a receptor, like a specifically localized for the mesosomatic zone. So that's why like after we treat peptide, and then I try to understand for the downstream mechanism, only root tip, so that we correct for the mesosomatic zone after peptide treatment, like a very small tissue, and then extract of the RNA from this small tissue, like to see the what kind of gene like a change for the expression in the RNA-seq analysis. So we did that for the RNA-seq analysis, like a DG analysis compared to the mock versus RGF1. We found that almost 500 genes like upregulate or downregulation. So after gene ontology analysis, we found that many genes like related to the ROS signal, like a reactive oxygen species signal. And also like previously, we already know like 2 ROS. One is a superoxide, and hydrogen peroxide is very important to regulating for the root development. So white type have a normal growth condition in Arabidopsis root. So you can see like a superoxide have a highly accumulation in the mesosomatic zone. And the hydrogen peroxide start accumulation to the elongation like a differential zone like this. So previously paper like talking about for the if we change for the this distribution, we can control the size of meristem. So that's why like I expected probably like a RGF1 control to the ROS signal. So beginning like I try to understand for the hydrogen peroxide localization. So I use hydrogen peroxide base AC. This is a fluorescent indicator. So this chemical like originally have a transparent chemical. After react to the hydrogen peroxide, you can see like a green fluorescent signal like this. So we take a picture for the confocal, and this is a superimposed picture like this signal like for the combined to almost like a 30 layer image. And here is the surface, the cell, and you see that this is like for the root cap, we call the outside cell. And here is one cell, like a very long cell and a very high signal to the hydrogen peroxide. But small cell is relatively like a lower signal. And also like we go into the deep section inside of a cell. So this small cell like almost like an undetectable signal. And also here like a start elongation and also differentiation. Like a big cell usually have a high accumulation like a hydrogen peroxide like this. So and then correlation to the elongation differentiation versus for the hydrogen peroxide localization. Here is for the after peptide treatment. Here is you can see the very dark spot in here, and then also have you can see the many small cell. So in case of RGF1 treatment, they produce so many small cells, and then you can see the very big dark spot. So suggesting is that RGF1 like reduce hydrogen peroxide and produce many cell, many small cell. So and also like a significantly reduce hydrogen peroxide signal. And then next, like we detect the superoxide localization by NBT staining. So NBT staining like a highly accumulation to the mock treatment in a Y type, the Arabidopsis root. After RG1 treatment, we detect the NBT staining, like you can see the very broadly localized for the superoxide signal. In this case, is the superoxide originally accumulation to the like a young tissue and then increase more small cell by RG1 treatment. You can see like a broad localization of the superoxides. So superoxide also like have a correlation to the cell development, also like a distribution. So finally, we understand for the highly accumulation of the superoxide like this. So the normal growth condition like a superoxide accumulation to the mini-symposium zone, and they start like accumulation to the hydrogen peroxide accumulation, the elongation like a differential zone. After peptide treatment for that, they change for the disbalance. They produce more superoxide, they reduce have a hydrogen peroxide. And then finally have a mini-symposium zone is very big. So, and then next question, like how this ROS like modulate for the root meristem sites. So we try to understand how that mechanism, and then also like we already know like two transition factor, like a pre-solar one, and then two is a measure for the root meristem for the master regulator, and they are very similar to the pre-solar 1, 2, and also they are localized like a root tip for the meristematic zone, and also have a they are redundant phenotype, have a compared to Y type, have a single mutant. The root length is almost same, but after making a double, like a root is suddenly have a very short, and also meristematic zone like a very small. And also we already know like for this protein localization like a pretty much overlap in the root tip. So what I just wondering for the which one like a downstream of a RG1 peptide signal, and I try to treat for a peptide for the in this single mutant background. So Y type have a like a response to RG1 produce have a very big meristematic zone, and PLT1, PLT3 mutant also have a larger meristematic zone compared to mock treatment. But interestingly, the PLT2 mutant are pretty much resistant to the peptide treatment. So meristematic zone size is still similar, even treat after RG1. So that's suggesting that PLT2 is one of the target of RG1 signaling pathway. And then next time we look for the reporter gene expression, like using for promoter CFP, also like a whole genomic PLT2, the translational fusion line. So first we look for the promoter, like a CFP of a promoter reporter line. The PLT2 also like localized around for the root tip in the in situ hybridization. Like this promoter activity is almost same. And after peptide treatment or even big meristem, and that reporter is expression like still very limited to the root tip. And this is the translation fusion line for the YFP. So you can see the very bright signal in the root tip because of the expect of the newly synthesized protein from mRNA, but other later like they have no mRNA and the protein start degradation like this, and then finally they are making a gradient like this. But after peptide treatment, you can see like this is you can see a very big meristem here, and also like a protein localization like a pretty much broad like this. So it's suggesting is that peptide treatment about post-translational regulation to the make more stable to the protein. So finally, we understand for the ROS after peptide treatment, we can change for the 2 ROS and also like a protein stability control. Like we try to understand how this ROS regulating for the PLT2 protein stability. So first, like we use have a DPI. This is NADP oxidase inhibitor. Try to inhibit the superoxide production to see what's going on like a PLT protein stability. So we use the DPI for inhibitor because this is toxic. Like we relatively use the lower concentration, like make sure like this DPI concentration doesn't damage about root development. And after that, we compare to the PLT1 single treatment versus RG1 with the DPI double treatment. you can see the protein is a more or less accumulation, like a DPI inhibit for the protein localization. So next, we used another inhibitor, like a hydrogen peroxide scavenger for the potassium iodine in like a reduced hydrogen peroxide signal, and to see that what's going on like a protein stability. So this case is RG1 single treatment versus RG1 plus potassium iodine. You can see the protein is very broadly localized. So suggesting that reduced hydrogen peroxide, the proteins make more stable, like a broadly localized like this. So finally, we understand for the using for two inhibitor, one reduce hydrogen peroxide, and in this case, proteins more accumulation. In the time that we can expect the hydrogen peroxide as a negative regulator. And also we use DPI inhibit superoxide production. This case is the protein is negatively regulating. So that's why
也是谁呢？客服啊？
superoxide have like a positive regulator for the protein stability.
你从
So
订单
next-
里找客服，直接不就是显示你这个订单了？
ROS regulating for the protein stability. So actually have a cysteine residue in the protein is very sensitive to the ROS. They are related to the protein modification, also protein stability, also like a protein activity in a many different organisms. We expected probably like a cysteine in a PLT2 have a key regulator to the
We're killing the microbes in the soil, and we're probably
stability,
killing the
the
microbes in our
ROS.
stomachs, which are there to defend us
Sorry.
from disease.
Sorry, Michelle. Could I just make sure that everyone is muted, please? Because whoever is speaking is disturbing the speaker. Please make sure you're muted.
So luckily, like a PLT protein have two cysteine. One cysteine is a DNA binding domain, another is a outside DNA binding domain. We replace this cysteine to serine to see that how this cysteine affects the protein stability. So we make the construct of the Y type GFP with a Y type PLT with GFP and transform this construct to the PLT mutant background. And this construct rescue the PLT mutant phenotype, and also like we can see that one layer of a confocal picture. You see that here is a very bright protein that start degradation, and after like a cell elongation, cell differentiation, so this signal is almost gone. But this like a construct, only one cysteine change. Promoter, exon, intron are all the same. So, but this protein is more stable. Like you can see that even have a longer cells still have a protein localization. And also here is the boundary for mesmatic zone. You see that mesmatic zone actually have a bigger than Y type, like a PLT2 localization line. So PLT is more stable and then make more bigger mestem. And also like this, about Y type quickly reduce the protein level, but like a C212 mutation have a reduced more like a protein degradation, like a very slowly reduce of a protein level. And also like a next time for the, we treat peptides for the in like a background of this like a transgenic line. So here's a mock treatment, 0.3 RGF1. Here's a 5 nanomolar like RGF1 treatment. So these proteins are dose dependently localized, have a and broadly localized for the mesmatic zone. So here is a C212 mutant line, have a very sensitive to the RGF1 treatment. So even 0.3, like this protein is already broadly localized more than 5 nanomolar like RGF1 treatment in the Y type. C112 also like still like a response more sensitive to the RGF1 treatment, but it's less sensitive to the C212. And the double mutant have also have almost same like a response to the C212. So like suggesting the C212 probably have a main the system residue to that related to ROS response. And next time for we treat like a hydrogen peroxide, very high concentration, like we use 1 millimolar, like also completely inhibit root growth. And then like this hydrogen peroxide treatment, like start the destruction of the protein. So, and also like a C112, another like for cysteine mutants have a also very sensitive like a Y type. Root is very short and also have a proteins that start degradation. But surprisingly, like a C212 replacement have a very strong resistance to the even treat very high hydrogen peroxide treatment, and also double mutant also root is very healthy. GFP localization is almost same. So suggesting is that hydrogen peroxide targeting for the C212 make some modification maybe related to the protein degradation. And also like we make sure for the protein level, like using for the Western blot, for the cutting for the many small little tip, because protein localize only little tip, and then we can make sure that actually protein level increase by RGF1. Also like we make sure to the protein degradation about hydrogen peroxide, and about C212 line is a very stable protein. And also like next time for we try to detect for the like a downstream gene expression after the peptide treatment localized very broadly PLT2 protein, because it's also mutation is a DNA binding domain. We just expect for the they change for the promoter binding or they change for the target gene. So we do the RNA-seq analysis, these 3 different genetic background and also 3 different consideration about RG1 treatment. So, and here is a Y type of a 5 nanomolar, like you can see like almost 3,000 gene, like up and down and regulation. And here is even 0.3 in the mutant line, like almost 4,000 genes up and down regulation, suggesting is a highly accumulation proteins more regulating for the gene expression. And also next time for the use just the only specific gene expressing for the mesomatic zone, like how much control by these 2 lines, Y type versus for the C212 mutation. C212 mutation that increase more like a mesomatic zone specific gene in the mock treatment. After 0.3 treatment, like J increase more like a expression to the mesomatic zone specific gene. So 5 nanomolar is kind of saturating for the both is increase the more like a mesomatic zone specific gene. And next time for look for the elongation zone specific gene. So this case is a pretty much reduction to the C212S line, and also like a 0.3 is a highly reduction to the elongation zone specific gene. So finally, we compare to the Y type and the C212 line, like how many genes have a downstream. So actually, like a downstream gene is almost similar number, like almost like a 90% is overlap. So suggesting is that that mutation doesn't change for the promoter structure, like binding, like still target gene same, but probably like a value, like expression value is different because based on the protein amount. So like this is we compare to the expression level to the same gene, and actually for the C212 increase more, because probably have a based on the higher protein amount, and also like a same gene they reduce more like a C212. I think it's also like a more protein reduction to the downstream downregulated gene. So finally, we try to do the what cell event happen after peptide treatment in a different genetic background. And we found that for the geoanalysis, have a cell proliferation genes upregulation, and cell related for differentiation, like a more downregulation. So also we make sure this gene expression by qPCR. So finally, we understand for the PLT to have a highly correlation to the stability based on the ROS localization, and RGF1 like a change for the this ROS distribution, and finally like a PLT2 localized more broadly localized. And this is our model have a root is always have a growing PLT mRNA synthesized like protein. But like in the future, this protein is going to the hydrogen peroxide area. They start for the protein degradation by oxidation. And also we already detect for the this modification in the mass spec analysis. But if PLT have no sustain, they are more stable and then like making more broadly localized and then making a bigger meristem. And also like a peptide treatment for even lower than hydrogen peroxide and then white type proteins are broadly localized, but mutants have no system, even more broadly localized. So this is our model. And also like now is we already understand what the downstream have a transition factor. Like now is we try to understand how this transition factor regulating ROS. So I came to the Taiwan from the US in 6 years ago, like start my lab, and then like always have a recruiting for the PhD student and a postdoc. Like if you interested in our research, please contact us. Thank you very much for the listening. So please ask question like somebody. Thank you very much for the listening.
Thank you very much, Professor Yamada. Let's see, we have time for 1-2 questions. If you can please write them in the chat, I can read them. I can start maybe while people start to put some questions in the chat. So I'm curious, it's really nice to see how this plethora genes can be regulated by this signaling. And I wonder if you have an idea of how do cells perceive this gradient of ROS? Do you have like think that there is a missing key regulator to integrate this information?
Okay. Yeah, that's a very nice question. Like for actually, this research we understand for the how ROS change for the PLT localization, but we still don't know like how this signal control for the ROS. So we just find the transition factor. So the transition factor cannot directly control ROS. So also we try to understand the downstream this transition factor. So right now, and try to understand for the how ROS change, but still have a we have a candidate, but still we don't have any clear for the pathway they are using, and also have a 2 ROS change for the different the development zone. I think maybe they are using for the different system for the different development zone, and then maybe different gene. But actually, I still don't know. Like that's very good question. Actually, I really want to focus for that this kind of thing. For that now is we try to do, but still we don't understand yet.
Okay, thank you very much. So, well, I'm sure there will be more questions coming. Please stick around. Probably Professor Yamada can follow up with you some discussion during the break rooms or these meetings.
Okay.
Thank you very much.
Thank you very much.
Then to try to keep this on time, let's move to our second talk. So yeah, so I'm going to is Professor so now our second talk is given by Professor Jürgen Kleinenben from the University of Freiburg. As you know, he explores how plant growth is regulated and how this occurs to different cellular levels. And he will tell us more about auxin dynamics and root plasticity. Thank you very much for being with us, Professor Jürgen. So the floor is yours.
Yeah, I would like to thank you and also Gwen to give me the opportunity and to join this this online symposium. I have to say I feel a bit like a flashback to Corona era. So standing in an empty office and talking to myself in front of a screen. But I think it's a great opportunity to share our work with a wider international community. So I'm very happy about this. And the work I would like to discuss today with you, yes, there we go, basically deals with signal integration. So we really like to understand how different signals are integrated into architectural traits. And what we did in the past typically is like we looked at one of these input signals and we followed what it, if we change this input, what it does to the system. And we only start to begin to actually trigger two different input signals and see what it does to the system. And I believe that this is a general aspect in the field. So we typically concentrate on one thing, and maybe by concentrating on one aspect, we actually completely lose how signal integration works at the systemic level. And this is something that I want to discuss with you today. And it's largely unpublished work. I have problems with changing the slides, but let's see how it goes. It's largely unpublished work done by Chengzi Ren, a very talented doctoral student in our lab. And he looks into root organogenesis as a model to see how different signals are integrated together. And here in this aspect, he's using temperature and light. And in order to look at root organogenesis, so de novo organogenesis, so it takes, of course, time. And here we will look at a very particular tool that allows us to look at growth and signal dynamics over time. And here we use an auxin-responsive promoter element. It's called DR5. And this one is driving a luciferase. So we look at bioluminescence over time. And you see here 3 spots. So these are spots where lateral roots are developed. And you see that auxin signaling maxima kind of nicely marks these spots where we do get a development. But if you look carefully at this movie, you also see that there are other spots, and we eventually lose the spots, and there's no lateral root development. And this is exactly what we want to look into. But it's a bit difficult to look always at these movies and explaining what is happening there. That's why we transform this movie into a 2D image, and that's a so-called chymograph. And I will try to talk you through this because it's very important to understand what we are seeing here, because you will see a lot of chymographs in this talk. So we have here basically the segmented root from top to bottom. So all the root segments on the top, younger root segments on the bottom. That's why you see also this growth pattern here. And then you have the signal intensity from the left to the right. And these auxin maxima that you see here, these spots, correspond to a line in these chymographs. And whenever you see a line, so this is a pre-branch site, and if this pre-branch site is around for 3 to 4 days, then we will have a lateral root development. And this we also call like a persistent PBS, or persistent pre-branch site that has auxin signaling all the way through the time that we observe here, and then eventually we see the lateral root coming out. But we also, as I pointed out, we have some spots where we have initially some auxin signaling, but then in time we lose the auxin signaling, and that we call transient PBS. And in this case, so without auxin, they cannot develop into lateral roots. And the question that we're having here, or that Chengzi has, is he uses this system, so basically a decision, to understand how systemic signals are integrated into this local response, so into this local decision. So how and what are the signals that kind of regulate this? And looking for systemic regulations, Chengzi figured that maybe it could be even auxin itself. Because when he looked at these chymographs, he realized that auxin is fluctuating. So if you look, for example, here in this pre-branch site, so there's a high auxin signal, low auxin signal, high auxin signal, low auxin signal. And even in between these PBS sites, so you also see like higher signal, higher signal, higher signal, and lower signal in between. So there's an oscillatory behavior. And if he plots these signal intensities over time, he sees that these signals are actually overlapping. So it's a systemic input here. And he looked at the frequency, and we see that we get a period of roughly every 24 hours, we get these systemic auxin peaks. So in the beginning, we didn't know what it is good for, but we kind of hypothesized that maybe this oscillatory behavior is contributing to the identity of the PBS and decides whether the PBS can be maintained and develop into lateral roots or not. So we can tune the signal intensity by line intensity. So I guess it's just an open microphone.
Sorry again.
So here we have
sorry, Jürgen, can I just make
our normal light condition where we have this nice oscillation. If we reduce the light intensity roughly by half, then we still see the same period. We see the oscillation. It stays 24 hours, but we see that the amplitude is clearly reduced. So this is the main thing that we see for light intensity. So light intensity can set the amplitude of this systemic oscillation. And if we go further down with the light intensity, we can go to a condition where we completely lose the systemic input. So you see this black screen here. So auxin is still there in the lower part of the root, defining pre-branch sites, but these pre-branch sites we lose when we also lose the systemic oscillation. And while in the higher light and medium light condition, we see that, that roughly the number of persistent and transient PBS is the same. Once we lose the systemic oscillation, we get a dramatic change, and we have mostly transient PBS leading to a more or less naked main root. Then Chengxi turned actually to high temperature responses, and there it started to become really interesting because we see multiple dynamics when we grow them into high temperature. So what you see here is a control condition, again at 21 degree. And this recording here starts when we transfer the plants to 29. The first response we see is that there is a ramping up of auxin response in the area where these pre-branch sites are defined. That's why we also get quite a number of pre-branch sites established. But these sites are initially transient. And they are transient likely because we get a systemic downregulation of auxin. So you see this nice black area here. And this downregulation is followed by a systemic peak of auxin signaling coming back. And this signaling coming back correlates with a reactivation of some of these previously established PBS. So we assume that here high temperatures leads to a resetting of the systemic aspect. And interestingly, this resetting reactivates a number of PBS that is very similar to the number of PBS that we get in if we just keep the plants on 21 degree. And the consequence is actually that if you just look at these two plates growing at 21 or the ones that are shifted to 29, based on the root system architecture, we see hardly any difference, at least in terms of lateral root number. So the hypothesis here was that high temperature induce as a recovery of systemic auxin, and this is safeguarding lateral rooting. So Chengxi said, like, okay, so now I would just have to downregulate the systemic recovery, and then I can look whether this theory is right. And as he knew that he can downregulate the systemic level by changing the light intensity, he combined these two aspects. So while we have this typical high temperature response at the higher light condition at 29 that I just discussed, he then did the very same experiment, but lowering again the light intensity roughly to half of the control condition. And initially the response is very, very similar. So we get a ramping up of auxin response in the area where the PBS are defined. We get a high number of PBS initially formed, but then we have a systemic downregulation of auxin, which is followed by a systemic peak of recovery. But there are two differences. So one thing is this systemic peak, the recovery takes longer to occur, and the amplitude of the recovery is much lower than if we grow them at higher temperature. So again, suggesting that light regulates the amplitude of systemic recovery. And as a consequence, what we see is that the number of PBS that are reactivated is much lower now at the lower light intensity. So here's the medium light. And this can be also reflected by looking at the root system. So you see that in contrast to what we saw just before with the higher light, now the 29-degree shift by lowering at the same time also a bit the light intensity has a tremendous effect on the lateral routing, so severely inhibiting it. So what we have now, we see that there are two signals, and they get integrated with each other. So the high temperature response, so the initial response, so the perception and the change in auxin is very similar at high or low signaling, but the consequences for the recovery of the systemic signal is different. So meaning if a plant sits outside in sunlight and faces 29 degrees, it will not inhibit lateral roots. But if the same temperature is faced in the night or in a cloudy environment, then it will drastically reduce it. So having a mean to regulate whether the temperature is already a stress response or not. So now Chengzi wanted to go on and he asked, okay, what is the molecular mechanism here that integrates these two responses into the systemic auxin oscillation and thereby the lateral routing? And I think he did a quite smart move here because he combined two aspects at the same time. So he did auxin profiling, so targeted metabolomics, and he combined that with transcriptomic analysis, so with an RNA-seq analysis. And for that, he synchronized seedlings. So he took 7 days old seedlings that were grown at day-night shifts. And he transferred them to constant light to synchronize them. And he grew them either at 21 or 29. And then he harvested every 6 hours to looking at the dynamics of both the auxin metabolism as well as the transcriptional response. And one of the first important finding he had then is actually that not only auxin signaling is oscillating, but endogenous auxin itself. So we have here the free IA, so the indole 3-acetic acid, which is let's say the most important endogenous auxin. And you nicely see that it has like a 24 hours oscillatory behavior. And this oscillatory behavior allows us also to look now at the downstream genes, and we see that with this DR5 signaling reporter, we have a lot of auxin-related genes that are in phase with this, and we also see that, and we also know now what works in antiphase. So there are also some transcriptional regulators of auxin that also works in antiphase, and that may explain the stability of the oscillation. And both aspects are actually sensitive to high temperature. So we lose, if we shift plants to higher temperature, we lose this oscillatory behavior of auxin itself. And we also lose the oscillatory behavior of these auxin-related genes. But there's much more happening. So it's not only that we lose the auxin behavior, but we lose rhythmic transcription in general. So it's a general response. It's not specific for auxin. So suggesting that there's a systemic mechanism in place here. And one of the systemic mechanisms that could be in place, I think also thinking of the fact that auxin is oscillating in a 24 hours rhythm is of course the circadian clock, because circadian clock is actually responsible for such oscillatory behaviors. So if we just have a very brief look at the circadian clock and how it works, so there's a very complex interaction between the different components, and this interaction allows actually these components to oscillate and to regulate transcription in an oscillatory fashion. And we can discriminate here between the morning loop and the evening loop. And now, thanks to the fact that we combined auxin profiling and transcription, we can actually look into our data set and see what correlates here from the clock with our auxin measurements. And then we see very clearly, if we look at this oscillatory behavior of auxin, we very clearly see that the morning loop components, so CCA1 and LHY, correlate very nicely with the dynamics that we see for auxin. So looking into the genetics here, then we see a full agreement with this tendency. because if we choose conditions where the control shows a sensitivity to the high temperature, using a medium light condition, in these conditions, both single mutant CCA1 and LHY are largely resistant to this effect. So you see also here in the root assay, so they do respond to the high temperature in terms of main root growth, but they do not respond in terms of lateral rooting. So the response is downstream of this morning loop. Chengzi also tested other circadian mutants where he sees that they are largely still sensitive. So it seems to be really hitting this loop, correlating with profiling data. So in our profiling data and in our transcriptional data, we of course also can look now at regulators that could contribute to the regulation of auxin. And one gene that showed both circadian oscillation as well as that it lost it in high temperature was the DAO1. And DAO1 is an enzyme that is able to oxidize either auxin itself or an auxin that is conjugated to an amino acid. And the consequence of this oxidation is that it is not reversible, and these molecules are no longer active auxins, so it's a way to degrade auxins, the oxidative degradation. So we got interested in DAO1 in particular because not only did we see that the gene was oscillating, but also oxidative degradation products of auxin showed a very similar oscillatory behavior that we before already saw for the free IAA. Yeah, here as an example is a OX-IAA that is conjugated to aspartate. And in both cases, in case of auxin, I mentioned that already, that we lose the oscillation in higher temperature, and also the dynamics of these degradation products is also changing upon auxin. So suggesting that this is a very interesting branch and candidates to look at. Chengzi went on, and he actually placed now the DAO1 promoter under our reporter system, so using the luciferase. So again, allowing us to look at the dynamics of this expression over several days. So in this case, 3 to 4 days. And what is very clear is that indeed, yes, so we see in time this very nice oscillatory behavior of this gene, and we also see that it oscillates roughly in a 24 hours fashion. And this behavior completely changes when we transfer the plants to high temperature. So again, the recording started at the time of the transfer, and we see immediately this oscillatory pattern is gone, and we see a ramping up of the signal. So it gets continuously everywhere stronger, and we also see that particularly in these, so we see a lined fashion here, so suggesting that particularly in pre-branch sites, the DAO1 is now upregulated. And what we see in time, we see a systemic downregulation of the expression of DAO1, which is kind of reminiscent to the DR5 peak that we saw before. And after the systemic downregulation, then basically the systemic signal and later the oscillation of the DAO1 is getting back. So what might be the consequence for that? So looking at the DAO1 mutant, in the standard condition at 21 degree, we see in the control this nicely oscillating auxin signal. So again, we look here at the DR5 promoter. In the DAO1 mutant, we do not see much of a change. So we still see auxin is oscillating. The amplitude is very similar, and also the period is very similar. So we have to conclude here that at least for the normal oscillation at standard condition, DAO1 doesn't seem to be essential for the systemic auxin. But the picture changes quite a bit if we draw our attention to high temperature, because now so typically in the wild type, we see the systemic downregulation of auxin. This is not what we see here. And typically in wild type, we see the systemic recovery of auxin. This is also not what we see here. Instead, what we see is that we do not get a downregulation of auxin in most of these predefined pre-branch sites. So leading to the situation where in this light condition, high temperature induces a transient nature of this PBS, while here in the DAO1 mutant, they seemingly don't care about it, and they keep the identity of the PBS. Which basically leads also to the fact that at the root system level, so while the wild type shows a strong reduction at medium light when we shift into high temperature, and this reduction is severely disrupted in the DAO1 mutant. So suggesting that DAO1 controls the light context-dependent root acclimation to the high temperature. All right, what could I show you in these 20 minutes? So I showed you that the Auxin oscillator controls the progression of a pre-branch site. So it's a bit like a checkup system. So if the pre-branch site see several of these oscillations, then they actually are allowed to go towards lateral root development. And we know that that light regulates the amplitude, and I didn't show you the data, but Chengzi could show that the light is perceived in the shoot via the phytochrome B and cryptochrome receptors, and this shoot-derived signal is coming via the root clock to control the amplitude, and we have the high temperature that resets the systemic auxin oscillation by impacting on auxin catabolism, and then this resetting basically is contextualized by the light intensity. So if this resetting hits a condition where light intensity is high, then actually the amplitude of the recovery will tell the system it's all fine, go ahead, develop lateral roots. But if the resetting hits the system when light intensity is low, then the system says, okay, it's not fine, there's a stress condition, stop lateral rooting. And more conceptually, I think what the story shows is that auxin oscillator function as a state variable, integrating systemic information into local root acclimation. So the auxin has been considered as a local regulator and also as a systemic one based on transport processes. But what we show here is the link to the circadian clock and its catabolic control is doing actually something very different. So it allows the system to sense the status, so light and temperature together, and informing a local decision for the PBS and for the routing. And with this, I'm already at the end of my talk. So I already acknowledged Chengzi. So this is the main driver of this project. He also directly supervised a bachelor student, so Jule, on this project, and he's currently co-supervising another PhD student, so Shasha that just joined the group. And we got great support by our collaborators, so Miguel, Andres, and Krishik were very important. And of course, without funding, we couldn't do what we are doing. And I think another shout out goes out to the entire group, because nobody works in isolation. And I think we have a fantastic group, and some of them are watching, I guess. And if you're interested in joining this fantastic group, so there are actually some position available right now. So we are lucky enough that in the frame of an ERC synergy grant, we just opened some PhD positions, and this one is available. So the deadline is 6th of February via a PhD school that is associated to the Max Planck, and very soon we will get another opening in the frame of a new excellence cluster that deals with forests, and so we can offer someone to work on Papla with us. So with this, I'm very happy to get your questions.
Thank you very much, Jorgen, for the really interesting work. Maybe a quick question from the chat. So by preventing persistent pre-branching sites, how does light prevent the lateral root formation?
So the idea is that light regulates the amplitude of auxin, and the system, if high temperature is not yet coming in, the system is that as long as there is a a kind of a threshold, and then we get lateral root development. But if we get below the threshold, then lateral root development or the auxin signaling in the PBS ceases out, and we do not get a lateral root development. So this is the idea that this is like a threshold that needs to be reached. At least that's our hypothesis here. And then in terms of high temperature, again, like this would reset the entire aspect. And then the resetting is very sensitive to the light intensity, because I showed that under normal condition, the higher light and the medium light had a very similar amount of lateral roots, but their high temperature response was very different, right? So meaning it provides really context in a quantitative way.
Thank you. Sorry, because we don't have more time to read the next question, but I'm happy that Jürgen will stick around, I guess, for the breaking room so that we can discuss later. So I would like to pass it
Jasmine,
to
could I
Thank
just
you.
could I just make a couple of announcements? So basically, thanks for that session. That was really great, really interesting. If people have any questions for the speakers, then please feel free to put them in the chat, and I'm sure the speakers will be happy to respond to them. And as Jasmine mentioned, there's breakout rooms in a second when we have coffee breaks. So if people want to ask questions directly, they can. One of the things, it was clear that there's quite a lot of disturbance in that last session from people not keeping their microphones muted. So please make sure that you keep your microphones muted so we don't disturb the speakers. The other thing that I've been made aware of is that not everyone can see what's happening in the chat. So if you can't and you want to ask a question, then please raise your hand at the end, and we'll try and make sure that we get to everyone if we can. But obviously we're running a bit late already. So we'll move on to the next session, which is the working group for roots and ecosystem function, which is led by Marie-Noëlle. So Marie-Noëlle, if you want to take over proceedings, that would be fantastic.
Yes, hi everyone. My name is Marie-Noëlle Tivierge, and I am a research scientist at Agriculture and Agri-Food Canada. I'm the leader of the Working Group 5, in which many plant and soil scientists work together about roots and ecosystem function. So I will be happy to speak with you at the coffee break if you have an interest in this working group. So today we have two distinguished speakers, and let me introduce the first one, Dr. Diego Abalos. And Diego, if you want to share your presentation during the time I introduce you. So Diego Abalos is a professor at Aarhus University, Denmark, whose research focuses on the nitrogen cycle in agricultural systems. His work addresses nitrogen use efficiency, nitrous oxide emissions, and mitigation strategies, integrating laboratory, greenhouse, and field experiments with modeling approaches to reduce the environmental impacts of nitrogen while sustaining agricultural productivity. He is widely published and actively engaged in international research collaborations on sustainable nitrogen management. His research group places particular emphasis on the role of plants in the nitrogen cycle, and today he will present selected examples of this work. So thank you, Diego, for being with us today.
Hello. Can you see my slides?
Yes, but it's not in presentation mode at the moment.
How about now?
No, not yet.
So Diego, I saw it in presentation mode, and then it's now flipped to the speaker mode. So maybe if you can flip it back.
Okay.
Yeah, that's good.
That's fine.
Is it okay? Yeah, it
Yes,
works now?
Yes.
Good. Okay. Thank you. Thanks, Marie-Noëlle, for the nice introduction. Thanks, Tim, for organizing this very interesting symposium. It's a pleasure to spend some time with all of you. And yeah, I have, I think, 20 minutes to talk about root traits to minimize nitrogen losses. How to split the talk in. So the first half would be about the general background on nitrogen cycling and the importance of nitrogen losses within nitrogen cycling, and how plants can conceptually modify these nitrogen losses. And then during the second half of the talk, we'll give examples of different experiments for the first two with grassland species, and then the last two with barley mutants. So since the application of fertilizers became widespread in the first half of the last century, the amount of nitrogen that has entered the Earth's soil has doubled. So tremendous increase in nitrogen inputs going into the soil. Around 50% of that nitrogen ends up in the plants and in the crops, and that's fantastic because that means that we have more food, more fuel, more fiber, higher availability of bioproducts. The problem is that the remaining 50% of this nitrogen can be lost from the agroecosystem in the form of ammonia volatilization. Ammonia volatilization, nitrate leaching, water bodies, and emissions of nitrous oxide. And these nitrogen losses are of tremendous environmental and economic concern because they cause climate change, the depletion of the ozone layer, eutrophication, economic loss, biodiversity loss, and harmful air pollution. So luckily, most governments across the world are now well aware of the importance of these nitrogen losses. And that's why, for example, in the EU and Denmark, we have the ambition to reduce nutrient losses by 50%, at least 50%, without decreasing soil fertility, reduce fertilizer use by at least 20% by 2030, and reduce greenhouse gas emissions by 50 to 70% by 2030. In my research, so within the field of nitrogen losses, N2O emissions are particularly important. And this is because these emissions, so N2O is a very powerful greenhouse gas. So one molecule of N2O has the global warming potential of 273 molecules of CO2. And N2O is also the single most important substance responsible for the depletion of the ozone layer. A very powerful greenhouse gas, very harmful gas that we need to mitigate. In my research, and in this field, there's a lot of things that we can explore because plants can modify several of the controlling factors of nitrogen cycling and nitrogen losses. For example, we know that some plants, legumes, can establish an association with bacteria in the soil, rhizobia, and that some of the nitrogen that the rhizobia fix and provide to the plant goes back to the soil. That changes nitrogen cycling. Of course, plants, different crop species and plant species in general differ massively in terms of the quantity and quality of their biomass, both above ground, of course, and in the root biomass. So, maize can have around 200 kilograms of nitrogen above ground, and barley, just to name two very common crops, can have half of that amount. Different, similar differences in below ground nitrogen that goes back to the soil once plants decompose, and that affects nitrogen cycling. Plants release this complex mixture of chemical compounds through the roots. That's called exudation, and specific compounds from certain plants can trigger mineralization of soil organic matter, and therefore they can increase the amount of organic nitrogen that becomes ammonium to be used by microorganisms and by plants themselves. This exudation can also change the microbial community in the rhizosphere. Some plants can increase, for example, the abundance of the nitrifying microorganisms in the rhizosphere, and that in turn increases the potential of a certain soil to generate N2O emissions. Plant nitrogen uptake is a very obvious and yet arguably the most powerful tool by which plants can modify nitrogen losses. So because plants are fierce competitors for soil mineral nitrogen, both ammonium and nitrate, and whoever wins that competition, microbes or plants, defines the amount of nitrogen losses in a given system. Nowadays, there is a very hot topic in soil biogeochemistry and nitrogen losses is the biological inhibition of nitrification. This is a biotic trait of some plants. So some plants can inhibit nitrification temporarily through the root exudates. It's only a few plants, but now there are big projects trying to, through breathing, increase the amount of BNI exudation, biological nitrification inhibitor exudation in wheat and barley. Some plants can also exudate a compound that blocks denitrification temporarily, but this is not so well understood, and there's only a few plants, but still, it's another root trait with strong potential to modify nitrogen cycling. There's also, all these are biochemical factors, but there's also an often overlooked physical factor of importance for nitrogen losses. It is just the root growth changes the entire soil structure. For example, thicker roots can act like a chimney for gaseous emissions to leave the soil. Through exudation, they change soil aggregates and also all the water retention and all the soil structural properties that modify nitrogen leaching and N2O emissions as well. So there's a physical factor by which root growth can also modify nitrogen cycling. And this last but not least, most vascular plants, 80% of vascular plants, establish an association with vascular mycorrhizal fungi, and this symbiosis has consequences for phosphorus acquisition by the plant, but also for nitrogen acquisition. And so it also has consequences for nitrogen cycling and nitrogen losses. So these are just some of the many mechanisms by which plants can control nitrogen cycling. So in agriculture, people tend to think that it's just to add nitrogen uptake, and that's it. That's not true at all. There's a lot of biotic interactions between plants and soil microorganisms. So that's sort of a general conceptual overview of this relationship between plants and traits and nitrogen losses. Let me show you examples of 4 experiments that we have conducted within this domain. So the first one was focused on these plant nutrient acquisition strategies. Ecologists have been using already for quite some time, for more than 20 years, this concept of the leaf economic spectrum. According to the leaf economic spectrum, plants can be categorized according to the way they interact with the soil for nutrient acquisition. On one side of this spectrum, we have acquisitive plants on the left of this slide, and those are characterized by having a high specific leaf area, low leaf dry matter content, and high nitrogen content. And these plants take nutrients fast from the soil, but they have a short lifespan. And conservative plants have the opposite traits. Plants are thought to be believed to be functionally coordinated above ground and below ground. So acquisitive plants have high specific root length, high nitrogen content, and low diameter, and conservative the opposite traits. We now know that this is not entirely true. It only tells part of the picture, because roots have this multi-dimensional nature. It's not only an economic spectrum, but they also can affect this nitrogen acquisition through another gradient. There's the acquisition gradient and there's the collaboration gradient. So by establishing a stronger association with mycorrhiza, for example. But again, here we focused on the leaf economic spectrum. Out of all these factors that I just described, 2 are arguably the most important from a quantitative perspective. And according to the leaf economic spectrum, acquisitive plants induce higher soil organic matter mineralization and have higher nitrogen uptake than conservative plants. These have been tested in several experiments in natural ecosystems, but the consequences of this for nitrogen losses have never been tested. So to see if there was something interesting going on, we formulated these 2 hypotheses. Acquisitive species reduce N2 emissions after a high nitrogen addition, because acquisitive species have higher plant nitrogen uptake, and that would reduce soil mineral nitrogen, soil microorganisms producing N2O. And the opposite will happen if there is no high nitrogen addition. So conservative species in this case will reduce N2O emissions, because in this case, mineralization of soil organic matter will be key, and conservative species induce lower soil organic matter mineralization. We tested the hypothesis in a greenhouse experiment using monocultures of 6 grasses grown under 3 nitrogen levels of 0, 100, and 200 kilograms of nitrogen per hectare, and with 5 replicates. These are the grasses. You cannot see the trait differences with the naked eye, but I can tell you there were. And this is how it looked, rather big experiment some time ago. We measured N2 emissions repeatedly over the duration of the experiment. We analyzed the main variables related to nitrate losses, above ground traits, the typical ones and below ground traits, specific root length, diameter, and loop density. So a focus on morphological architectural traits. Once we finished the experiment, the first thing that we did is to categorize the plants according to the leaf economic spectrum using a PCA with leaf dry matter content and a specific leaf area. And we could see that these two plant species were the acquisitive ones, these two the intermediate, these two the conservative. No need to remember the specific species. And these are the results for the 0N nitrogen level, but for the 100 nitrogen and for the 200 nitrogen, the results were exactly the same. So very clear differentiation of these species according to the leaf economic spectrum. The main finding of this experiment was that the amount of N2O released per unit of nitrogen uptake was 80% lower for the acquisitive species compared to the conservative species. And this was observed for the 3 levels of nitrogen addition: no nitrogen, 100 nitrogen, 200 kilograms of nitrogen per hectare. So part of the hypothesis was correct. Acquisitive will reduce N2 emissions with a high application rate, but the second part was not correct. So in this case, the acquisitives were still performing better even without nitrogen application. The effect of the intermediate species was interesting because it was similar to that of the conservative without nitrogen application and similar to that of the acquisitive with nitrogen application. So, and this is primarily because Lolin perenne was classified as intermediate, and it showed a lot of plasticity, especially in relation to the nitrogen content in response to the amount of nitrogen that we applied. So it was very plastic in the amount of nitrogen that can take according to the nitrogen regime. And then we conducted a structural equation modeling, and we found 2 traits, one above ground and one below ground, so specific leaf area and mainly root length density were very strongly associated with total plant biomass. In turn, plant biomass was associated with some soil properties. More important here is that increased biomass was associated with lower soil nitrate content, and then nitrate content was related to N2 emissions because nitrate is the main substrate for denitrification. So the conclusion here is that these 2 traits, and specifically root length density, is a very good predictor of how a plant is going to impact N2 emissions during their growth. So a very important trait to understand the climate change impact of certain crops. In the second experiment, here the focus was on mixtures, so not on specific plant components, but on plant mixtures, but it also had a root trait component. So we know that when we combine different plants, there's 2 possible outcomes. There's a negative interaction when plants compete with each other, and the outcome is that one of the plant eventually loses, so it disappears from the system, and that decreases biomass. But we know that luckily, very often we see positive interactions when we combine different species. This has been found most often in natural ecosystems. You can see here natural grasslands, these results. And this can happen due to complementarity when we combine, for example, this species here with deep roots with this other one with a more shallow rooted system. They will explore the soil very efficiently, taking nutrients from different soil layers, and that complementarity increases biomass production. And another mechanism is facilitation. In this case, is when one species benefits from another one. Grass legume mixtures is a typical example. The legume fixes nitrogen, it releases some of that nitrogen to the soil pool, and the grass is happy with it. And the legume is more neutral to the presence of the grass. Overall, increased biomass production with these combinations. As I said, this positive biodiversity productivity relationship was found in natural grasslands. It was also confirmed for forest ecosystems worldwide. And then recently, I would say this, a few weeks, not even months ago, we could also see that in fertilized grassland system, we can also see a positive effect of mixtures. So here, Marie-Noëlle and myself together. And this is nice work. It's a multi-site, 26 sites with the same experimental design. Led with this work, led is the legacy net, led by John Finn, Carsten Mallis, and Caroline Brockley. And we could see that the benefits of mixtures are almost universal. So across the 26 sites, mainly in the US, Canada, Europe, and China, many of the mixtures outperform the monocultures. But now the key goal is, okay, so how do we identify the optimum trait combinations to maximize this productivity while at the same time reducing nitrogen losses? So in order to start this work of identifying optimum trait combinations, we formulated these two hypotheses. Larger plant diversity reduces N2 emissions due to higher plant nitrogen uptake, so more complementarity with more species, more biomass, low nitrogen in the soil, lower N2 emissions, and then including an acquisitive species based on the results of the other experiment will be key to reduce nitrous oxide emissions. There was another greenhouse experiment with a very similar experimental design, but in this case we had monocultures and 2 and 4 species mixtures. We deliberately did not include a legume because, of course, legumes in terms of nitrogen cycling have a too powerful effect. So we wanted to keep it to this complementarity without including any nitrogen fixer. And then it was same measurements as in the previous one. So now I will not go into the details. Okay, so the hypothesis was that with increasing species richness, with more species, we would have higher biomass. That was a total failure, as you can see. It is not what we observed at all. And then the idea was that this increased biomass would translate into lower emissions with the highest level, but that, of course, did not happen at all either. So the first hypothesis was not correct. The higher species richness level had even higher emissions without nitrogen, for example. But we could see that there was something going on with the 2 species mixtures. So zooming in and seeing the results at for all of the treatments, we could see that without nitrogen application, the combination of Festuca and Fleum had the lowest emissions. With nitrogen application, the other 2 species mixtures, so Lolim Perenne and Pua tibialis, had the lowest N2 emissions. And what was the reason for the good results with these mixtures? Biomass production was important, so these combinations were productive, but definitely not the most important factor. So of course you can see here is these combinations had the highest root, or among the highest root biomass. So root biomass was important, but there were other factors at play. And a key one was that this is a combination of an acquisitive and a conservative species, and this is a combination of a conservative and an acquisitive species again. So that complementarity was important. These 2 species are commonly found in natural systems with low nitrogen levels. These 2 species are commonly used found in intensive systems where the nitrogen in the soil is higher. So combining species with contrasting acquisition strategies below ground and adapted to the specific nitrogen level is the key to reduce N2 emissions. So there's a strong potential of using mixtures, targeted mixtures, to reduce N2 emissions. The third experiment, the last two go a bit faster, they're a bit more simple. Here we were interested in a more, just one specific trait, root growth angle. So we wanted to see the effect of steep root growth angle on nitrate leaching and N2 emissions in spring barley. The hypothesis was that higher allocation of root biomass to deeper soil layers would be good for leaching, but bad for N2. This work is still not published. Was conducted by Marvin Eguizabal Garcia, a PhD student who successfully finished not long ago. And the plant material that we use is the Tilmore from the Tilmore population. You can see this is the Morex, the reference cultivar, and this is the mutant TN194 that with hypergravitropism, so the roots grow vertically. This is the second mutant, just to confirm that it was not to that specific mutation, but to the phenotype. We used another mutation also based on the Morex as the lead cultivar. And in this case, it was the experiment was conducted under control conditions in a climate chamber with 2 nitrogen rates, 7 weeks with the 2 mutants. Okay, so the first thing that we did is to confirm that the mutation was expressed, and indeed, this is the reference, and these are the 2 mutants. It's just an example, but we could see also surprisingly consistent effect, because all of the seeds that we tested had this mutation expressed, and these had strong consequences for nitrogen losses. So you can see TM194 had on average 64% higher emissions than the elite cultivar Morex, and the other mutant had on average 100% higher N2 emissions than the reference cultivar. We did not find an effect on nitrogen leaching, perhaps because the pods were only 25 centimeters long. They were not big enough to detect the differences in nitrogen leaching. And for the fourth experiment, in this case, the root rate of interest were root hairs. We wanted to see if having root hairs would have an impact on nitrogen losses. To speed up because I'm a bit over time. Again, the Tilmore population that had 3 mutants here: no root hair, short root hairs, and long root hairs. This team was part of the collaboration. And we selected the hairless mutant and the long hair mutants for the experiment, exact same setting as in the previous one. And we confirmed the phenotype, so the seeds that we had were hairless. And this really had a strong impact on nitrogen losses. So N2 emissions were more than 2 times higher for the hairless mutants compared with the reference Morex, and nitrogen leaching was 4 times higher for these hairless mutants. So very strong impact of this particular root rate on nitrogen losses. So summarizing the results of all of these results that I just been talking about, the strategic selection of plant species and traits can enhance nitrogen rotation in the plant and in the system and support climate change mitigation through reductions of N2 emissions. Targeted genetic approaches offer a powerful pathway to identify and enhance nitrogen-efficient root systems in modern crops. We can use it. And root and leaf functional traits should be central to crop and cultivar screening as we move towards systems in which the anthropogenic nitrogen inputs in the form of fertilizers need to be lower. So thank you very much. And that's it from me. I'll be happy to answer any question you may have.
Thanks a lot, Diego. A very interesting presentation. And in the chat, there are 2 interesting questions, but we're really running late now. So if you could respond to them in the chat, Diego, so everyone can see the response to those 2 questions. So thank you very much.
Sure.
Now let me introduce the second speaker, Dr. Vincent Poirier. And Vincent, if you could share your presentation while I introduce you. So Dr. Vincent Poirier is a professor at UCAT and director of the Agriculture and Agri-Food Research Institute at UCAT in Canada. He holds a bachelor's degree in agronomy and 2 master's degrees in soil and environment from Laval University, as well as a doctorate in renewable resources from McGill University in Montreal, Canada. He completed postdoctoral fellowships at Laval University in collaboration with the Center for Functional and Evolutionary Ecology at Montpellier, France. His work focuses on carbon storage in agroecosystems and the roles of roots in soil functioning. He is currently involved in several regional, national, and international research projects. So thank you for being there, Vincent, and you can start when you are ready.
Thank you very much. So do you hear me well?
Yes.
Okay, great, because I don't see you anymore now, so I just want to make sure that everything is okay. You see my presentation as well in presentation, not in presentation,
yes,
but in
we
full
do.
screen mode. Okay, great. So thanks a lot for this invitation. I'm really happy to be here, really excited as well. So it's a great initiative, and yeah, so let's jump into it. First, my plan for you today. I'm gonna first talk about the past, a bit of history and some definitions. Then jump into the what I call the present, the main mechanisms and characteristics of interests. And third, the future. We're gonna end this talk with a discussion about avenues to explore for further investigation. So to begin, let's go back a couple of years ago already, 14 years ago. So for me, it all began in Porquerolles, South France. So as you can see, this is a really beautiful area. Porquerolles is an island on the coast of South France, and there was a summer school there on plant functional trait that was organized by Alison Munson, former postdoc supervisor. So I'm here, and Alison is right there, and there are many nice people from plant functional ecology. There's Eric Garnier over there, Marie-Laure Navas, Bill Shipley, Sandra Lavorel. So for me, this experience was really eye-opening because I discovered a new world. And it was somehow a revelation because I found out that there are two parallel universes, at least two parallel universes. There was this whole world of functional ecology that I just discovered on this Porquerolles Island, where we speak about plant characteristics, growth strategies, and leaf and root traits. And there's another world, which is the world where I come from, the world of soil science. I was trained as a soil scientist, and where we talk a lot about biophysical, chemical properties of soils, pedological processes regarding soil formation, and soil organic matter, which is a living aspect of the soil. And despite the importance of roots into the soil organic matter cycle, these two worlds communicate little with each other. So that was for me the revelation. I said to myself back then, so I have to bridge this gap, or at least attempt to. So before we move on further, I just want to clarify what is the root functional trait. So maybe it's going to be redundant for some of you, but so a trait is any morphological, physiological, or phenological characteristic of the root system, in that case, that can be measured at the individual level. And that definition is not from me, it's from this nice paper by Cyril Vial and colleagues. And the idea over there is that this approach seeks to overcome the effect of species in order to link the measured traits to functions within ecosystems. And regarding soil organic matter, well, soil organic matter is a continuum, okay? This is the important keyword here. A continuum of organic residues and compounds that are at more or less advanced stage of decomposition and more or less associated with the mineral matrix of the soil. This quite interesting and really important paper came out 20 years ago, and this was somehow the foundation for what we know now as the main mechanisms controlling the stabilization of organic matter in soils. So there are three main mechanisms. It's maybe it's a bit more complicated than that, but overall we can resume it to three main mechanisms that control the stability of organic matter. The first one is selective preservation, which is highly linked to the intrinsic biochemical recalcitrance of the organic molecules. There's physical inaccessibility / occlusion, and referring here to the aggregation of soil particles. So I'm mostly going to focus on that. And the organomineral and organometallic interactions that stabilize the organic carbon and organic matter in the soil. So after reviewing more than 200 articles, spending several hours of reading and working for many months, our efforts have paid off, and we published this this paper of which I'm really proud, and almost 8 years ago. So, but it seems that it is still relevant, because that's why I'm here for today, to talk about it after the invitation of Marie-Noël. So the idea in this paper is to try to bridge this gap that there was in the literature between the world of root functional ecology and soil organic matter. So we identified 18 root traits. There are many more traits than that, of course, but what we could find in the literature was mainly 18 root traits that we split in 5 categories that are shown to influence the soil organic matter stabilization in soil horizons. So the figure that you see is in French. I'm sorry, it was a bit complicated for me to translate it because it's there's a lot of details, but très chimique means chemical traits, très architectural means architectural traits, très physiologique means physiological traits. Here we have symbiotic traits and morphological traits here. So for chemical traits, we found that we had some nice information in the literature regarding the impact of soluble compounds, hemicellulose, lignin, phenol, calcium, aluminum, carbon, nitrogen, and suberin concentrations. For architectural traits, we found that root depth distribution, root branching intensity, root land density were the main traits that we could relate to soil organic matter stabilization. I would add root mass density, but surprisingly, there was not that much information in the literature. For physiological traits, we found links with rhizodepositions and formation of proteid roots or cluster roots. Symbiotic association, of course, mycorrhizae and nitrogen fixing bacteria. And for morphological traits, the average diameter, root average diameter, and root specific length, or specific root length. And I would add also the distribution of length in the different size diameter. For example, the proportion of the total length that attributed to very fine roots, for example. So first thing to say is that the influence of roots on soil organic matter varies with soil depth. So here we have a little picture of some unpublished stuff. So soil depth is on the y-axis, and percentage depth distribution is on the x-axis. So what you see here, for example, in the upper layer is that 60% of the root biomass that can be found in the upper meter of soil can be found in this layer, while we have about 40% of soil organic carbon. So superficial horizons contribute more to the total root biomass found in the soil profile, while deep soil horizons contribute more to the total amount of carbon found in the soil profile. So there's a disconnection that's happening within the soil profile. So the root-derived carbon that is entering the deeper soil layer is probably retained more efficiently than it is in upper soil layer. So that was also a driver that led me to question myself. So why is that the case? Which specific root traits are more likely to impact stabilization along the soil profile? So I don't have all the questions to these, all the answers to these questions, of course, but it's an ongoing process. So I'm going to guide you through the 3 main mechanisms. There's a lot more information in the paper than I'm going to show you in the time we have. But first, let's start with recalcitrance. So this is an extract from the table that's in the paper. So what we should retain from this is that regarding morphology, morphological traits, there's very variable effects in the literature, so it's not always clear. However, regarding symbiotic and chemistry, we found marked effects that are clearer. And maybe one of the most important thing that came out of this work is that there are two different conceptual approaches that exist. So one is what I call the root-centered approach. So in this vision, labile roots that are rich in nitrogen and soluble compounds, that are low in lignin and structural compounds, they decompose rapidly and it ends up to a low stabilization of soil organic matter. And in the opposite, these roots that are really recalcitrant, rich in lignin, they decompose slowly. And in this vision, they lead to a high level of soil organic matter stabilization. But when we look at the soil-centered approach, what the literature tells us is that this is some kind of the opposite. So the labile roots that are rich in nitrogen and soluble compounds, they get decomposed more efficiently by the microorganisms. And they produce labile compounds, and then these compounds, they get stabilized onto the mineral surfaces, and it leads to a higher stabilization of soil organic matter. And the counterpart is that the recalcitrant roots, they are rich in lignin, and they get decomposed much less efficiently. And the microbes, they spend much more energy to get what they need. So there are a significant fluxes, CO2 fluxes, and then this leads to lower level of soil organic matter stabilization. The second mechanism is regarding occlusion. What came out of this is that root traits can influence certain aggregate size classes differently. So it depends on what you're looking. And that a combination of traits sometimes better explain the impact of roots. So an example here, on this figure, what you see on the y-axis here is the amount of large macro aggregates, so the aggregates that are between 2 and 1 millimeter. It's in the rhizosphere. So an abundant biomass, you have root mass density here. So if you move on further towards this direction and further to in this direction, so what we can find out is that an abundant root mass that is low in lignin promotes the stabilization of large macro aggregates. And the high density of fine roots promotes the stabilization of very large macro aggregates. So in that case, we are at the millimetric level, 6 to 2 millimeters. And if you look at this graph here, we move on to soil organic carbon that is retained in the form of particulate organic matter. We see it's highly correlated. We see it's the same soil by the way. So we have a strong relationship here with root length density. So the increased root length density promotes the stabilization of carbon in the form of particulate organic matter. And the last mechanisms is that through interactions with minerals and metals. So here again, the extract from the paper. And this mechanism is dominant in deeper soil horizons, because in deeper soil horizons, the mineral surfaces are less saturated with organic compounds, so there's more reactivity. So there was a positive impact of the most traits studied, but rooting depth remains the most important trait. It's kind of logical, because roots must reach the subsoil for root-derived carbon or organic matter to get stabilized down there. And here in this slide, what we see is that very fine roots that are rich in soluble compound, they stabilize more mineral-associated organic carbon in the rhizosphere. So there's specific root length here and root-soluble compound here. And then there's an increase when these two traits interact. So moving on towards the end of the talk, the future now, avenues to explore for further investigations. So first for me is the impact of living roots versus dead roots. This is two things, you know, while the roots is the plant is growing and when the roots are decomposed. And in parallel to that, it's the impact of stabilization versus destabilization, because roots also have a destabilization effect. So what you see now is a sort of a mind map that I created a couple of years ago, and I go back to this slide once in a while. So the dotted points represent the impact of the dead root, the full lines represent the impact of the living roots, and the blue lines represent the impact of stabilization, and the red lines represent the impact of destabilization. So eventually, I would like to maybe put some coefficients in there and try to relate this to root traits and soil type. But all that to say that there's many things to explore for further investigation. And another avenue, really important, is that the influence of root traits may depend on soil organic matter stabilization capacity. So I don't know if you're familiar with this MEMS model from Francesca Cotrufo that came out in Global Change Biology. But basically what the model says is that there's a relationship between the microbial substrate use efficiency and the matrix stabilization capacity of the soil and the formation rate of soil organic matter. So when you're in a situation where you have high microbial substrate use efficiency, labile compounds, labile root fragments, rhizal deposits, this stuff gets easily processed by microbes, and it can lead to the formation of stable soil organic matter. But in the last years, I worked with Raoul Ruiz, Grégoire Fréchet, and colleagues from South France, and we found something that was counterintuitive. Here, what you see in the graph is an increase in the mineral-associated organic matter, the carbon in that fraction, with the level of lignin in plant tissues. So this somehow as I said, it's a little bit counterintuitive. And I have to say that this was done in soil where it was, I think it's 95% sand, really coarse texture soil, really close to its maximum capacity for organic matter retention. So, so the soil type that we are working in is an important driver. And all that I've done in the past years, I was mostly working with clay and heavy clay soils because this is the type of soil where I came from. But that work with Grégoire and Raoul made me believe that the impact of traits may be highly modulated by the soil type. So to conclude, two characteristics stood out and positively influenced the three mechanisms. I haven't spoke about it in this talk because I didn't have much time, but you can find information in the article. Mycorrhizal association and root sovereign concentration are two important traits. A lot of questions regarding conventional morphological traits. They show variable response. I wonder why is there such a lack of precision, and maybe there's a need for a refinement or analysis at the level of root orders or different size classes. It gets a bit complicated to go into very much details, but maybe that's a way out to get more clarity. I think that studying the stabilization and destabilization balance of the soil organic matter on surface and subsurface soil resins using root traits is a promising approach. And at last, the influence of root traits may vary depending on soil type, texture, mineralogy, initial organic matter content. So yeah, we have to dig deeper on this matter. And I think that I am done now. Thank you.
Thank you so much. Thank you so much, Vincent, for the presentation. And we won't have questions, but I'm sure you can respond to them in the chat if some people have questions to add. So thank you very much for this, this session.
Okay, so thanks for the 2 sessions this morning. That's been really good. Well, this morning in Europe anyway. It's been really interesting. We're going to maintain the breaks. So we're running a couple of minutes late, but I think if Will's okay, he's going to be running the next session. Then we'll come back in 10 minutes or so. If you want the opportunity to speak to the working group leaders who are in the meeting at the moment, then there is an opportunity to go to a breakout room to do that. So if you go to the bottom of your screen, you'll see a breakout sessions button. If you click on that, you'll see that all the working groups are there. If you have a particular interest in any particular working group, then please join that working group breakout session, and hopefully the working group leaders who are here will go to those as well, and you'll have a chance to have a chat about the work that's happening in those working groups. But otherwise, take a break, 10 minutes, we'll come back and we'll start the next session at 12:05 UTC. So see you in a few minutes. So here we go.
Perfect. Thanks, Tim.
Over to you, Will. Cheers.
Joe, are you back from your tea break? Are you around?
I'm here.
Beautiful. Okay, so hi everyone, I'm Will King. I'm the working group lead for Rhizosphere and Signaling. It is my absolute great pleasure to introduce Joe Edwards today, who's going to be giving a really cool talk about switchgrass and root microbiomes. Joe did his PhD at the University of California, Davis campus, before picking up a USDA NIFA fellowship at the University of Texas with Thomas Junger. And now he is an assistant professor at Texas A&M University. So looking forward to this talk, Joe. Are you able to share your screen?
Yes, can you see it?
Yes, that looks beautiful, and we can hear you. Go for it.
Okay.
Cheers.
Okay, great. Thank you. Actually, let's see. Oh, let me try one more time because I clicked on something and it went away. Okay, that's not good. Oh,
So
is that looking weird?
I know that looks fine for us and for me, and I can hear you as well, and I can see your normal presenter view, which is good.
Okay, great. It's looking very weird for me though. So but I'll try to do this. Okay, so yeah, as Will mentioned, so I've started my lab almost 2 years ago now at Texas A&M, and we've been really taking our time looking at switchgrass microbiomes or different microbiomes of different grasses. So we study plant microbiomes. So that is a complete community of microorganisms that colonize a certain environment. We focus on roots primarily, and that's because we think these microbes are important to the plants. So, for example, they mineralize nutrients that otherwise the plant would not be able to absorb. They heavily colonize the plants, so they just take up space, which means they can outcompete pathogens. They boost immunity, they boost growth through various mechanisms. So we think these microbes are important. And so for us, we're interested in how these complex communities, which can sometimes contain tens of thousands of different species and strains of bacteria, come together, and in particular, how the plant influences this process. So, so I can't tell if that actually progressed the slide or not. I'm sorry,
No,
I'm
it
having.
seems to be stuck on the first slide.
How about now?
Yep, that moved. Yep.
Okay, perfect. Okay, so in my lab, we study various different systems. One system that we study is switchgrass. And so this started when I was a postdoctoral fellow at the University of Texas. So switchgrass is a interesting plant species to study microbiomes with. It's a native plant to North America, so it grows from northern Mexico through the United States into Canada. It's a very large plant. It grows in marginal soils with very little nutritional input and water. So it's really been championed by the U.S. Department of Energy as a biofuel feedstock. And what I really like about studying it is it's a wild plant. It's undomesticated, which means it hasn't gone through these genetic bottlenecks, that's happened through breeding with other plant systems. So it gives us a nice kind of natural plant that we can look at the ecology and the evolution and how that interacts with different microbes. So when I was a postdoc, I got involved in this project. We were putting together a natural diversity population of switchgrass. What that means is we were driving around the United States, digging up switchgrass along the road. So we would do this, our collaborators would do this, and then we'd send it back to the University of Texas, where we would take a leaf sample, grind it up, extract DNA, send that out for resequencing so we can call SNPs and variants against the reference genome of switchgrass. And then what we would do is we take each one of these plants, we clonally propagate it, and we plant them in different gardens, common gardens around the United States. So we have around 600 to 800 different accessions in each garden, and we have around 10 gardens, but I'm going to talk about 3 today. So we have a garden in Texas, a garden in Missouri, and a garden in Michigan. This is an example of one of our gardens. I believe this is in Michigan. So you can see kind of the phenotypic diversity that exists in this population. So we have waxy plants and large plants contrasted against small plants. So there's lots of phenotypic and genetic diversity in this population. And I was really interested in asking about the microbes that colonize these plants, particularly in the roots, and just putting together kind of a large survey effort of understanding who's there and the different factors that drive differences in their community compositions, including, do these communities look different across these different field sites? Do they look different across different genotypes? And ultimately, can we use kind of the genetics of the host to try to map places in the host genome that impact the abundance of different microbes? So this is first bit of data from this. We published this a few years ago. So this is our 3 field sites that I'm going to talk about today. So we have our Texas field site, Missouri, and Michigan. And so what we did was we went to each plant, we dug up some roots, we extracted DNA and did our kind of standard amplicon sequencing pipeline for 16S sequencing to analyze these communities. So this is kind of what we see after we do this. We see 3 large clouds of points. So each one of these dots represents a microbiome from a sample from a plant. And what we can see is that primarily these microbiomes cluster by field location. So all of these samples came from the different genotypes in our Texas site. This is our Missouri site and our Michigan site. So this is not surprising that we see that the environment, the field site, is the largest driver of this community composition. In fact, this is actually a little bit comforting to see. This is, for us, a kind of internal positive control. And it's also not surprising because these field sites are very different in terms of soil texture. So our Austin site is a heavy clay, our Texas site is a heavy clay, and then our Missouri and Michigan site are more agricultural sites. So loamy texture. These are also agricultural sites here, while this is in the middle of a city. So even though these field sites have very different microbiomes overall, when we're looking at the roots, we can still identify these 92 microbes that we always see on the plants, no matter whether they're in Texas or in Michigan or they're across all the genotypes. So these 92 microbes we call the core microbiome. They're always there, and we're very interested in what they do and how the plant influences them. One benefit of this experimental design is the population that we were able to use. So we were able to use a technique called GWAS, genome-wide association studies, to try to find loci in the genome that impact different microbes. So here we're using the abundance of different microbes that we're detecting as our trait. And so for our core microbiome, we're able to find significant GWAS peaks for about 2/3 of them. So 62 of our 92 core microbes were able to find a significant peak. I've pulled out a few here just to show examples. So this is kind of a graphical representation of the host genome. So the different chromosomes and markers are along the x-axis. The strength of the association is along the y-axis, so we can find regions here. So for this Titanophagia, we find a peak near this early nodulin-like 18 gene. This free-living nitrogen-fixing bacterium, we find a peak near the cysteine-rich receptor-like kinase, so potentially plant immunity related. We find peaks near root hair genes, and we find this really nice peak near this plant hormone receptor, this abscisic acid receptor, pyrobactin resistance 1 like 5. So what this is telling us is that the microbiome and how the plant controls the microbiome, this is what we would consider a complex trait. There's lots of different genes and pathways that impact many different microbes. There's no one master regulator for this. So the data that I just showed, it actually came from one time point in the season. So it came at this mid-season time point. Switchgrass is a perennial plant, meaning that it goes dormant in the winter and comes back in the spring. So that experiment we just collected at mid-season, but we know that plant microbiomes are dynamic, that they change over the season, particularly in annual plants. So there's a large turnover in the community that exists on these roots from the seedling stage into the reproductive stage. And since we know that these change over the season, we wanted to know how our switchgrass microbiome is changing across these different phenological stages of the plant. So once a plant breaks out from dormancy, we call that the green up stage in the spring, into mid-season and into the senescence stage where they're remobilizing their nutrients below ground. So in each of our field sites, we again went to each one of our plants and collected roots at these three different time points. And what we found was a little bit surprising. So here are samples. This is just one site. Here are samples taken from our green up stage. This is the mid-season stage and the senescence stage. There are not really huge differences in the microbiomes of these plants between these different stages. And when we look at our core microbiome, those 92 microbes that are always there, so this is a plot of those. Each one of these dots along the x-axis is a different microbe in our core microbiome. The color tells you what type of microbe they are. And then our three field sites are along this y-axis. I don't have them labeled. But what it means is if there's a point here, we're still detecting that microbe as belonging to the core microbiome using our cutoff criteria for that. So what this tells us is that the switchgrass microbiome is stable across these, relatively stable across these three time points. This core microbiome that we detected in the season previous is stable across these three time points and across different years. So what we think is happening here is because switchgrass is a perennial plant, there's likely this phase that they go through to assemble their microbiome, but after that point, it's relatively stable for the rest of the plant's life. So this is one interpretation that we have of this data. Usually this is part of a larger talk, so I have a little summary so far as we go on. So what we know is that the environment that you're growing the plant in, that's the largest determinant of which microbes are going to be there. We can still find this core microbiome, and that we know that this core microbiome is influenced or under some level of control by the host plant, and we can find different places in the genome that appear to control or be associated with the assembly of the core microbiome. So now we're really interested in actually looking at very early stages of seedling stages into this first year, because we think this is where the plant may have actually the most control over its microbiome, and that may determine the trajectory of how that plant grows for the rest of its life. So again, we know a lot about this, but we know very little about this stage in our perennial plant systems. So I described a big survey effort of trying to figure out who's there, how abundant they are, who the most abundant microbes are on these plants. Of course, we want to know what these microbes are doing for the plants. And our way of doing that is by trying to culture as many of these microbes as we can get. And our method for doing that has not been the traditional kind of plating technique and picking colonies and streaking them out, but we've actually got our hands on this instrument that's called a prospector. And what I love about it is it's a black box. So, but we know how it works. So the way that this instrument works is you take your sample, you dilute it down to a certain degree, and then it has this microscope slide. They call it an array. It's the size of a microscope slide. And on the slide, there's these etched wells that contain, there's about 6,000 wells that contain some nanoliters of media. And you wash your sample over this array, and every once in a while, a single bacterial cell will land in an array, and it will grow and divide. And this instrument has a way of detecting that and picking it, transferring it to a larger, like a 96 well plate that we can culture these up even further. So what this allows us to do is kind of automate the process, and it's very quick. So we've been able so far to culture over, I think at this point, it's about 50,000 different strains of bacteria. We've run into the issue of how do we figure out which microbes we've cultured. And so we started Sanger sequencing, but we decided that was actually a little bit expensive. It's about the same price just to sequence genomes now for bacteria. So we're moving to pretty much sequencing all the genomes for this isolate collection that we have. So far, we've sequenced over 3,000 of the isolates using Illumina. So we have draft genomes for about 3,000 different strains here. In our collection, we have about 500 unique bacterial species. And from here, we're hitting the major plant phyla or microbial phyla that are associated with plants. So this has been a nice resource for us to play with. There's a whole other seminar here to talk about these genomes and everything that's contained within these microbes. It's really fascinating, but we won't go there today. Importantly, for our isolate collection, we've been able to capture quite a bit of our core microbiome. So we have 92 strains of microbes that belong to the core microbiome that we've detected through our survey efforts, and we've been able to culture around 34 of them. So what I'm showing here is our core microbiome that we detect out in the field. The abundance of each microbe is along the y-axis here. And for microbes that we have a representative isolate for, I've put a red arrow underneath them. So we have quite a bit of these core microbes in our culture collection and a diversity of them. They don't all belong to one phylum or a different phylum. So we have a diversity of these core microbes. And importantly, I'm going to get back to this in the latter part of the talk, but we've been able to capture the most abundant microbe that we always see on these plant roots, our switchgrass roots. And so, like many other groups with isolate collections, we've been interested in putting these into synthetic communities, so very controllable communities. And the way that works is we culture each one of these microbes separately. We pool them together in a community that we can control, and then we inoculate plants in these micro boxes that have a little filter for gas exchange, but don't let other microbes into the system. And so we're playing around with the system quite a bit. We're doing some fun ecological experiments where we're playing around with the nutrients in the media or the conditions that the plants are growing in. We're taking, for example, we're changing the order of arrival for different microbes. So we'll put a community together and then add some different microbe to it later to see how that perturbs the microbiome. We're removing microbes from the community and asking how that impacts the assembly of different microbes onto these plants. And we're actually playing around a lot with different hosts. So if we take this switchgrass microbiome, we add it to a different host, what happens? So I'll describe this experiment quickly. So we wanted to know, switchgrass is kind of hard. It's a hard plant to work with. It's a large plant. It's outcrossing. It's not a great lab model to work with. So we have a relative of switchgrass that's called Panicum hollii. It has great genomic resources. It's smaller in stature, a lot easier to work with. And what we were hoping with this experiment is that we could take this lab model of Panicum hollii and have it assemble the microbiome in the same way that switchgrass does. So we were testing that. And what we found out is that if we change our host species, it really changes how this core microbiome assembles on that plant. So over here, we have our 34 core microbes. We've pulled them together into a community. We've added them to these different plants. These are our switchgrass plants. We have a couple varieties over here. This is a coastal ecotype of this Panicum hollii and an inland ecotype. And we can see that they assemble this core microbiome very differently. So switchgrass over here, coastal Panicum hollii. And so we wanted to know what exactly are the differences between these switchgrass plants and this P. hollii, which microbes are driving those differences. And what we found is that there's this, our most abundant field microbe is also, it's completely taking over the community with Panicum hollii. So it's making up about 90% of the microbiome or more when we add it to Panicum hollii. Whereas in switchgrass, it's still very abundant, but it's somewhere around 60 to 45% of the community. And we also see kind of the opposite trend where there are some microbes which completely go extinct when you add them to Panicum hollii, but they remain in relatively higher abundance on switchgrass. So the assembly of this core synthetic community, it really depends on the host species. So given that this microbe is the most abundant microbe out in the field, and it seems to be taking over this community, it seems interesting, and we want to know what this microbe is doing. So we've been adding this microbe to the system and testing different things. One thing that we found is when we add this most abundant microbe, so we call it RCP500, the genus and species is Rosiatiles chitinivorians. I don't think many folks in the audience who have looked at plant microbiomes even know about this microbe. It's hard to find publications about it. But when we add it to our switchgrass plants, we see that it boosts growth. So this is mock inoculated plants. These are plants inoculated with this strain of microbe. It boosts above ground weight, and the same for Panicum hollii. We took we made a large effort to develop a pathosystem for switchgrass, and what we found is when we found this fungus that will, if you add it to a leaf, it makes a nice large lesion. So this bipolaris fungus makes this lesion on the leaf. If you add this bacterium into the soil, we can see that it significantly decreases the size and severity of the bipolaris infection. And this is true across switchgrass and Panicum hollii. And so we published a paper recently showing how, if you're interested, I won't go into it, but how jasmonic acid contributes to this and how this is a relatively ubiquitous microbe across different grass systems. But for this symposium, what about what's happening below ground?
Everybody try their eggs.
So, for root,
oh my God, these are so good.
Sorry, would you
I mean,
mind?
it tastes like any other ice
Would you
cream.
mind
Luis,
muting quickly?
can you please mute your
I
microphone?
made them.
Luis,
Have you? Of course it's good. It's good because I made them.
Okay.
This is not good.
Oops. Okay, so for our root systems, so typically for roots, when you grow them on agar dishes, and you grow them vertically, the roots will grow straight down. So this is our switchgrass plants growing on a petri dish. Roots are, growing down. When we add this microbe to the root tip, what we find is that these roots kind of they look like they're losing this gravitropism a bit. They start to wave around quite a bit, and they start forming these lateral roots in kind of weird spots. So this is a zoom in on this section. You can see even at the root tip, there's a lateral root emerging. And another oddity here is that the root hairs also become quite weird. So they start to secrete, they're in weird spots, and they start to secrete something from the root hairs. And we found this is also the case in Arabidopsis. So this is in our switchgrass. We've also started to look in Arabidopsis. We see again a lot of ectopic lateral roots when we add this microbe to the mix. And I think you can see that the rosettes are much larger for these plants than untreated plants. So a postdoc in my lab really wanted to get some experience using scanning electron microscopy, and we thought this might be a fun use for that. So Dr. Solon started looking at these roots. So this is a azenic root, so no microbe present. So you can see nice straight cell files, intact root cap. When we add this microbe to the root tip and it begins to grow a little bit more, we see that it's a little bit misshapen. We get a lot of these root hairs at the inoculation site, and that this root cap seems to be quite different than the control plants. When we look at where we inoculated, we can actually see the microbial cells at the inoculation point, but that's not where they really colonize the heaviest. When we're looking at the root tip, we see that these root tips are swarming with the microbe. So this microbe really seems to like the root tip. We did another experiment to ask, is this perhaps a metabolite that is produced by the microbe that is causing this phenotype, or does the microbe require direct contact with the plant? So Peijing, he did this experiment where he took a membrane that will allow for metabolite exchange, but won't allow for entire cells to fit through. And he would grow this plant on top of the membrane and either put the microbe underneath the membrane or on top of the membrane so that it could directly contact the plant. So these are root traces of, so no inoculation of the microbe. So this is what our roots would look like. When we allow the microbe to directly interact with the roots, we see the beginning, the emergence of the phenotype, which starts out as a hook. And then if we stick the microbe underneath the membrane, we don't get any phenotype. So what this tells us is that the microbe requires direct contact in order to form this phenotype. So we're trying to figure out currently, so this is new data that has not been analyzed to its fullest extent, but I'm trying to talk about unpublished data here. So we've been trying to figure out exactly what's happening with this microbe, and we thought, well, maybe we could try RNA-seq, although I think this usually opens up more questions than answers. So we tried an experiment where we mock inoculated plants at 3 days and 6 days, or we treated them with different microbes that we've isolated from our switchgrass plants as well. So we have a Pseudomonas fluorescens. These are root traces. You can see that they don't have a severe root phenotype. This is a closely related microbe to Rosy-Tieles chitinivores. So in this Commomonaceae family, you can see that we get a little bit of the phenotype starting to emerge with this relative, but not quite as severe as our major culprit here. And so when we do RNA-seq, we can see that we have a ramping up across these 3 different treatments. So Pseudomonas fluorescens, we only have the order of hundreds of differentially expressed genes, whereas we have thousands of differentially expressed genes for this RCP500. One area that I've started to get a little bit interested in is plant immunity. And so are the roots short and are they, do we see this phenotype because they're having a severe immune response? So we started to play around with different treatments. So this is how you look at pattern triggered immunity. Typically in plants, you look for a ROS burst over time. So you add an elicitor like FLAG22 and you get a ROS burst. We added our microbe, P500, to these plants to see if we get a ROS burst. And we did a treatment where we actually heat killed the microbe. So we heated it up so that they're not active anymore, or we left them alive. And we see we get a much larger ROS burst when we kill the microbe. And what we think this is that the microbe actually has some capacity of muting the plant's immune response. And we see that this is not always the case. So different microbes show much larger response ROS bursts when they're alive. So we think that it's not necessarily a PTI response, and that's why we see a shortened root. We compared this to FLAG22 elicited upregulated genes, and we see that there's some overlap, but it's relatively minor. So we don't think that this is necessarily a PTI response that is causing this root phenotype. But we know that because the phenotype of this root, that auxin must be involved somehow. Since we have the genome of this microbe, many soil microbes actually have the capacity of biosynthesizing auxin. This microbe does not have any canonical auxin biosynthesis pathways. And we've also tested this in vitro. It doesn't seem to make any auxin by itself. And so when we look at auxin-related genes in our RNA-seq, what we see is that, so here are the different treatments. So 3 days after inoculation, 6 days after inoculation, gray is control. These are different microbes. And then this is our favorite microbe, P500. What we see is that only with P500 do we get this upregulation of these auxin biosynthetic genes, yucca5 and yucca6. So this appears to be treatment with this microbe upregulates auxin biosynthesis in the host. We see a lot of auxin signaling genes that are only upregulated in response to P500, but not the other microbes that we've isolated from these plants, including this 10 protein, which redistributes auxin in roots. So I'm going to wrap up here, and I hope you've seen kind of our workflow for how we are trying to figure out how these microbes are working with plants. So we go from this very large survey data where the plants have told us which are the most important microbes for them. We've cultured them so that we can start to play with them and under different scenarios and ask, how are they important for the plants using synthetic communities? And we're narrowing in on certain microbes that we think actually may have some benefit or to the host plant, and we're trying to figure out the mechanisms for how they work. So I'm going to wrap up here and say thank you to everybody for listening to me ramble on, and give thanks to the folks in my lab that are working on this and our major collaborators who are doing lots of the plant sequencing for us and our funding sources. So thank you very much.
Perfect. Awesome. Thanks, Joe. Great talk. We're a bit pushed for time, so we won't take questions, but you have something like 9 questions in the chat, which if you have a chance, could you just go through, maybe answer them yourselves? That's a fantastic talk again. Really cool data. It's amazing. The manipulation of RCP 500 on these roots is really, really cool. But thank you again for giving a talk. Okay, so next up, second speaker in our little session here is Catherine Whipple. So Catherine, it's my great pleasure to introduce her. She did her PhD in Germany in plant physiology, and then jumped over and did a couple of postdocs, one at Stanford University looking at root nodules, and the other one at the Max Planck Institute in Cologne. Now, Catherine is an assistant professor at the University of Amsterdam, and she's going to tell us some cool stuff about functional plasticity and root microbiomes.
Yeah, thanks. Thanks a lot, Will, for the introduction. I think I have trouble sharing the screen. Let's see. I think I have to allow it to share the screen. Okay. That's great. What can you see my screen?
I can see myself, which is also great, but probably the slides would be nicer.
No, this is okay. This is not nice. But it says I'm sharing. Okay, wait. Maybe then I have to just.
If you put your PowerPoint up,
yes.
yeah. Okay.
And do you now see it in presentation mode?
I see that fine, and I can hear you, and that looks good to me. Yeah.
That is amazing. Thank you so much, Will. Yeah, thanks a lot for the invitation to present my work here today. I'm very excited, very honored. I will talk about plant microbiomes, and I'm actually quite happy, and it's quite handy that Joe gave a talk before me because I can cross-reference quite a bit. There are quite some parallels in the research, which is great, I think. So Joe already introduced that there are beneficial functions of the microbiome to plants, and I want to just now illustrate with this figure here how the shaping of the root microbiome actually happens. And this is really just a brief overview to show that we have a lot of signaling going on underground between the roots and the microbes that are living in the soil. We have reviewed this last year. And I just want to highlight that we have, of course, primary metabolites here, sugars and amino acids that microbes can use to grow on them. We also have antimicrobial compounds. Benzosasanoids and coumarins have been extensively studied the last few years to show that they are involved in microbiome shaping. And we have something that we termed microbiota recruitment signals, and that examples for that could be strigolactones and flavonoids. And the idea is that these signals are there in lower concentrations, so the microbes would necessarily not use them as carbon sources, but they do affect the functioning and the actions of microbes. And flavonoids, for example, are a signal for beneficial rhizobia to interact with legumes. And now, if we look a little bit into these microbiomes, for example, on different plant compartments, and even if we just focus on the root here, then we see that we do also get different microbiomes in the rhizosphere, in the rhizoplanes, on the surface of the roots, and in the interior of the root. And we have very diverse communities of microbes here. And what I want to point out here with this slide is that of course, we have interactions, specific interaction types between the host and the microbes, but we also have interactions within the microbiome. And I just want to point out that, yeah, we have mutualism, which is a classic, mandatory interaction between two partners. We do have commensalism, and I want to mention this because we often refer to the microbiota members as commensals because they benefit from living with the plant, but we don't know for sure if the plant is benefiting from these microbes. And we have antagonism and also competition for resources within the microbiome, and I will get back to that in a second. But essentially, we also heard in the earlier talk that there is a problem with fertilizer and nitrogen input in the soils. And basically, to make use of microbiomes as alternatives for pesticides and fertilizers, we want to understand how these microbiomes work together with the plant. And for this, we need functional and mechanistic studies. And then again, we already heard about synthetic communities, and I just want to spend a second here on describing this in a bit more detail again. So basically what we use is we use culture collections that can be hundreds of different strains, and Joe already illustrated that there is now really great new methods on how to isolate very quickly very important microbes. So we have these culture collections. We also have the genome sequences. So we have taxonomic and genomic information. And with this, we can build defined synthetic communities or syncoms to whatever experiment we would like to do. And I want to highlight here a couple. One of them is a drop-in or drop-out experiment. So where you have a control community, and then in your test community, you leave out a strain or you add another one. And by this, you can identify keystone species, or you can look for competition. And similarly, you could do invasion experiments where you have one community to start with, and then you add another one, and then you can even investigate ecological phenomena like priority effects, dominance, or competition. And then, of course, we're also looking into mechanistic aspects, because we can genetically modify those strains if we have them isolated. So this is great. And the biological questions that I'm interested in is essentially what determines the microbiota composition. And we already also heard that environmental factors are influencing the root microbiome. And if you go out into a field, or if you do a greenhouse experiment and all these environmental factors stay the same, you still find distinct microbiota compositions on the roots of plants. And these are driven by host species specific factors, at least in part. So this could be the immune system, could be the chemical composition of the plant, or the ability to form mutualisms with microorganisms. And at the time, we were essentially, to begin with, interested in the question, can we use these controlled SynCom experiments to recapitulate these kind of results, these findings that we get when we look at natural communities? And the model plants that I'm working with and have been working with for a while are Arabidopsis, which doesn't need an introduction. And the other one is Lotus japonicus. That is a legume species that we use in the lab. And this is engaging with Rhizobia bacteria to form little nodules on the roots that you can see here. And this is a beneficial interaction because the bacteria are fixing nitrogen for the plant. And the beauty of this is that for both of these species, we have whole genome sequenced culture collections. And these culture collections derive from plants, from these two plants that were grown in the same soil that we collected in Cologne in Germany. Now, this slide here is showing the culture collection phylogeny. So on the left-hand side, you see this tree. And this is really just to illustrate that we have a lot of different bacterial families and we have overlap. So we have from both plant species representatives for all of these families. So indeed, we have taxonomic similarity and also some functional overlap. But essentially, then we were asking, why do these different plant species, if they have similar strains, what is driving still these differences in the communities associated with the roots, and are there strain-specific dynamics? And for this, we designed an experiment that we call a competition experiment. So essentially, we took strains from Lotus and strains from Arabidopsis and mixed them together, and we're asking if we then re-inoculate the two different plant species in a system, would then these strains again preferentially colonize the host from where they were derived from. So we do that in a gnotobiotic system that we call the flow pots. This is sterilized soil that we repopulate with these syncoms, and then we grow it in a controlled environment. Again, also these plastic boxes with filters on top. So this is really very handy for these kind of experiments. And then we assess the community, again, also by 16S gene profiling. The syncom that we use for this is meant in a way that we use a composition, a very diverse composition, with representatives of each of the bacterial families that we find in both culture collections, and then we have one representative each of Arabidopsis and Lotus in each for each family, and we can still distinguish these strains on the 16S basis. Now, when we do this experiment in our gnotobiotic system and we profile the communities in the roots, then we see here a really nice separation of the microbial communities between Arabidopsis roots and Lotus roots. And here we even included a Lotus mutant that does not engage in symbiosis, so they don't have nodules. And still, these root communities are very different to the Arabidopsis ones. And then, without showing the actual graph or quantification for this, but we took a step further and we're checking if the abundance of Lotus strains and Arabidopsis strains differs as a total on these plants. And then what you see here is essentially that the blue strains, which would be the ones that are from lotus, they are more abundant on lotus roots compared to the Arabidopsis roots, and the other way around. The Arabidopsis strains are more abundant in Arabidopsis compared to lotus. And this is what we termed the host preference of commensals, where when these strains are in competition with each other, they prefer the host of their origin. Now, this was really interesting, and we wanted to understand what does this actually mean for the plant? Do these specific communities have an effect? And we were first looking then at a transcriptional response in the roots. And for this experiment, we kept the communities separate. So we have lotus strains and Arabidopsis strains separately. And then we profile the transcriptional response in the Arabidopsis roots, shown here on the left-hand side, and on the lotus roots on the right-hand side. And I want to highlight a few clusters here that we deemed interesting. And these are the ones on the top, where you see that there's an induction of certain genes by the Arabidopsis strains on Arabidopsis, so by their own strains. And also in lotus, we find genes that are induced just by the lotus strains on lotus. And then when we take a step further and we compare these genes, we actually find homologs in these clusters across the two different plant species. And these are, in fact, related to immune responses. So here we have a few candidate genes or a few interesting homologs, and these are transcription factors that have been shown previously to be involved in immune signaling. And we thought this was interesting because we hypothesized that maybe the recruitment of these strains, of their own strains, is inducing an immune response, one part of the immune response, to maybe prime the plants against a coming pathogen attack. And to test this, we were then going into another type of experiment where we wanted to test if there's differential protection by these different syncoms against a pathogen. And for this, we used, again, our system where we keep the two syncoms separately, Arabidopsis and lotus strains. And then we have a pathogen or an opportunistic strain in our Arabidopsis culture collection. And this is a Pseudomonas strain. We call it root 401. And this is detrimental to the plants when you just put it by itself on the plants. And we indeed do this experiment in an agar plate system. So here the syncom would be in the agar of agar plates. We let the plants, the Arabidopsis plants, grow for 2 weeks and then inoculate them with this detrimental strain and then check how the plants look like. And this is the result. So I'm going to walk you through this. Basically, on the top panel here, you see when the plants were inoculated with root 401, that if the Arabidopsis strains, the Arabidopsis plants are only on MS medium, they die after a few days, so they are really not happy. If the Arabidopsis syncom is in the background, and here the number doesn't mean anything, it's just it's 17 strains still, then you still don't get a protection. But with the lotus strains, we do find this protection against the Arabidopsis derived strain. And this was a bit surprising because we thought that the own strains would be better in protecting against this pathogen. And one hypothesis why this is happening is that maybe the lotus strains can antagonize root 401 and the Arabidopsis strains cannot. We tested this in vitro, but we couldn't find any inhibition of root 401 by these other commensals. So we were asking, well, another possibility is that because root 401 comes from Arabidopsis as well, maybe it is more dominant and more competitive among these Arabidopsis strains. So we essentially made use of this in vitro test where we first tested if root 401 is antagonizing the commensals. And here, this is a halo inhibition assay where your strain of interest is in the agar, root 401 is in the middle. And if you get this halo of inhibition, this means that root 401 is secreting something that is indeed inhibiting the other strain. And then you can measure this halo. And here on the graph, you see that indeed root 401 is inhibiting a lot of these strains. So essentially all of them are inhibited by root 401. So then the next step was essentially to say, okay, what happens if we take this antagonism away from the strain? So we had already a mutant in the lab that was defective in 2 genes to disrupt pyoverdin production and DAPG biosynthesis. And DAPG is a known antimicrobial compound. Pyoverdin chelates iron, and this is also an effector for or a factor for antimicrobial activity. So if root 401 does not have these compounds, then here in the middle you see what happens to our infection assay. The strain itself is still detrimental, but now testing two different Arabidopsis communities here, actually here we get protection all of a sudden, and then also the Lotus communities can still protect. So this was interesting because this means that if the strain is less antagonistic, then it cannot thrive among the Arabidopsis strains. We wanted to understand a bit more how exactly this strain is influencing the bacterial community. So we did a profiling on the roots of these plants, and this is a complicated graph, but I will walk you through the most important things. So similar to what Joe showed earlier, the circles here are the relative abundance of a certain strain here on the left-hand side in the Arabidopsis syncom, on the right-hand side in the Lotus syncom, on the roots of these plants. So the bigger, the more abundant. The color here shows the change, so the increase or decrease compared to the mock condition. And if there is a circle around it, it is a significant change. And I just want to drive your attention to this, to these quantifications here, which are the Pseudomonas commensals in the respective syncoms. And here we see that in the Arabidopsis syncom, the Pseudomonas commensal is decreased whenever the root 401 strain is present. And remember, root 401 is also a Pseudomonas strain. And then in the Lotus syncom, we have a Pseudomonas strain where there is no change happening if root 401 is there or not. So we hypothesized that there is indeed some sort of specific interaction between these Pseudomonas strains. And to test if this is true, we did a dropout experiment in this case. And here on the left-hand side, you see the plant phenotype. On the right-hand side, you see the quantification of the shoot fresh weight. And here on the top row, you see that the Lotus syncom is protecting against root 401. And then if we leave out the commensal Pseudomonas strain from this syncom, then we lose protection. And on the other hand, if we add this Lotus commensal strain to the Arabidopsis syncom, all of a sudden we get protection here, full protection. And this was super interesting because it means that we can have one particular strain in a complex syncom that can be a competitor against a detrimental strain. So we were interested in seeing if this is true for other Pseudomonas strains as well, and how widespread it is, and if it depends on where the strains were deriving from. And we tested a bunch of other ones that we have in the culture collection. And here you see a phylogenetic tree of those. We have Lotus strains, we have Arabidopsis strains. And here in the plant assay, basically to make a long story very short, we see that we have different protection types, or we have protection or not protection for several different strains. And this does not correlate, indeed, with the in vitro capability of these Pseudomonas strains to inhibit root 401. So this is, again, a halo assay, but against root 401. So this means these Pseudomonas commensals, some of them can protect, some of them cannot. And this does not correlate with how inhibitory they are against the detrimental strain. So there must have been something that is driving this protection. And one theory one concept that has been shown also for the gut microbiome field is that we can have competition for resources in these microbial communities. And this could be a way of also repelling or keeping out a pathogen from a community. So we tested by looking at the genomes of these strains that we have, what the catabolic potential is of these strains. So we looked for catabolic gene clusters in all of these commensal Pseudomonas strains. And what you see in the graph is on the x-axis, there are compounds that can potentially be catabolized if the gene cluster is present, which is shown then here by these blue squares. And essentially what we find is that the more similar this profile for catabolism of a commensal strain towards root 401, the detrimental strain, the better this explains if it can protect in planta. So what we show is essentially that there's an intragenous, so within Pseudomonas, resource competition that may influence colonization success on the host. So I'm gonna sum this up here now. This is basically the study that we just recently published also. So Root 401 is a highly competitive strain. Syncoms can diminish the detrimental effect on Arabidopsis thaliana, and Root 401 requires these exometabolites, so pyroverdin and DAPG, to be able to be competitive among other strains. And we show that at least one part of the protection is explained by competition between Pseudomonas strains. And basically what this means is that probably if there is indeed carbon resource competition, this can be linked back to exudate profiles of the plants and what these plants are exuding. And this brings me to the final slide, where I just want to briefly mention a little bit where we're now going with this in the future and just in general in my lab. So we've shown a few years ago that there is host preference of commensals. We did that with lotus and Arabidopsis, and now we're expanding this to at least tomato and also to other crop plants that are more interesting for agriculture. We're looking at exudates, we're looking at plant performance in different projects. So for example, we also look at the role of transcription factors in the plant that control community assembly. And competition and cooperation, so similar to what I just showed you now. And I want to highlight this last part here, which is colonization patterns, because we know that along the root axis, we have different exudate profiles. We also have different microbial communities, actually. And we want to know a bit more about how host preference is being displayed on a spatio-temporal resolution. And with this, I would like to thank you for listening and acknowledge the people that were involved in this work. So this is our big group in Amsterdam. This is 3 PIs, people of 3 PIs, and the people highlighted here were involved in the study that I just showed you. And now I'm happy to take questions. Thank you very much.
Perfect. Fantastic talk, Catherine. That was amazing data. I always love seeing the host preference stuff. It's one of my favorite things to see.
Thank you.
I can start off the question. So it may be a bit of a silly one, but so Root 401 is quite antagonistic in vitro, right? So it seems to harass other things quite a lot. I'm curious, and I know I've seen a little bit of this in the literature. Have you looked at whether there's a relationship with biodiversity? So if you keep on adding strains with Root 401, at some point, I assume that antagonism must relax a bit or stop or be inhibited by something else. I'm curious whether you've done something like that to see.
Yeah. Do you mean in planta or in the in vitro assays?
In vitro. Yeah.
Okay. So the in vitro assays we only did indeed individually. So only one. So Root 401 inhibiting just one strain. So we didn't do that with communities. Indeed, yeah, that could be an interesting way to assess that. Yeah, what we have done is we looked at how Root 401 is influencing communities. So the syncom in vitro, so just in culture, not with the root. And we do find some differences to that, which just really also shows us that the host is involved. So there's a host signaling, of course, going on that then also plays a role for this protection.
Yeah. Excellent. Yeah, I think that's amazing. Another quick one is you mentioned you're going to be looking at this anyway, this spatial colonization patterns, as for future work.
Yes.
It would be quite interesting to see whether Root 401 does localize quite closely with a lot of other bacteria than that and has a stronger effect on the host. But I guess you'll be looking at that and you probably don't know quite yet. Is that right?
Yes. So this is indeed, actually that, research idea is, it was even meant more for looking at the different syncoms, so not specifically Root 401, but indeed this would be a great follow-up. And the caveat with this is that we need to label the strains with fluorophores. And I mean, I mentioned earlier that it's nice to have those strains and to be able to transform them, but it's not as straightforward, unfortunately, as it sounds. So this is work in progress. We haven't done any, we don't have any specific results on that yet. But yeah,
Perfect.
This is on the list.
Okay, I think there's one more question and then we can move on to the next session. So Louise here asks about when you evaluated your synthetic communities, are you planning to use any types of stress events like drought, nutrient deficiency, etc.?
Perfect question. Yeah. So essentially, what we did with this, with the story that I told you now, is to use biotic stress with this detrimental strain, and this was easy because we had the system already established in the lab. So indeed, now in a different project, in a PhD project, we're looking into other types of stresses to see if, how these communities can differentially protect or not, so that we indeed can make informed decisions on what a beneficial consortium could be like that we can use in agriculture. Yeah.
Perfect. All right, excellent. Thanks again, Catherine. That was a fantastic talk. And I'll hand over now to Tim, who will pass it to the next session. Thanks again.
Perfect. Thanks, Will. And thanks, speakers, for that session. It was really interesting. Just for anyone who's joining now, we are running a few minutes late, as you're probably aware, but that's okay. We'll make up some time in the coffee break, etc. So we'll pass over to John Atkinson now, who's the working group lead for phenotyping method development, and he has a couple of speakers he's going to introduce. So over to you, John. Thanks.
Hey, can you hear me okay?
Yeah, perfect.
Cool, brilliant. Right, Alex, if you're there, would you like to load up your talk, please?
Yep.
So it's my great pleasure to introduce Alex Grandon. He's a research scientist at IRD in Montpellier. He completed his PhD in 2011, and since then he's worked all over the world, really, spending a bit of time at University of Nebraska-Lincoln, IRRI, and ISRR in Senegal as well, who I think he still has quite strong ties with collaboratively. And today he's going to be talking about root xylem area and how it influences drought tolerance. So take it away, please, Alex.
Yeah. So John, I should use the share button, but it says that it's impossible.
Ah, you've not got permission. I don't know if you're there, Tim, and you can enable that.
No, you should. I mean, everyone should have access to the share. So if it's not there for you, Alex, then there may be just an issue specifically. So there's no way of giving you access, as it were, I don't think. So you should have it. So if you press the button, there's nothing happens, right?
Yeah, it's not working.
Okay.
Are you in browser or app?
Firefox.
Okay.
Right, I'll make you a co-host. I'll make you a presenter and see if that makes any difference.
If not, I had some issues earlier with the chat and had to leave and come back in to get it going. So,
has that made any difference at all?
Yeah, I try again. No, it's not working.
Okay, what I suggest, if it's okay, could we go on to Larry's talk if Larry's there, and then maybe if Alex, you can send John your slides, then maybe John can project them, or I can. If you send me and John the slides, and then we can project them instead, because I don't think there's any other way. I'll try making you a co-host now, just in case.
I just remember that Alex might have had a scheduling conflict, or.
Oh, is that right?
Yeah, but I'll connect. I'll try to connect again with another browser also. Maybe that's the issue. But yeah, go ahead, Larry, and I'll continue after.
My talk should be a little faster, I think. But
okay. Sorry about this disruption, but these things happen. It's fine.
Yeah.
So, thanks, Larry, for jumping in.
Oh, sure.
Yeah, thanks, Larry. Do you want to load your talk up, Larry, just to double check that's going to work?
Good idea.
I'll introduce you. Yeah, sorry about having to jump in there. Thank you for that.
Am I sharing the right screen?
It's loading, so while it loads, I'll just quickly introduce you. So for those who don't know Larry, he's a senior staff scientist at Oak Ridge National Lab in Tennessee. He completed his PhD in the Lynch Group in Penn State before moving to Nottingham in 2014, which is where we first met. Actually, we were postdocs on kind of aired projects, right? If I think back all that time ago. Yeah. After that, Larry moved back to the States, and he's done some time in Missouri and then the Noble before moving to Oak Ridge. And I think he's probably most Well, he's very well known partly for Riser Vision Explorer, which I'm sure loads of people who are watching this have used over the years and are very grateful for the open software that you've released. And today you're going to be talking out beyond Riser Vision and hyperspectral phenotyping, which I'm sure will be really interesting. So yeah, great. Thank you, Larry, for stepping in and take it away.
Yeah, thanks, John, and yeah, thanks to ISRR. It's great to see all these activities from the largest society, for root biology in the world, and really excited that we're doing these presentations and having chances to interact with the rest of the root biology community. It's a really special community to me. Today, yeah, I'll talk about root phenotyping kind of broadly, and in fact a bit of microbial phenotyping too. So it's kind of a mixed bag. So bear with me because that's just how I think and operate. So we'll get into it. I want to talk a little bit about the rhizosphere first. So this is actually a composite of a image I took at the Hounsfield facility in Nottingham, and I've used this for a review paper on the rhizosphere. So if we have roots growing in soil, Lawrence Hiltner, 1904, described the rhizosphere as the soil influenced by roots, and so I use that really simple definition. And as you know, roots are exudating, so you can barely see this like yellow film around them, and then there's different zones of uptake and gas exchange, microbial activity. There's larger zones maybe of water uptake and nitrate uptake. So the rhizosphere is really all these mixed zones. And if you kind of zoom in, you can see that it's you could describe it as a holistic rhizosphere of many interacting zones. And so in this review paper from my time in Nottingham, we described these group of equations as a way to kind of abstract all these interactions. And so I think that's what a lot of the talks today are talking about. And these are really complicated to study and break apart. And that's certainly what a lot of us are trying to do. And one thing I wanted to kind of update some of these ideas with a paper I'm about to submit. So I wanted to kind of, I think we all know that basically this rhizosphere bulk soil thing that we all use because of the methods available is basically a FOSS dichotomy. And so I wanted to get a bit more explicit about that and some ideas I've been developing. So yeah, we have things like rhizosphere development, rhizosphere conditioning, and even pedogenesis, or the formation of soils. These are things that occur that are influenced by roots across space and time scales. But as far as I know, there aren't very many sort of conceptual integrations of these ideas. So this is what I've been working on. So rhizosphere development occurs from seconds to weeks, and it involves more like individual roots. I think this is what most of us are typically accustomed to thinking about. This diagram on the left is sort of based on some earlier work from Jones et al. That actually focused on the mycology of this, but this rhizosphere development concept is really useful. So individual roots select, so the community in the rhizosphere is always there. It's sort of like the source. And then there seems to be a selection process within the rhizosphere. And especially root tips are really active. Then of course, eventually roots die. And when I wrote my review paper on the holistic rhizosphere, I was already kind of thinking about, like, well, really all soil has been influenced by roots. So if I look outside my window behind me, it's all like trees and grass lawn. And if you could show me any soil that wasn't influenced by roots, it'd be really difficult. So I started thinking, well, maybe all soil is rhizosphere. But when you go down that route, it's not very useful then. So how do you describe sort of the extent of how rhizosphere is really the question, not whether it's rhizosphere or not, maybe. And conditioning is another concept that people have used, and you might think of it as going from weeks to years, and it's within and among root systems, and it occurs as individual roots influence the soil and change it. And that change may not be quite permanent because it's always going, flux, and new roots are coming in, and but these rhizospheres can actually influence the development of other rhizospheres. And finally, at the largest scale, you can think of pedogenesis. So we know that soil formation takes a long time, and that it says I stopped sharing. I don't know why.
Yeah, I seem to have lost your slides, Larry. Sorry.
Should be sharing again, I believe.
Yep.
Okay. So this can occur over much longer times and occur over much larger scale. So I kind of was trying to zoom in on sort of a ecosystem and above. So yeah, in the end, I just wanted to kind of share these ideas to encourage us to think more broadly and holistically about the rhizosphere. So in my work at Oak Ridge, I kind of shifted from my work at Noble, where I was kind of getting really integrated into breeding pipelines. So a lot of my work there was focused on how to actually do root phenotyping to integrate root traits into breeding. And then my work at Oak Ridge has kind of pivoted me more towards more basic kind of research, but still working on phenotyping. So some concepts I've been sharing here for people, because when I got here, a lot of people were doing imaging, but I think they needed some help in kind of how to conceptualize and standardize things. So phenotyping in general is really about getting the right image in the right place at the right time. And we want to optimize the imaging to highlight the objects of interest and store the data. And we want to optimize image storage and analysis, use reproducible code that hopefully you're sharing. And so the right image is kind of like, here's one example of using a device called a rhizochip, which is a microfluidics device developed at Oak Ridge and also used at another national lab in the US National Lab Network. So then you can use tools like here I was trying out Dragonfly as a free commercial software for segmentation, and finally using Rhizovision for the feature analysis. But this basic pipeline is generally what people are doing with image analysis, and currently segmentation is really where AI is coming in a lot and doing model training. So when we decide what to phenotype, we usually have to make some type of hypothesis about what's important. And then once you decide that, then you have to develop a platform to measure that thing as quickly as possible. So like with roots, maybe numbers, links, and angles. And then we're typically measuring large populations, and sometimes that's for genetics. But to me, the more important thing is just to get the phenotypic variation. And of course, you can use that for mapping and getting genetic regions. But for me, a lot of the interesting side comes into looking at how all the traits are related to each other and depend on each other. So you have sort of these statistical analyses where you might identify interesting correlations. But as we like to say, correlation is not causation. So then to determine causation, you really have to follow up with manipulative studies, like with detailed physiological studies where you go more in depth. And then of course, you can also tie this all back into simulation modeling, where you can explicitly test your ideas and even new ideas. And I think Johannes Postma and others will talk about this in the next session. So this should tie in there as well. So a few years ago, I was doing this is also from Nottingham. I was doing imaging of this type, and I had been kind of told, like, yeah, just throw up some roots on a black background and you'll be fine. But what I eventually found out that it's not really quite that easy, and so that you can still do further optimization from techniques like this. And so when I got to Noble, I actually designed a different hardware setup that has a backlight and then the camera on the other side. So you're basically capturing shadows of the roots, which should be familiar to people because that's basically the same idea that we're using traditionally with the flatbed scanners that have the transparency light. So using a backlight can just give you really nice images where the image you see in that software is not thresholded. That's just the raw image from the camera. So it can just make image analysis really easy. So this software is also available, and I've seen that people do use this imager software with these Bossler cameras. Machine vision cameras are really well suited for these tasks compared to handheld kind of point and shoot. Yeah, so me and Anand have been working on these software for some years and continue to work on it. And I release everything as open. So one thing that I've noticed that like the software has impact, but so do your hardware plans. So I know that John has interest in that and shares like the hardware plans and stuff, and people actually use it. So here's an example where this platform was basically commercialized and is sold by Vienna Scientific, and they do like cite my work and everything. So that's nice. And on LinkedIn, every once in a while, I notice that people are actually using their own setup, so you can see the camera above the roots, and they're using the imager software. So yeah, I do take a lot of pleasure knowing that these tools help other people and want to continue to work on things like that. The Wish Roots project in Europe also makes use of rhizovision for wheat breeding. So if you think about going back to this type of image analysis, I don't know if any of you have done this before, but back in 75, the method was to lay out a bunch of roots on a grid and count the intersections. And you can work out an approximation of length from that. And it was in 87 that Alvin Smucker and others, as far as I know, this is sort of the first complete algorithm for how to do root image analysis that's in the literature. A few years later, people started to do papers on flatbed scanners. And then in around 1991, the Regent software company released WinRhizo based on all this work and the commercial availability of flatbed scanners. So arguably root phenotyping was sort of the original plant image-based phenotyping, and it has been going on for 30 years. But we did see some gaps a few years ago. So I ended up having an opportunity to work with Anand to develop Rhizovision Explorer, and so that's been released for a few years, and I think it's proven to be pretty useful and popular. It doesn't do everything, and so there's definitely lots of room for other software. So I really view it as a community or ecosystem of software that we can work to make as interoperable as possible. But the new version, which is actually on GitHub right now, so if you want to try it, you can. It's a beta. It doesn't have all the features I want to get into sort of like the full release, but the important things that it does have already is a command line interface, and you can now compile it for Linux for the first time. So that allows us, for example, for us here at Oak Ridge, we're setting it up to work with our phenotyping facility, which now has rhizoboxes in it. That's automated. And so that's allowing us to call it from scripts and things like that. So you can automate it. So I made the GUI to make it easy for everybody to use it, but then in automated phenotyping, that becomes a limitation. So this kind of solves those limitations. And we're going to add a few more traits to it, like for rhizoboxes and stuff, and then release that as the version 2. And then in the future, I would like to continue to work on things like how do you identify lateral roots from the axial roots and things like that, which we still only do by diameter classes. So some of my work here now is shifted to using the rhizoboxes. So here I'm just giving some examples of using the rhizoboxes for imaging and even collecting exudates and things like that. I'm trying to work up this as a hardware release as well. And then for this project, I've been proposing to use these new hyperspectral cameras we have that originally when I heard about hyperspectral, I thought it was like something really nuts and everything, and it sounds really fancy. But in the end, it's just that normal cameras have 3 colors, and hyperspectral cameras, like mine, for example, has 300 colors. That in it, one camera goes from 500 nanometers, which is bluish, out to 1700 nanometers, which is in the shortwave infrared region. And so a lot of these bands are related to things like water content and other things. And then after you get used to the image analysis of them, you can see that it just generalizes from having 3 colors to 300 colors. So you need more computational power, but conceptually it's not actually all that different. But yeah, it takes a lot of work to work through it. And so we've been working through all this analysis, and I hope to share more about that soon. And also how to co-register the sampling events with the hyperspectral imaging so that we can tie things together, like the signatures from hyperspectral to things like microbiome, community, or other types of soil properties. So we can measure things like the water content, and hopefully we can get some prediction based on where all the roots are over time. I believe that we can explain some of the, quote, missing variation that we typically see with microbiome studies about what is driving sort of the variation that you see in the microbiome, like from the genotype. We see some genotype effects, but it's relatively small compared to like the error term in a lot of these studies. So I'm hoping that these methods will help to address the error term a bit. So some early data from our phenotyping facility here is that we see color images of roots growing of populous species trees, which is a big focus here at Oak Ridge, growing on the left, the color images, they're a higher resolution. And on the right, this actually isn't hyperspectral. This is a near infrared camera that gets strobed with 2 LEDs at 940 nanometers and 1450. So 940 is less responsive to water, and 1450 is more responsive to water. And so when you do pixel level math and divide them, you get some index of water content, which I believe that we can see here in these images. This is more qualitative at the moment. But what I think that you can see is that there's more blue at the bottom, which is typical because we do see the water kind of drains in these. And also you see that the plant itself is wet, which I'm well aware of from my time at the Hounsfield as well, because one of the hardest parts with X-ray imaging is that you're imaging wet tubes of roots inside of a wet soil, which makes the contrast at times be a bit low. So we're starting to get this type of data, and we're working through how to link. So we have ground truth measurements of the actual water content with these images, and we're working through sort of all those analyses, and also how to register the color images with these, and then of course do all the segmentation. So we're hoping to do like different species, different textures, and different stresses in these systems, and quantify things like the image-based data, as well as like microbiome analysis. And there's a lot of work here at Oak Ridge with mass spec for metabolites and proteomics. And we have a really cool new mass spec instrument that allows really high resolution measurements, where we can even do isotopic differences of things like the proteins. So there's a lot of opportunities to do isotope labeling as well. I want to talk a little bit about microbial phenotyping. So this is me actually last Christmas. I was actually here because I was trying to get a data set daily. And so I didn't want to skip it. So I came in for an hour to do this imaging. And we bought this colony docket thing that's really meant for colony counting, but we just wanted a standardized way to measure the fungus. And so here's a way that on petri dishes you can get these type of images really quickly. And you could probably just do it with other types of cameras too, but I wanted to explore overhead lighting versus transmission lighting and things like that, to kind of understand like what type of lighting is good for this. And what I really like about fungus compared to plants is that like the variability is really low. And you can basically see the results of this experiment like this. So this is all the images that I collected, 360 images. 2 different species, fungus and bacteria, can also be quite beautiful. And so there's 2 species. Within each of these groups of plants, there's 5 replicates. So it's really amazing how low the differences are among replicates. And then also there's 3 levels of nitrogen in this glucose minimal media on agar. And you can see those results, that when it's really low nitrogen, you see very faint organisms, and it's darker when there's higher. Here's some close-ups. I can't belabor it, but I think this one looks like the eye of Sauron from Lord of the Rings. And then this species also has a quite different architecture. So we've been working on things like how do you get the colony size and things like that, but I think there's a lot more hiding in that data, like how there's little rings of stuff, and how do you quantify all that. I also worked out a method to get the respiration of these. So here we have a closed loop with a Lycor 850, and you can delid the petri dish and put it inside of this chamber and measure the CO2 flux going up, and then you can fit curves to get the actual respiration rates. So for this small data set, I do have sort of one physiological measurement plus the image data, and we're working through how to combine it. We've annotated a bunch of these and trained a unit on doing the segmentation, which works okay. So we'll be sharing all that data soon. And on the trait analysis side, so if you get something like area, just like we could see in the pictures, we can see it in this data as well, that you have the time series here, you have the size or area by pixel count. We have the 2 strains in red and blue here, and then the different levels of nitrogen. So, and like, for example, in the FG, you can see that the high levels of nitrogen grow almost the same. And only when you get down to 1% of the normal levels is where you get sort of a reduction in growth. So it means that we probably use a lot more nitrogen than we need to in these mixes. So I think this is basically some examples of nitrogen use efficiency in fungus. And I'm trying to work through how do you kind of present. We have a ton of data, and it's so much that how do you narrow it down to a cohesive story for like a paper is really the challenge with phenotyping data. One cool thing is doing basically regression across all the time points against the image data with respiration. You can see that you would think that the respiration was only done at the last time. So you would think that the last time would have the highest correlations to respiration. But in fact, we found a lot of earlier events that predict the endpoint respiration. And I'm seeing that broadly with time series data, that it's not necessarily the endpoint data that is the most predictive. So that's important to work out. And so in this project, I was doing all this work as this manual labor to image, and I thought, man, it'd be cool to have a robot. And so like a year ago, I started to look into it, and I looked into a bunch of different vendors that sell these things, and eventually I figured, well, I'm gonna try this robot and the gripper you can purchase for less than 10,000 US dollars. So I thought, well, I'm just gonna go for it, and so I did. And over the past year, I've basically learned how to control the robot, which all these things I'm using have Python SDK, so it's becoming much more approachable. And I'm hoping to release this as well as like a full hardware spec. And what I've worked on most recently is actually like things like the safety controller, so that I actually have this up to industrial safety standards as well, with like a door interlock and things like that. So this thing is capable of imaging. I vibe coded, I'll be honest, a GUI to control it and images. And in fact, I included, I don't show it here, but it actually can upload the image to Chat GPT and describe it to you. So you can start to think about how do you bring in things like agents, right? I think we'll be hearing a lot more about that. Here's what the software looks like. You connect to the robot. You can select which plants to image. I've turned on the AI agent here. You can choose which shelves. And then in a moment, you can't see what the robot's doing, of course, but we should get the image and then the summary. And that will be near the end of my talk. I think I'm getting to the end of the time here. So. So my daughter's really into K-pop Demon Hunters. So you can see it actually gets, there's a purple marker drawing that says there's tape and cracks on it. Maybe you should look at that. It's like, yeah, probably I should really. But yeah, these are my test plates. So another work, I'm working on a data analysis agent using a local model, and that's been pretty fun. We're hoping to tie that into like live phenotyping over time. So I guess like agentic phenomics might be sort of the next step for us. And I want to thank all my colleagues here at Oak Ridge. And a lot of this work came from the Plant Microbe Interfaces SFA, which is a long-term DOE funded project. So thanks to the DOE for the support, and thank you all for listening.
Thanks, Larry. That was really interesting. There's a couple of questions in the chat for you, but before we go, I just wanted to ask you a little bit about your plans for sharing hardware and what you think about how to actually best do that, because it's something that I know we run into real difficulty with, right? Like publications and the publishers papers are not designed to allow you to share hardware easily. And I just wondered if you had any thoughts on the best way to do that, where it actually can be used by other people and benefit them.
Mm-hmm. So what I've been doing, is like basically sharing PDFs and plans on Zenodo, just as complete as you can document it. I'm trying to get internal funding here now that would like, part of the proposal is doing like videos and things like that too. If we could have more videos of like the assembly process. Because I definitely realize that you could release the plans, but still like these things they are hard to do. Like none of this is easy, but I think we can just do our best to lay it out as clearly as we can. As far as publishing, I generally put software and hardware on Zenodo, and I side it with the DOIs and like the kind of biology publication. And I agree it's hard to like publish a paper like just on the hardware and things like that, but maybe journals, especially like phenotyping journals, should be more open to that, because it's so much work, and we need to get deliverables for all that work too. So it'd be more fair to us that do a lot of method development if we did have those opportunities. So I feel your pain.
Good points. There's a question from Alina, who's curious about the chamber for measuring CO2 level, and is it ventilated somehow? How does it work?
So yeah, what I actually found, I'm using it for a couple of places, that if you need like sealed chambers, that electrical enclosures are good options. So that is actually more sold for like electrical enclosure, maybe for small computers or boards and things like that, just happen to be clear. It has a rubber grommet, so it seals pretty well. I haven't tested leaking, really well. And then those closed loop systems, I just have the bulkheads that you can buy from Lycor, so I can plug in the tubes that make a closed loop between the enclosure and a Lycor 850, and that closed loop lets you measure the accumulation of CO2. So the data that you get is a time series graph, basically, of CO2 accumulation.
That's a good solution. In the interest of time, we should probably move on to Alex. Are you happy to answer the couple of other questions in the chat for me, if that's okay?
Yeah, of course. Thanks so much,
Thank you.
John. That's great.
Yeah, thanks, Larry. That was a great talk. Alex, should we have a go and see if you've got sharing permission now? And if not, I've got your talk here, so I can share it for you.
Yeah, so I'm trying again.
I can see me, so I think it might be working.
Yeah, it's working. Yeah, perfect. So can you see the presentation?
Yeah, you just need to put it into full screen and we're good to go.
Is it
wonderful?
Full screen now?
Yeah, perfect. So the next talk is Alex Grondon, who's going to be talking to us a bit about root metaxylem area and how it influences drought tolerance in pearl millet. So yeah, take it away, Alex.
Yes, thank you, John, and thank you all. Good afternoon or good morning. Yeah, pearl millet. It's a tough crop. It's a tough crop because it grows in arid and semi-arid areas, mostly in West Africa and in India. And it's yeah, very important for food security in these regions. And pearl millet is actually one of the only crop that will not fail in these conditions, but still, it's very sensitive to water availabilities during the season. And so it's a very good model actually to study drought tolerance mechanisms. So that's what we do by employing a water-centered view in which we sort of view water as flowing across the plant in the different compartments. And this is a result of a fine equilibrium between root water supply and shoot water demand. And all the plants will adjust this equilibrium when the conditions become limiting, whether in the soil or in the atmosphere, will impact the plant's water use and the dynamic of water use a long time and eventually its yield. So within this framework, we hypothesize that root hydraulics play an important role in plant hydraulics. And so we try to answer the questions. We tried to answer the questions of how root hydraulic traits influence pearl millet water use dynamics and yield under drought. And since yield is a very important component for us in our research, we decided to address this question by going directly to the field. And so we grew a diversity panel of pearl millet lines. So we grew 160 lines in irrigated conditions and drought stress conditions in the field in Senegal over 2 years. And we applied the vegetative drought stress because it's one of the most impactful and recurrent stress for the past 20 years. And we looked for links between root anatomical traits and yield yield. So root anatomy was phenotyped on node 4 at the base of node 4 after the period of drought stress, which was 3 weeks after irrigation and 3 weeks of a dry down. And at the end of the cycle, we also measured yield and yield maintenance in the drought and look for links between these traits. And so when we did that, we observed a robust and significant correlation between metaxylem area and mean area of metaxylem over the 2 years, which was actually a strong result for us. And the correlation was observed with yield maintenance under drought and yield under drought. So basically what this means is that plants that had larger metaxylem were more tolerant to drought, or were able to maintain their yield higher under drought conditions. So to go a little bit further into the understanding of how metaxylem traits influence water use dynamics in Pearl millet, and how does this impact yield under drought conditions, we did not really add any genetic material to really address this question directly. So we had to deal with the genetic genetic diversity. So we selected a panel of inbred lines that contrasted for metaxylem related traits. But within this panel, we wanted to make sure that what we would observe in terms of water use would be comparable and really due to the differences in metaxylem area. So we had to make sure that some other traits might not hide or influence the impact or the function of total metaxylem area. So for that, we tried as much as possible to fix shoot dry weight. So you can see that in the panel, the shoot dry weight is more or less stable, similar, and also the root dry weight. Having said that, you may have noticed also that these are from field experiments where we measured the root anatomy or the metaxylem area at the base on node 4. So we did not know at the time if that was really representative of the whole root system. And also we did not know, or we wanted to check whether metaxylem area would also influence root hydraulics in Pearl millet. So to address the first question, we measured root anatomy in the contrasting lines. So here I'm just showing 2 lines, but that's the same similar results for all the lines along the route from node 4. So the node 4 is here. And what we can see is that when plants, when the root has larger metaxylem vessels at the base, it's actually conserved along the roots, as you can see in both of these images. We also checked whether this phenotype was conserved between roots from different nodes. So we sampled node 3, 4, and 5. And so this is the total metaxylem area at the different nodes. So node 3 is the latest, the earlier, the oldest, and node 5 is the youngest here. And these are the different genotypes, and you can see that it increases, the metaxylem area increases when in newer nodes. But as you can see in the color code here, the ranking is quite conserved, indicating that measurement at the base of node 4 is actually representative of metaxylem area variations along and across roots. So to address the second question, we also measured the root hydraulic conductance of the different lines that we selected in our subpanels, and we did that by growing the plants in hydroponic conditions and using a show lambda bomb to measure sap flux, and then we scanned the roots. So that was using WinRhizo actually. And this, by this method, we were able to calculate the root conductance, and we found a significant and positive correlation between the conductance and the total metaxylem area and number of metaxylem, indicating again that when we measured root anatomy at the base of node 4, that was representative of the root system, and that allowed that increased the root hydraulic conductance of the root system. And actually, to go a little bit further into this root hydraulic traits, we also collaborated with Juan Baca Cabrera at ULISH, and Juan did some granular mecha simulations on the sub-panel, which suggested that the radial hydraulic conductivity within our lines was not significantly different, was very similar, but in fact we could definitely, that confirmed that we could discriminate these lines based on axial hydraulic conductance. You can see that in this graph here. So all these results in mind, we were quite confident that using this sub-panel, we would be able to show the impact of metaxylem on metaxylem area on water use dynamics and the drought. So that's what we tried to do by measuring the impact of metaxylem size on plant water use in a lysimetric platform that we have here in Montpellier. So this is composed of 240 load cells that can measure the pot weight dynamically a long time at intervals of 30 minutes. So we applied more or less the same drought stress that we applied in the field conditions. So 3 weeks of growth and then 3 weeks of drought. And we did that also thinking that, okay, the soil has a strong impact on hydraulics, so we want to be as close as possible to the soil texture that we find in the field. So we developed a soil which is composed of a particular sand with particular granulometry mixed with peat. And Mathieu Javot and his colleagues at University of Louvain measured the hydraulic characteristics of this soil, and we actually confirmed that it had sandy soil hydraulic characteristics, which means low water retention ability and also a sharp decrease in soil hydraulic conductivity upon soil drying as compared to the peat soil that we used currently previously. And actually this sandy soil that we use in the greenhouse had a very similar characteristic to the soil that was present in Senegal in the field. So we used that soil and we grew the plants and applied the dry down. We were able to control the dry down. So what you can see here is, for example, for 2 load cells as an example, and you can see that from saturated soil, we stabilize the irrigated plants at around 80% of fraction of transpirable soil water. And the stress plants were dried, the soil of the stress plants were dried using different thresholds, so 80%, 60%, 40%, and we tried to reach around 20% at the end of the experiment. And that gave us the daily water uptake because we could collect the weight of the pots every day. But we could also do this at the daily scale, so we could also measure the transpiration response to the evaporative demand at different days. So here you can see the transpiration increasing early in the morning and then decreasing in the afternoon, nighttime, and so on. So using these results, we were able to measure the transpiration drops in irrigated plants as compared to well-watered plants. And that's what you see here. So this is a normalized transpiration. So this is, and this is the fraction of transpirable soil water. So this is irrigated soil. And if you go in that direction, the water content within the soil decreases. And what you can see is as compared to well-watered plants, the drought stress plants start to decrease their transpiration at some fraction of transpirable soil water until a point where the plants stop transpiring completely. And what I've plotted here is the plants from that was grouped into narrow metaxylem vessels and large metaxylem vessels. And what you can see is that the plant with larger metaxylem vessels tend to close their stomata or to drop the transpiration earlier during the dry down as compared to the plants with narrow metaxylem vessels. And that was significant. So the inflection point of plants with larger metaxylem vessels was earlier during the dry down. Okay, so that was observed in the groups. And when we did that on all the plants within the greenhouse, we saw a similar pattern. So a positive and significant correlation between the inflection point, so somewhere around here, and the size of metaxylem area. And as you can imagine, the inflection point translate also, earlier inflection point during the dry down also translate in water savings. So the earlier the plant would drop its transpiration or inflect its transpiration, the less it will use water. So all this indicated that plants with larger metaxylem vessels exhibited more conservative water use as compared to plants with narrow metaxylem vessels. And that was in line with what we sort of observed in the field, where larger metaxylem vessels was associated with drought tolerance. So at the daily scale now, we observed similar patterns where when we looked at the transpiration along the day, so this is time of the day, transpiration, under irrigated conditions, you can see that large metaxylem vessels transpired more than plants with narrow metaxylem vessels, which is somewhat expected. But under drought conditions, as observed before, plants with larger metaxylem vessels actually transpired less than plants with narrow metaxylem vessels. And when we looked at drought tolerance related parameters, so where we did not observe significant differences in terms of stomatal conductance measured using a Likor 6400, but we did see a significant increase in assimilation under drought conditions in the plants with larger metaxylem vessels, indicating again that these plants conserved some water and were in a more tolerant stage during the drought stress, at the end of the drought stress. So to sort of summarize these results, what we have now, what we would like to propose as a working model is that in dry soil, in pearl millet, plants with larger metaxylem vessels are actually restrict their transpiration, are more sensitive to drought and restrict the transpiration during the day, which potentially induce water savings during the drought period and tend to improve drought tolerance. Now we know that this is sort of contradictory with some other results that are in the literature on other species, but we think that this has to do with the soil hydraulic conductance and the fact that we are working in sand. And in fact, I'm not showing the results here for the sake of time, but we did exactly the same type of experiment in a loam soil, and we see exactly the opposite results. So we think that in a sandy soil, the larger metaxylem vessels are more, plants with larger metaxylem vessels are more sensitive to soil drying, and the soil becomes more quickly limiting for water uptake as compared to plants with smaller or narrower metaxylem vessels. Okay, so with that, I'd like to thank all the collaborators and all the people who did this work in Senegal, in Europe, and in Montpellier, our colleagues at Montpellier. And I also like to thank our founders that allowed us to make this work happen. Thank you.
Thanks, Alex. That was fascinating. I really enjoyed that. You've had a few questions in the chat. The first one's actually from Larry, who's been curious about using weight data over time. And he was asking you, what analysis are you using specifically to do your calculations on weight, and if you've got any code that's available for that?
Yeah, we use a code that was published actually. It's a paper from Carr et al. in Journal Experimental Botany. It was based on data from a larger glycemetric platform developed at ICRISAT by Vincent Vades, but it works equally well for smaller platforms the one we have in Montpellier. So we do have automated codes to generate the data quite fast. Yeah, the weighing data quite fast.
Maybe you could drop a link to that in the chat after this under Larry's comment, just so people can access it if they're interested. That'd be really good. You have another question from Johannes, which I think you have started to answer. But it was that plants with larger metaxylem vessels and greater axial conductance should have less negative water potential in the leaves. So why would they close their stomata sooner?
Yeah, so we measured actually water potential in the leaves along the dry down, and what we could see is that the plants with larger metaxylem vessels would have indeed less negative water potential at midday, but earlier during the increase in the evaporative demand, so water potential started to drop, and we think that possibly, so this is our hypothesis at the moment, larger metaxylem vessels would sort of reach a limitation more quickly during the day when the evaporative demand in the rhizosphere, leading to a drop in soil water potential, and as a consequence, earlier drop in leaf water potential, and then stomatal conductance, and then reduction in stomatal conductance. So that's our hypothesis. But then when we measured it, maybe we were already past that time, and then we sort of observed that plants with larger metaxylem vessels had lesser drop in leaf water potential, which sort of indicated also that they were sort of recovering also from the stress.
Interesting. You've got one more question from Amelia, which is asking about your thoughts on larger xylem vessels, drought, and then the possibility of embolism as a result.
Yeah, that's a good question. This is definitely a possibility that has been measured in roots, in stems, in peduncles, in wheat. In pearl millet, we did measure it also with collaborators, and the P50 was actually very low, below -3 megapascals. So we did in the field, we never measured soil water potentials. We did measure water potentials in the soil in our experiments in Montpellier, and we never reached at 20% field capacity. We never went below -3 megapascals. So this indicates that potentially we are still not at this threshold yet. But that would need to be confirmed in more detail, but that's not easy to do in roots.
Yeah, interesting. Well, thank you for a really interesting talk and for answering the questions. And yeah, that just leaves me to say thank you to both speakers. Those were really interesting talks, and pass back to Tim. I think we're running a little bit behind, aren't we? So I'm not sure what your plan is now.
No, that's fine. So yeah, again, thanks, Larry and Alex, for great talks, but also for being flexible, just to try and keep us running as smoothly as possible. So we are about 15 minutes behind, so that's okay. I think we'll take a shorter coffee break. We're supposed to take 20 minutes, but I think we'll take 15. So if we can all come back at 2:10 UTC. The breakout sessions are still open, so if the working group leaders want to go into the breakouts, then they can have a chat with anyone who wants to go along there. So again, at the bottom of the screen, there's a breakout session button. Press the breakout session button and then join whichever working group you're interested in talking to. Otherwise, 15 minutes, have a break, have a walk around, have a coffee, and we'll see you back at 2:10 UTC.
Session. We will have two speakers today, Johannes Posma and Mona Giraud. So I will introduce the first speaker, Johannes. So Johannes is a current senior scientist in the Forschungszentrum Jülich. Dr. Posma completed his PhD at the Lynch Lab in Penn State University, where he focused on integrating the carbon cost, among other parameters, into SIMROOT. And after his PhD, he moved to the Forschungszentrum Jülich to start the root dynamics group. There he further developed SimRoot, and he now transitioned it into an open version of it, the Open SimRoot, which include many more parameters and variables and submodels. But I think Johannes will talk more about it. So please join me to welcome in Dr. Johannes Posma.
Well, thank you very much for your introduction. Yes, it was a long day. We had a lot of information, really cool talks. And so maybe this one is going to be relatively light because the message is basically this is school modeling that you could use. But at the same time, it's going to be a bit heavy because this model has been used for many different kinds of studies, and I want to show by example basically how it could be used. Let me first start sharing. A moment. Share. I think you should be seeing my slide now.
Yeah,
Is that
it works.
correct? Yes. So OpenSimRoot is a code for constructing root architectural models and has been around for quite some time. Started indeed as SimRoot in the Lynch lab, and then in 2017 we open sourced it. Now as part of the Root to Resilience project, there was a new release of that, in which we've tried to pull together all the different bits of codes that were in forks and branches for different projects, and really try to thoroughly test this in terms of backward compatibility, in terms of benchmarking, etc. And this is what I want to show you, what we did. So OpenSimRoot not only simulates root architecture, but we also do the shoot growth so that we have leaf growth and leaf senescence and photosynthesis, and then of course root growth, and then we have a carbon model associated with that, a water model that does hydrology similar to the previous talk, and then nutrients, mostly N, P, and K we focus on. That looks like this, right? So I hope that over the internet this animation came across, where you see in this case a simulation of a root architecture of a sweet potato plant growing in a soil with some colors that represent, in this case, nitrate concentrations. Yes, so first part is that we parameterize for new crops, and I think that's interesting. Since 2017, there was a rice model published, a cassava model, a pearl millet model, and a barley model. We already had maize, bean, squash, and lupin, and currently under construction, which will be published probably also soon, is wheat, sweet potato, faba bean, and Arabidopsis. So that means that we have parameter sets for quite a few crops. Maybe your favorite crop is among them. What we do is model a root architecture. Here you see an example where we try to go from phenotyping of field and greenhouse to a simulation, right? And here's an example of rice, similar, an example of cassava, where in the blue background is a photo box, somewhat similar to what Larry York presented, but not with his nice backlight, but just with a blue paper in the background. And then we made a model out of that to understand cassava root systems and also their storage roots. And then, of course, you can compare the data in the literature, the measurements, and the simulation to see if that data is similar or not. Here's an example of an MRI image is converted to a model, and we've done that also with CT images. So from phenotyping to root architecture to represent that root architecture as well as we can, so that we can ask questions about, well, what does it mean to have a root architecture like that in terms of carbon use, in terms of nutrient uptake, water uptake. But first, we have to confirm that that root architecture is actually properly represented at the plants that we observed. And this is part of a benchmarking paper that is in preparation, that's led by Delori. He made a very nice RSML package for R, right, that can read in RSML files and then can analyze a lot of traits of from those RSML files. And so what we did is different models wrote the RSML file for a certain phenotyping exercise, and then it was compared to the original phenotype roots. And what you see here in the middle, right here, the purple triangle, these were the observations, and the green one here is the OpenSymRoot results. So the PCA, they are very close. We are, with respect to many traits, very similar to the phenotyped traits, showing that we can represent roots quite well. If you then can represent roots quite well, can we actually develop phenotyping software on simulated images? Yes, this can be done. 2 examples that were done in the last 5 years. Here's an example of a cassava root. Already said we had these images or videos actually from rotating root crowns, and we wanted to analyze those. And what we initially did is make simulations of a similar root crown and try to train our software on those simulations, and then try to use that to see how well it worked on the real root systems. And so we can use actually the renderings of OpenSymRoot to improve our image segmentation methods. Another example was a paper on virtual coring, where we try to find out best practices for positioning the core with respect to row and inter-row spacing in the field. When you place a core in the row or in between the row, you will get a different root length density profile. Then these are still architectures that are basically observed and reproduced, right? We're not predicting architecture, but we would like to move towards a predictive model of root architecture. And for a predictive model, we need to have understanding of the root plasticity growth responses. Here's real world data where we placed a fertilizer patch in a rhizotron, and then you see these cassava roots that they heavily proliferate around that patch. And can we simulate these kind of responses? And so we tried to develop the code so that we could have different kinds of plasticity responses. Here is kind of an extreme example for a split root system, and then for the kind of patches as we saw in the previous observation. Still, parameterizing these plasticity responses is quite a challenge, mostly from a data perspective. We can also ask from the other side, are these plasticity responses actually useful or not? And here we did a study where we compared plastic versus non-plastic root systems. What you see here in the blue line is the best plastic root system that I could find. And in the orange line, is here basically giving an optimized non-plastic one. And so you see that the plasticity does give you some edge over a non-plastic one, but if you can optimize for a certain condition, right, then you wouldn't have to be plastic anymore. Yes, but that's the whole point, that a plant is plastic so that it can handle several conditions, right? It's to be able to stable across environments, not to be optimized for a single environment. So if we know the environment very well, a optimized genotype probably could do nearly as well as a plastic one. But if the environment is unpredictable, plasticity is going to help us. This was very nicely worked out in a very recent paper by Harini Ragaranya, where they looked at lateral root penetration ability. So how well are roots able to penetrate a hard layer? And they compared lateral roots versus axial roots. and in native soils, right, and in cultivated soils. And what they found is a different combination of an optimum. And mostly here, I find this very interesting. We see that in both cases, there's an optimal penetration ability. That means that these roots shouldn't penetrate too fast through those layers, neither be too slow. And I think that's a very, very interesting result, suggesting that actually an arrest of roots, right, here's an arrest, slower than 1 and here 2, is actually suggests that this is adaptive. Modeling root anatomy, we have used the model to do several anatomy studies, and here are listed the papers that have all root anatomy and OpenSimRoot combined. I highlighted here one study that I thought was very interesting that came out in 2024 recently, which was about the size of the parenchyma wall width in the cells in maize, suggesting that thick width actually is beneficial under low nitrogen, whereas under high nitrogen, the model will not predict much difference between these traits. So it can be used to explore basically the benefit of these anatomical traits and scale it up to the whole plant. All the anatomy papers, just look them up, and they have all kinds of different anatomy traits there. We model carbon. Of course, we have a labile carbon pool that's basically a non-structural carbon pool that can be allocated to leaf stems, roots, to carbon exudation and to respiration. And of course, we have input from photosynthesis, seed reserves. And of course, when we have leaves grown, it goes back to photosynthesis. This is the very basics of basically a crop model or any plant photosynthesis carbon model. But of course, this idea can be expanded, and then when we expand it, we get maybe a box model like this. This is an example of OpenSimRoot's network that's simulated for cassava. These kind of networks, the model can actually output automatically, so you can know what you're simulating. And then we went one step further and said, well, can we add metabolic models to OpenSimRoot? And this is an example of a flux balance model that describes the metabolic network of photosynthesis in cassava. So this input of CO2 here and of light from a photon flux density, and then in the end is output of biomass. But this biomass is a certain amount of fiber, protein, lipids, and carbohydrates, etc., as basically measured. And then we can try to work out this all the fluxes in this network using flux balance modeling techniques. And then also what comes out of that network is the efficiency of it. And so this we wanted to couple to OpenSimRoot. So we had a flux balance model of a source leaf and of a sink leaf. So source leaf doing photosynthesis, sink leaf is growing. And then we had also a root that is producing starch, storage root. And so different flux balance models that we couple to OpenSimRoot in order to scale them up and to understand these models in a dynamic context of a growing plant. For this, we developed a Python interface so that we can use the flux balance models and the Cobra network that is used for that. And then, of course, in the end, you want to validate the carbon model to say, okay, are we predicting the plant dry mass well that we measured, the leaf mass fraction and stem mass fraction, etc. Modeling water is then also something that we put a lot of effort in to improve that. And this led to a very good, I think, hydrology model that was part of the benchmarking exercise by my colleague, led by my colleague Andreas Schnepf, and where I think we did quite well with our code. Here's just one example of evaporation from the soil. So as the soil dries, the evaporation goes down. And this, in principle, the switch from potential to actual is a really hard one numerically. And here you see comparison of two implementations that we have in OpenSimRoot. One that is basically slow and off, and one that is very fast and very accurate. So when we then have a good hydrology model, we can start using it for studying drought. And here's an example of a drought study that we did, but not published yet, in which we try to understand how root diameter affects basically the growth of a plant under different drought scenarios. And so here you see after 24 days, the model says, well, have thinner roots, but then if we wait a little longer and we wait till 90 days after germination, we actually end up with an optimal root diameter, which is slightly thinner than what our reference was. And so I'm very happy. Last ISRR, I presented an optimal root diameter in maize for nitrogen and phosphorus uptake. And now here for barley for water uptake as well, that we can start understanding, okay, why do we have plants certain diameter roots? Does that work with real world data? This is field data, variation in genotypes and locations from the Root Resilience Project. And there is also a slightly negative correlation, meaning that in these barley lines that have more thin roots, right, they tend to have a little bit better growth. So that's a correlation that kind of fits with the results that we have. Find that fine roots tend to be doing a little bit slightly better. Deep rooting was also a big question that we tried to study. Here's a correlation between rooting depth in terms of D90, so the depth above which 90% of the roots reside. So this is deeper on this end of the axis. And here I suggest that the deep rooting barley lines would grow slightly better, but this depends really on the environment. Do we have a drought environment with low precipitation, or do we have a situation where we have rainfall, but we start with a dry soil, like at the end of the dry season, the soil is very dry, rain is falling on top of it. And then, of course, we have much more surface water, and the relation between root depth and shoot dry weight is basically lost there. Then the last thing I want to say is something about code, right? Because if you would like to work with a model and do one of your own studies, of course, you should know something about the code. It's C++ code, and that is open source that you can download. It's modular through a plugin infrastructure so that it's easy to extend it. We have a highly flexible XML-based definitions that define the models and the parameters. We made it extensible using a Python or an own formula interpreter. And we tested for backwards compatibility very carefully. And I think that's very important that this model is stable and old studies are still valid today. And so my suggestion is, if you're interested to use it, try it out. If you then still like it, fork the code to create your own private fork and make your changes to either the input or the code, publish your paper, and then most important, after you publish your paper, send me a merge request so that your code can become part of the main repository. How do you get started? We wrote 3 practice abstracts, and the one that gives you a good way to start, you can find under this QR code. And it will list you in 5 small steps how to download the code and get a simulation running on your computer. You could also visit our website, of course, and find more information there. And thereby I want to thank you for listening. This is the root group at Forschungszentrum Jülich. And I also want to have a special thanks for these three guys because they are the main contributors to the code beside myself, Christian Kope and Schaefer and Chris Black. And thereby I am open for questions if there's time.
Thank you, Johannes, for the great presentation. There is a question in the chat. It says, Johannes, nice presentation. Do you think root and tuber crops like cassava pose particular challenges quantifying and modeling their root system architecture, or model renderings of other crops can seamlessly be adapted to RTCS?
Yes, so the storage root crops definitely are a special case, and so we put quite a bit of effort in building simulations for cassava, and now we are working on sweet potato in the root-to-rest project. They are special because, of course, they have this big carbon sink, which is basically means that they have all the carbon of the world, if you like, right? But they would like to put it into the storage roots. So these storage roots, they could be a tremendous source of carbon. So you can't really talk about carbon limitations, but at the same time, they're a great competitor for carbon as well, right? And the other thing that I think makes these storage roots, these storage crops quite challenging is when you think about water relations, as when those storage organs become bigger, they actually contain a lot of water, right? That buffers basically your transpiration, and thereby basically they also have an effect on the drought tolerance of these crops. Then last, these crops, I think the reason, the ecology why they make those storage roots is because they want to overcome, for example, drought period. Like cassava is very tolerant to drought, but basically when the drought season comes, it will drop most of its leaves and it will live, and it assumes that after the drought is over and the rain comes, it can regrow from that storage root, right? Of course, the farmer will harvest before that because he wants his starch. Yeah, so the ecology of these plants is also quite different from our normal crops, I think, and that makes them quite different. Yeah.
Thank you. And I think we have still 2 minutes for one question, and I can make one question because so
sure.
Regarding to the root plasticity, I was just curious how to define in the soil matrix the specific nitrogen spots, or how normally do you do that?
Yes. So mostly we have been using this in the context of studies where people have, for example, a riser box like I showed, right? And they specifically placed a piece of fertilizer in a box or as a line, as a spot. So you kind of know your map, and then we try to specify that. We specify it basically by giving a list of 3D coordinates with concentrations, and then the model can in 3D interpolate basically that. So the interpolation methods for then to get a concentration across the whole space. We also implemented the ability to do kind of random spots, right, to generate random patterns, finer or coarser. But I've not really used that in any publication yet. It was more for fun than for reality. Yeah, I guess. Yeah, maybe
Very
in the
cool.
future. Yeah.
Cool. Thank you, Hannes.
You're welcome.
Okay, so so now we are out of time, so we can go to the next speaker. Thank you so much, Johannes. And the next speaker is Mona Giraud. She's a final year PhD student at the Forschungszentrum Jülich at the University of Bonn, working with Dr. Andreas Schneff, and her research focused on the development of the C-plan box, which is another functional structure and plant model for roots. And she focuses specifically on the water and carbon fluxes through the continuum of soil, plant, and atmosphere. So, Mona, if you're ready, Please, the floor is yours.
Yes, thank you very much for the opportunity to present today and for the introduction. I hope you can see correctly the screen.
Yeah,
and
we can see it.
yes,
Thank you.
Very good. So today I'll be presenting part of the work I did during my PhD, and it's related to, as it is written, the in silico analysis of carbon and water dynamics in the rhizosphere under drought conditions. So to get started with, so the rhizosphere engineering is the idea of shifting the pools or the processes in the rhizosphere in order to reach specific aims. So for instance, diminishing the CO2 output. In order to do that, a lot of different management practices are possible at different scale. So ranging from the from agronomic management in the field, or more chemical or biological change at the root scale. And so in order to know what management practices could be useful and when to implement them, one option could be to use mechanistic models that can represent different processes across scales and domains, where you see a graphical representation on the right. So a very simple example would be in a study from 2010, where we have one specific root system in the soil, and because we had different types of soil, the same water uptake led to different water potential profile. And so therefore, in this study, we were interested in setting up a multi-domain, multi-scaled, and multi-process framework, specifically to study the interaction of water and carbon at the plant and at the root scale. And so the process we wanted to look at were the plant development, microbial dynamics, carbon flux, water flow, and for a specific soil structure. And yes, if you're interested in, to go into more detail, what I'm going to show is related to two papers, one published in 2023 and another which is currently under review, but which is available as preprint. And so to do our simulation, we decided to use functional structural plant models, which is the same type as model as the one that was presented by Johannes in the previous talk. And so it's a scale that is between the molecular and the field scale, and it's interesting if we want to look at the interaction between gene, environment, and management practices. And also, if so, some common studies at this scale could be even looking at the resource acquisition or interactions between plants. And so we implemented this model to look at the effect of drought of 1 week on the plant and on its local environment to look at the effect on water uptake and the carbon allocation. And the two assumptions we started with and we wanted to verify was whether drought would lead to higher exudation rates, and if this higher exudation rate would lead to higher CO2 emissions. So now we'll talk a little bit about the models for the plant and for the soil. So starting with the plant, we represent the plant topology using a graph. So the plant is made of nodes linked by segments. So later in the talk, I'll be talking about root segments, and it will be then basically those small units of a plant. And the morphology is represented implicitly according to data that is stored in the plant. Then for the water uptake, we have the loss of water through the leaf by transpiration, the flow of water through the plant according to the pressure gradient, and the uptake in the roots. And then the plant growth is water dependent, where we need to have a minimum turgor pressure. For the carbon, we have the assimilation of carbon in the leaves, again the flow through the plant and the release in the roots, and plant growth become a carbon sink. So that was for plants. Moving on to the soil. So around each root, we represent what we call the perirhizal zone. And that is this gray area around each root, which does not have to be equal to the rhizosphere. And it's represented implicitly via this 1D model, which is this blue line. And what are the processes that we include? We have the flow of water and several carbon pools, so high molecular weight organic C compounds, a low molecular weight, so that can also increase when we have exudation from the plant. And it can either be sorbed on the soil or be taken up by the microbial community. And for the microbes, so they can be either active or dormant according to the stresses or resource available. And they are divided between oligotrophs and copiotrophs. And they are defined, so those two groups are defined according to their substrate affinity and metabolic rates. Finally, the microbes, when they die, their carbon goes back to the organic matter pool, and we also have microbial respiration. So that's for plant and soil, and they are coupled in this multi-scale framework. So we have here our plant model. For, as mentioned before, for each root segment, we have one 1D model that is implemented. And we also have a 3D soil made of soil voxel. And so when we look at the reaction of carbon, we have two options. Either we are in a voxel which has at least one root, like here. And so then the reactions are computed within the 1D models. Or we are in a voxel that has no roots, like in this case, and then the reactions are computed at the voxel scale. And therefore we have a variable resolution and we get a higher resolution in the area of soil that are of interest to us, which are the one near the root soil interface. And I'll be showing the results in the next slide and talking about two types of results. There are the microscale results, which would be the mean value in all the voxel where we had at least one root in one of our scenarios at one moment. So it would be then a fixed volume. And then the perirhizo scale results, which will be the data for each cell of a 1D model. So here we'll have a higher resolution, but the volume will change according to time and according to the scenario. And so we did a first calibration using experimental and literature data to calibrate a generic C3 monocot and several parameter sets. But yes, here I will show just one microbial parameter set. And we had three scenarios. One baseline where the water content was always high. One early dry spell where at day 11 we had a sharp decrease in soil water. And then the water potential kept on decreasing as the plants transpirated. And then at day 18 we went back to high water potential. And the late dry spell where the same thing happened, but 1 week later. And so now for some results. So here you see a presentation of the output that we got. And so on the first panel, you see the mean plant water potential according to time with the early drought, which is between the first two dotted line and the late drought, which starts afterwards. And in green, you have the early dry spell, and orange, the late dry spell scenario, and in black, the baseline. And so without much surprise, the plant water potential decreases during the droughts. And in the second panel, we see the effect this has on the amount of carbon used for growth. And so in this simulation, the drought had a very strong effect on the growth, and it almost completely stops during the droughts. And outside of the drought, also during, so around midday when we had a high transpiration, also we have some pause in the growth, which starts again in the evening using the starch reserves. And we can see the effect of drought on the exudation as well, where we had diverging results. For the late droughts, because we have all of this carbon which is not used for growth, the leftover carbon is then in part exudated, and we get a higher cumulative exudation compared with the baseline. But for the early drought, because the plant had not had as much time to grow, the leaf surface area was quite small, as well as the root surface area, and therefore we got a lower total exudation, cumulative exudation. And if we look at the free square at the bottom of the graph, so that's the output of all of our 1D models. So you see a little example on the left where we have for each cell of a 1D model a data point. And so here, because we have all of 1D models, we have a lot of data points, and they are ordered on the x-axis according to their distance from the roots they are allocated to. So the one at 0 are at the root surface interface. And on the y-axis, we see the concentration of dissolved organic carbon. And outside of the dry spell, it increases during the day when you have high oxidation, and it decreases at night. And the increase in solutes makes the point turn to yellow. And that represents the fact that the ratio of active to total microbes increases. And so by this balance, we see that we have a type of microbes that have a starving survival lifestyle. So as soon as resources are available, they develop quickly and take up what is there. And then at night, or when there are less resources, they use up what is there, and we get even a lower, amount of carbon compared with the initial conditions, and the microbes become dormant again. And during the dry spell, we get even higher concentration of dissolved organic carbon because we have less water and because we have a more constant exudation. But in spite of this, the microbe, we still see the point turning blue and green because the microbes suffer from water stress. And here we move on to some macro scale results. Here we compare the amount of carbon in our different pool according to the baseline. So when we have a 0 value, it means we have the exact same amount as in the baseline scenario. So here we start from the bottom where we have our amount of carbon in the solutes, and we see that during the drought we have first an increase of carbon, even though we had for the early drought a lower exudation. And that's an effect of the root system size. So it is exudated over a smaller amount of, a lower volume of soil because the root system does not grow as much. And so we get high concentration of carbon, and it's because it's less well divided in the soil, it's less available to the microbes that are here compared with the baseline scenario. And the same thing happened in the late drought, but also we have this higher exudation. And then we see that later in the simulation, we start decreasing again as the microbes have time to develop and take up what is there. If we move on to the microbial development, so for the early drought, we have lower development of microbes because we also have this lower exudation. And it's the opposite for the late drought where we end up with a higher microbial biomass. And one small remark, although we don't represent all the processes that could allow us to simulate the Birch effect, we still could see here that after the drought, we had a slight uptick compared with the baseline of activated microbes because the water stress disappears, and then the microbe can profit of the carbon that had accumulated during the drought. And here, because we had a high amount of dormant microbes, the amount of emitted CO2 was more collected to the total microbial biomass compared with activated microbes. And so now looking back at our initial assumption, drought did not always lead to a higher exudation rate. It depended on the initial development stage of the plant. And because we have these very active microbes with this starving survival lifestyle, when more carbon was available, we did get a relatively higher CO2 emission, but that was also dependent on our calibration of the microbial community. And also we did a first comparison of the results qualitatively with a meta-analysis of the effect of drought on the plant and the soil. Using the study of Deng et al. And so, as in their meta-analysis, the drought led to a lower biomass in the root and shoot. And partly because of a higher exudation in some cases, and also because of a lower soil water content, we got a higher dissolved organic carbon concentration. For the microbial biomass, so when you see this red arrow, it means that it's different from what was seen in the meta-analysis. And when it's red and when it's green, it's as was observed by Deng et al. And yes, and for the microbial biomass, whether we saw an increase or a decrease depended on the types of microbes that we represented. and the same for the microbial emission. So more active microbes were able to increase in spite of a drought and respirate more. And finally, the concentration of soil organic carbon, contrary to the study, we saw an increase generally, although it was a small one. And it's partly because currently we don't represent the fact that the dead matter of a plant is added to the soil organic carbon pool. And if we also represented this process and simulated drought over a longer period, we could see, as observed in the meta-analysis, a decrease of soil organic carbon in the droughts. And so as overall conclusion, so the model, so this first implementation of our multi-scale framework allowed us to see, to represent different strategies of the plant and the soil under stress, and to look at how the carbon allocation varies over time. It can allow us to look at the additive effect of different stresses, the effect of specific phenotype of management practices. And looking forward, my work was part of a first stage of a project called CROP. And in the second stage, we will use experimental data that is now available from the first stage of our project to do more calibration. Also here you see a representation of a field experiment where we look at the effect of nitrogen. And we will also include nitrogen in our model. And yes, and at the bottom you see other members of the CROP project which helped in this study. And the model I presented was further developed during the work, but it was an existing model. And here you see all so people that are currently or were formerly part of a group that participated in the model development, led by Andreas Schnepp, a group leader. And I'll leave you with a small video while we go into questions.
Thank you very much, Mona, for the nice presentation. And so I have a question regarding to the parameterization of the microbes. And so, well, that's the first question. How is the parameterization of the microbes for this particular model? And if can we implement the, I don't know, mycorrhizas or other microorganisms that interact with roots? And yeah.
So 1 second, I have a small, so regarding the calibration, I have a small slide on this. You see, hopefully. So we did what was called parameter and process constraints. So that was the work done by Ahmed Sirkan, where he defined a series of parameters to, here it's to look at the difference between copiotrophs and oligotrophs. And here would be specific values that should have at the end of our simulation. And so from those constraints, he defined several parameter sets. Hopefully you can still see the slide. And
yes,
from
we can
these,
see the slide.
Very good. And from all those parameter sets, we selected randomly 99. And then we simulated all their values. So for instance, here would be the fitness of oligotrophs according to copiotrophs, or the amount of emission according to the microbial biomass. And we selected 3 that should represent different characteristics. So the one I showed today is the one we call high MB, which tended to show more quickly development of a microbial biomass. And we had also a one that led to more CO2 emission and one that was less active. Yes, so we looked at the results with those 3 representative parameter sets, although I just showed one for today. And the second question was about mycorrhiza. So currently it's not part of the model, but we have another PhD student, Anna Heck, who is looking into adding this in our plant model.
It's super interesting. Thank you so much. And yeah, if anyone have any other question, they can contact Mona in person, so or by chat or email. So thank you so much, Mona. There is no more questions in the chat, so yeah. Thank you very much, and I will hand it to Tim.
Yeah, I'm sorry, I just have a small issue right now with my with the application. So if can you make me stop sharing? Or yes, here I can. I think I've stopped shared. Yes.
Yes.
Yeah, that's it. Perfect.
Yeah. Thank you.
Thank you.
Right, thank you, Ivan and Mona and Johannes. Really interesting talks. Thank you very much. And we'll move on to the next session now, which is roots. So it's for the working group Roots in Sub-Saharan Africa, and it's led by Michael Adu from Ghana. So, Michael, over to you, and you can introduce your speakers.
Thank you very much, Tim, and good afternoon to everybody online today. Yeah, so welcome to our session, Roots in Sub-Saharan Africa. We have two talks. The first one will be is by Dr. Daniel Nyadanu. He is the Deputy CEO of the Ghana's Cocoa Research Institute. That institute is mandated to work on cocoa, coffee, cashew, shea, and cola. Daniel tells me today his presentation will be on plasticity and genetic parameters in cola. I was just informed 10 minutes ago that Deputy Director, he's unavoidably been taken away. So a colleague in his lab, Dr. Edem Anyomi, will be presenting. So Edem, if you are ready, please share your screen and let's go.
Okay. Is it on?
I can't see your screen yet. Okay, now it's on. Yes.
Okay. Okay. So good afternoon, everyone. Hello. Can I go ahead?
Yeah, just go ahead. Just go on the presentation mode and go ahead. Yes.
Okay, good. So I'm presenting on the genetic variations and phenotypic plasticity of root system traits in the breeding of resistance to pre-establishment stress. So by way of introduction, cola crops trees are normally found in West Africa, and they are
If you could go into slide, can you go into slideshow? Yeah, in
yes.
Presentation mode, please.
Oh, sorry. Yes, sorry. Yeah. Is it now in? Hello?
Yes. Yes, it's now in. Go on. Yes.
Okay, good. So so cola is a crop you really found in West African sub-region, and there are several varieties of it. The two most economic ones are the Nitida and the Cola acuminata, and they have several economic importance here, and also they contain bioactive compounds, especially caffeine. So basically, that's it. You normally find them in farmers' farms, among other tree crops. But despite the socioeconomic and cultural importance of cola, it is affected by many factors. First and foremost, it wasn't really domesticated. So, I mean, varietal selection and everything has been all over the place. Now, the most important threat now is due to climate change, where the stresses are causing even the crop not to be really established even in the seedling stage. So water use efficiency and all those other parameters have not really been established for cola. And we know that if we want to actually look at adapting to environmental stresses, the main thing we start with is from the root zone. So this work seems to target that. The root traits are responsible for maintaining the plant growth and productivity under water stress conditions. And so parameters like root length, dry weight, volume, and numbers are very important and are very highly correlated with drought tolerance. In cola, there's little knowledge about the studies on the root system traits, that is under drought condition and under the normal watered conditions. There's also lack of such effort limits possibility to adapt cola to pre-establishment stresses. As I said earlier, that because we are not being able to establish them well we are losing a lot of them in our system. So the objective of this study was to select drought stress tolerant cola varieties and determine plasticity of root system traits under well watered and drought stress condition. So by way of added information, the materials we used for this work consist of hybrids, but two hybrids, the single cross hybrids and the double cross hybrids. So we are evaluating them for these root traits. So what's the result? So we found some quite interesting results. So the first figure here is the variation of the root length among the hybrids, root length. And we found that if we take a material like G24, under the drought stress condition, we are having more root length than under the normal conditions. That's the well-watered conditions. You can find that one too for some cross like G35 and another one like G11. So this early data is clearly telling us that, well, there's a whole lot of variation within this process for this root length, and actually a source for selection if we are to move forward. Similarly, when we take the next figure, which is the variation to the root volume, the entire volume of the root, we still see the same type of trends. For instance, the same G24 again, you will see that the drought stress condition produced more total root volume and variation than even at the watered one. You could also see that when you take something like G35, that one too has a very wide variation in this root volume traits compared between the hybrids we are looking at. Then if we take root network, the same thing, we can find that the materials, for instance, G10 had higher root networks than the well-watered condition. And for a cross like G29 and even G24 again, you can clearly see that in the drought conditions, they are having more root networks. So these are important indicators for establishment, because once you have better root network, it goes to help you in establishment to withstand the drought better than any counterpart that doesn't have such network. Then when we take the root perimeter also, we can still see that we could also see variations in that across these genotypes, with some genotype like G35 again having a bigger perimeter under stress condition, drought stress condition, than under well-watered conditions. And the same thing for G24. So clearly at this stage, we can even see that G24 is performing well under all the root measurements we've done so far compared to most of the other hybrids. When we come to variation in root depth, the depth of the root, again, the depth of root is very important because it is able to move and get more water, more nutrients. So under drought condition, if you have a good depth, you can pick more than somebody who doesn't have a good depth. Same when you look at the data, again, the results is showing that we have materials like the same G24 again, having it higher. You have materials like G36 also having higher than the well-watered condition, and then G43 also having a higher root depth than in the well-watered situation. Then we want to look at the root convex, that is the round area around the root, the convex side. Same way, you can see that there's so much variation, with some genotype clearly showing great difference for such traits. G24 again is in here, way ahead of everybody. And you take G36, sorry, it's also again higher for these traits. We also went on to measure variations of root average diameter, the diameter of the root. And the diameter is important because that is the whole tube that actually absorbs the water and the nutrients. Again, we see that there's clear variations among the genotypes, the crosses for these traits, and with G15, G24 again, G36 showing clear variation where the drought average root diameter is bigger than in the stress condition. And this will be a plus for your adaptability and your tolerance to the drought. Then we have variations in number of root tips. And we know what root tips are. Those are the direct area where they pick the nutrients and everything. Now we could see that one too, again, there was wide variation. You can see there were some small standouts, like for instance, G34. You clearly see that in the drought condition, it produced more number of root tips. So this means that during that time, it is still working, as opposed to other people who may be just minimizing their growth activities around that time of the drought. This guy will still be moving, still be actively involved in translocating water and nutrients across the plants. Then the variations in the maximum width of the hybrids. That one to root width. That one too, we can clearly see that there were variations in there. Then we are looking at the variations in the maximum number of roots. The higher the number, the better it is for you anyway. So you could clearly see also there were some genotypes like G24 again. Clearly, it had higher variations in the maximum number of roots, far more than its watered counterpart. Same applied to G36. Then the variations in the lower root area, the lower softer part of the root. That again was also clearly varied among all the hybrids. each other. And then we realized that root volume, root diameter, and then yes, and then we could realize that these traits were very, very close to each other. They were closely related to each other, whereas other traits were far separated from the pack here. So we went on to try to cluster the genotypes and the traits to see which traits are highly linked to some particular process that we made. And we realized that G35, G28, and some few processes are highly linked to these particular traits, giving us an indication or a basis for selecting those traits. So especially if I'm looking for leaf volume, I know that G28 and G39 are towards that traits. So these materials could be advanced in such research, in such with such goals that they are strongly linked to these traits. Then the crosses that we used were in two forms. We had the single cross hybrid and the double cross hybrid. We wanted to see if, these two traits will be separated. They will be able to be separated. But when we ran the PCA, we realized that they were all clustering together except G24. And this is the G24 that we saw throughout the work, always being different in terms of everything. So clearly, this G24 is different in all these root traits we've measured compared to all the other crosses. So what's our take home? So our take home is simple. The root traits that we measured so far are very varied in the polar hybrids that we've crossed. Drought and well-watered conditions influence root traits differently. Some of the hybrids we will say perform far better, significantly better under drought conditions than their other polar traits. And I mentioned something like G24 as one and G36 as another. Thank you very much.
Thank you very much, Adam. Yes, I think I have one comment in the chat.
Okay.
There's data on root tips that you presented. Do they refer to the number of the tips per plant, and what was the plant age? So the root tips that you reported, do they refer to the number of the tips per plant, and can you give the plant age?
Okay. Hello.
Yes, go on. We are listening, Edith.
Okay, so the work was done on seedlings at the age of hello. My network is beginning to mess up. What's wrong with my network?
Go on, we can hear you. Go on.
Can you hear me?
Yes, we can hear you, Adam. Go on.
Okay, so these seedlings.
It appears Adam is having challenges. Adam, you have a few questions in the chat, and the chat, if you could please respond to them. We have questions on the yield performance and how they correlate with the root trait. We have a question on how the hybrids perform in the field. Actually, I'm intrigued about, I'm actually happy about developing hybrids for these underutilized tree crops, which are very difficult to breed. So having hybrids for them, crossing them and trying to select for them is actually interesting. And I was also going to ask if they found maybe trade-offs between deep roots deep rooting and then maybe lateral root branching, but it appears he has some challenges with his network, so he probably would respond to this in the chat. So we'll go on to our second presentation for the working group. So our second presentation will be done by Dr. Vincent Opoku Ajimai. This is from my lab. Vincent recently completed his PhD from my lab under my supervision and my colleagues. He also did his master's in the University of Cape Coast, where he worked on rhizosheet in cowpea. In the PhD, he went to look at root system architecture phenomics in vegetables, eggplant and okra. Currently, he's working with this, the Ghana Crop Research Institute, I think now working on rice root systems. So today he's put presentation on all these crops that he's worked with, and he hopes to tell us about them in 20 minutes. So Vincent, if you can hear me, please share your screen and let's take it up. Hello, Vincent. Is Vincent here?
Yes, I think I'm online. Yes. Can you hear me?
Yeah, I can hear you. Share your screen.
Okay, good.
I still have Adam's screen shared. I don't know if it is me alone or everybody. So, listen, are you able to share your screen?
Hello.
Yes, I can hear you, but I cannot see your screen.
Can you hear me?
Adam, we've moved on to the next speaker. So could you please share your screen so that the next speaker can share? Yes.
So I think, Michael, I think Vincent is sharing the screen. I can see. If you can just put it onto the other display. So at the top, there's display settings. If you go to that and then change the view so that we can just see the main screen rather than the notes. So if you put it in presenter mode now, Vincent, it should be fine.
Go ahead, I'll get back to it. Go ahead.
I'm trying to share my screen, but it appears connecting and nothing is showing. Hello, good afternoon and good morning. I hope you can see my screen now.
We can't see your screen. We can see something. We can't see your slides though. That's it. Perfect.
Okay. So sorry for that. I think there was a little technical glitch with the network. I'll be very snappy with this. Thank you for the opportunity and good afternoon and good morning once again. I am Vincent, and within the next 20 minutes, I'll be sharing root traits for abiotic stress resilience in sub-Saharan Africa with some key examples from vegetables, cereals, and legumes. This will be the presentation outline. When we talk about food security in sub-Saharan Africa, mostly take the center stage. And this has, over the years, conditioned most of our research investments in this sector, vegetables being neglected, despite the role they play when it comes to improvement in hidden hunger and micronutrient deficiencies. Despite the role these plants play in ensuring food security in sub-Saharan Africa, they are cultivated under abiotic stress conditions, of which drought, heat, and nutrient deficiencies are amongst. And under such conditions, reduce the overall productivity of the plant. But despite numerous attempts or strategies to build resilience among these crops, most of this attention have been geared towards cereals. And even within cereals, most of this focus are centered on the above ground traits. But the question is resilience decided underneath the soil before we see the symptoms or not? And if it's so, is it possible for us to leverage this resilience underground to ensure functional productivity of these crops? So over the past years, from my master's to my PhD level, this is what myself and my team have been trying to do. We've tried to sequence or to investigate whether there is genetic diversity in below ground traits, which is the root system architectural traits, because throughout today we've all learned about the role roots play when it comes to crop productivity. So I wouldn't stress talking about the functions of food traits and how they contribute to productivity. So I'll just go straight to the various activities or the work that we've been doing so far in our lab. In our lab, we follow a simple protocol, and start by pulling together the population plants or the variety we want to create. Having put them together, we try to screen for genetic diversity within the population. And having screened for the genetic diversity, we use multi-trait analysis approach, which includes dendrogram, clustering, correlation, regressions, and ANCOVA analysis to select superior genotypes as well as poor genotypes. And having selected them, we screen them under either water regimes or nutrient amendments conditions. And afterwards, we try to assess or evaluate phenotypic plasticity among these plants or the selected genotypes. And in doing so, we use various systems in doing these activities. And with the systems, we mostly work with Rhizobox, and it's just a system that helps you to view the root growth through a transparent surface, because roots are opaque in nature. And we also use columns and also pot experimentations and also field screenings. We mostly use improvised systems in our lab. And when it comes to imaging of the roots, we have an improvised imaging box. And this was, I think, we had the motivation from Larry York, who happens to be here today. We built this system based on a prototype he is having. And after snapping our images, we use various softwares to extract these images into data. And with that, we use ImageJ, SmartRoot, and Rhizovision, which is also an open software built by Larry York. And I have to acknowledge him for that. So one of the works that I really want to talk about is vegetables, because it's the neglected, it's one of the neglected plants when it comes to building resilience. In our study, we studied the genotypic variation in rhizosheets and root hairs among okra and African eggplants. And during our investigations, we realized that there is the presence of root hairs in both vegetables. And this root hairs also contributes to the building of functional traits such as rhizosheets. And this was done at the seedling stage, that's 1 month old seedlings. And having done that, at the active vegetative stage, we also screen for genetic diversity in the root system architectural traits. And it was clear that we observe some variation genotypically among the genotypes that we screened. And here we could see on this panel, you have some of the genotypes showing a more profuse root number, volume, length, compared to the B, which tends to have a limited root number and length, compared to the C as well. And the figure beneath it just shows how the roots appear after we use ImageJ for extracting the data from them. So this is a few statistics behind what I just showed. In our study, it was clear that there is a positive association, a positive relation, or a direct relation between rhizosheet weight of these vegetables and their root length, which means root lengths are not just present in these plants, but they also contribute to the formation of rhizosheet. And we know rhizosheet has functional characteristics, and this is also genotype dependent, because you could see within this figure, those that are tagged blue, which are the superior genotypes that we selected, tend to maintain higher rhizosheet under 90% fold capacity, and even as water level declines to 60% fold capacity, you still have a more stable rhizosheet, which means the formation of rhizosheet is not only dependent on the genotype, but it's also influenced by the water level or the moisture level of the soil. And even so, superior genotypes will tend to maintain a more robust rhizosheet formation under such conditions. And it was clear that rhizosheet also showed a positive association between with relative leaf water content, indicating that rhizosheet could play a significant role in ensuring resilience by improving the hydration status of plants under abiotic stress condition. And here I'm referring to drought. So it was clear that within the superior genotypes, as rhizosheet weight increased, relative leaf water content of the leaf also increased. And when it comes to the cell damage, which was measured using electrolyte leakage, it was clear that as the rhizosheet weight increased, the cell damage level decreased, indicating that the water status of the genotypes with higher rhizosheet weight was higher, thereby reducing lipid peroxidation or the production of reactive oxygen, which could have caused cell damage among these genotypes. To know if rhizosheet could contribute to the final product in which farmers are interested in, because farmers are not interested in whether there is rhizosheet there or not. What they are interested mostly in is the yield of the crop, because that is what they are going to sell to get money. It was clear that there was a positive association between rhizosheet mass and total root length. And genotypes with longer root length also tend to increase higher yield. So which still highlights the role of food system architectural traits or below ground traits in contributing to yield under abiotic stress condition. And still I'm referring to drought, because it was clear that within the superior genotypes, even at 60% food capacity, at least there was a stable yield maintained. However, at 20% food capacity, there was a sharp decline, but still the superior genotypes were able to maintain, and it was able to do that because they tend to produce higher total root length, thereby enabling them to exploit soil resources within lower sections of the soil horizon. Another study that we also undertook was assessing how root traits could contribute to pea uptake and use efficiency among cowpea. In sub-Saharan Africa, mostly cowpea is cultivated within resource-poor environments where low P is a problem. So with the hypothesis that rhizosheet and root hair plays a significant role in the uptake of soil nutrients, we try to investigate this hypothesis. And in doing so, we first screened for the presence of rhizosheet and root hairs among population of cowpea genotypes. And here it was clear that rhizosheet were and root hairs were present in the screened genotypes. And where the genotypes had higher rhizosheet and root hairs or longer root hairs, they tend to increase the uptake of phosphorus and also improve the phosphorus use efficiency. still highlighting the fact that root system architectural trees will play a significant role, not only in ensuring resilience against drought, but also under poor nutrient conditions, root system architectural trees will play a sustainable role in ensuring resilience. Currently, what I'm investigating into is in Ghana, currently we are trying to ensure rice sustainability production across major irrigation schemes. And what I'm doing now is try to screen for trees that could ensure resilience or could ensure adaptation under AWD, which is now which has now taken center stage when it comes to water use in rice production. And here we are using three AWD or water depth level, which is the continuous flooding, where the farmers have been doing always. And here we are trying to regulate it under 2 levels, where we are doing the safe AWD or the moderate AWD, where we allow the water to decline down to 10 centimeters below the soil surface. Then at the severe AWD, where we allow the water level to drop down to 5 centimeters below the soil surface before we reflood. And doing this, I'm looking out for trees that will help us to select for genotypes that could adapt to such water fluctuating conditions. And here I'm working with 3 varieties, mostly cultivated in Ghana. And this is just to show the experimental setup. And in doing this, I first screened for genotypic variation in root system architectural trees, because with the idea that root system plays a major role when it comes to uptake of water, I centered most of the work in that direction. Though we consider the above ground parameters, but I'll be talking about only the below ground parameters. And at the seedling stage, we realized that there was diverse variation when it comes to root system architectural trees. For example, taking the panel A, you could see although there are some form of root hairs and the root length looks a little bit longer. However, comparing that to the panel B, you realize that there's more profuse and longer root hair traits compared to that of the A, that of C and D. And it means at the seedling stage, we were able to pick some form of variation or genotypic variation among the genotypes. So I better tested that when we expose this or these genotypes to these fluctuating environmental conditions or water conditions, what would happen? And this is just a synopsis or excerpt from that data. I have to sit down and run some field analysis out. And it was clear that the genotypes that showed longer root hairs at the seedling stage tend to maintain a more profuse and longer root system architecture under the moderate AWD and still maintain a more steep root system under the severe AWD compared to the A, where we use the normal farmers practice, that's the continuous irrigation. Also, it was clear that another category of genotypes, although maintained a good root system architectural traits under the moderate AWD treatment. However, when AWD tend to be severe, it tends to impede the root development, similar to that of C, where both the moderate AWD and the severe AWD tend to impede root development. And here, one of the key insights I picked during the analysis was that total root length contributed to higher relative leaf water content during the analysis, and this was genotype dependent. So it was clear that the genotypes that I earlier showed, S. bica, CRI agra, still tend to maintain a higher relative leaf water content at the continuous flooding, which is the normal farm practice, the moderate AWD, where we try to reduce it a bit, then the severe AWD, indicating that the root rates, the total root length is not only existent, but it's also playing a functional role by ensuring plant hydration. So this was done in a pot experiment. So afterwards, we went ahead to plant this selected genotype, that is the Agra and S. bica, which were selected on farmers' field. And it was clear that under both among both genotypes, the AWD tend to improve yield, where S. bica was recording 20.5% higher yield compared to that control continuous flooding, whilst the agra also tend to increase yield under AWD by 16% compared to that of the continuous flooding, which means when we use root system architectural traits to select genotypes, even at the seedling stage, we can still have functional productivity under field conditions. Despite all being said about the relevance and the importance of roots, as I've mentioned earlier and throughout today's presentation, when it comes to sub-Saharan Africa, this information is limited because I would say there is little or limited expertise when it comes to this aspect. As today, I realize most of the presentations coming from the other parts of the world we're having modeling, we're having hyperspectral imaging, we're having X-ray, CT scans, which don't have such facilities here, which tends to hinder most of the activities when it comes to phenotyping for these important traits. And since we don't have that facilities, we don't have the technical know-how, and these are some of the gaps. And also, although there are fundings that are coming to Africa, especially sub-Saharan Africa, to ensure building resilience, however, these funding are mostly geared towards building resilience when it comes to above ground traits, thereby still making the below ground traits less exploited. And I think maybe going forward, most of this funding should be geared towards building resilience in that direction than only the above ground traits. And the reason being is breeding remains incomplete without root system architectural traits. And I keep saying that maybe most of the improved varieties here in sub-Saharan Africa hasn't given the yield or the expected yield as we see, because during the breeding process, the root system architectural traits were ignored. And this could have contributed to still the reduction in yield that we see in the most part of sub-Saharan Africa. Before I end my presentation, I'll thank the organizers of this program. I'll thank my supervisors who have mentored me to this stage. And this presentation or this project wouldn't have been possible if funding agencies did not commit resources. And also, I'm coming from the Root Phenotyping and Ecophysiology Lab, University of Cape Coast, and I will thank my lab members who contributed in diverse ways to make this thing possible. Thank you.
Thank you, Vincent. I think we can just do about 2 minutes of questions. So if you could make it a bit snappy, we have about 4 questions, but make it a bit snappy for us. So the first one is asking which segmentation technique you use to measure the root hairs. So if you have information on the segmentation technique you use to measure the root hairs. Let me add the second one so that I can answer both together. And then the second one says, can you share some information or citation for the method you use to separate roots and rhizosheet from bulk soil in your studies? So maybe that one you would put the citation in underneath that question. But can you give an information on the segmentation method you used?
Okay, thank you very much. In our lab, mostly we do segmentation using ImageJ. I think there is ImageJ is really a click on tool, and there is a caption in ImageJ that allows you to do the segmentation processing. So it's kind of binary skeletonization. I think that is what I remember. There is a skeletonization section on ImageJ that allows you to do that. And when you are using Rhizovision, there is also a part, immediately you load the images, there's a part that allows you to automatically segment the image. It is automated. So you just have to tune it between 200 to 250 or below 200, which allows you to do that.
Okay, so for the citation, after here, you would put it there. We have a question from Tim. He observed that your vegetable genotypes have relatively short root hairs compared to some species. Do you see any compensation for this with mycorrhizal symbiosis? So that is a question from, do you see any compensation for this with mycorrhizal symbiosis? Any comment on that?
Thank you very much, Tim. As I said earlier, in our part, we lack such facilities, but currently those genotypes, I'm collaborating with a colleague in Belgium, and another one in Sweden, where they are working on the microbial aspect of those ones. So I can't talk much about it now since the data is not out. So I can't talk much about it now, but maybe later when the data is out and we are able to publish it, maybe I could share a link with you so that you have a link.
Okay, that'll be interesting to know. Then a quick one. Are the rice varieties you're working with, are they upland or lowland varieties? And if both are, there root phenotypic differences among upland and lowland varieties? Have you observed this?
Thank you very much. We are dealing with the sativa and the glyberimers, and you know the glyberimers are mostly the upland genotypes, and the sativas are the lowlands. So in the work, you realize that at the initial stage, since the glyberimers are mostly known for drought tolerance and they improve root traits, at the initial stage of screening, where we haven't imposed the AWD, you realize that they tend to show some form of improved system architecture traits. But under the AWD system, you have the sativa showing a more improved system compared to the glyberum, because the glyberimers are not used to excessive water. So I think that is what I observed. There are variations within them. Yes, please.
Okay, on that note, thank you very much, Vincent, and thank you very much, Daniel Nyadanu and them Anyovi for hosting us. That has been the Roots of Sub-Saharan Africa. Thank you. Tim, back to you.
Yeah, it is finished.
So back to you, Tim.
Hi, how's it going? It sounds like Vincent was very happy.
Vincent is very happy, too happy.
Yeah, exactly. So if you could stop sharing, Vincent, your screen, then we'll move on to the next work package, which is root systems biology. So, and Michael, thanks for that. We're kind of almost back on time, so we're only a few minutes late now, so that's great. So over to Amir, who is going to run the final session on root systems biology. So Amir, if you can introduce your speakers, that would be fantastic.
Great. Do you hear me well?
Hear you perfectly, yes.
Awesome. Hello, everyone. Good morning, good afternoon, good evening, wherever you are in the world. It's 7:40 a.m. in Pacific time in United States, around the west coast of US. Yeah, this is my pleasure to run this, the last session of this mini symposium on root systems biology. Very shortly, the Root System Biology Working Group aims to promote multidisciplinary collaboration and engagement among the global scientific community in the area of root systems biology in different areas, and capacity building and collaboration in integrative analysis, technology development and implementation, specifically on spatial and single cell biology, and translational research and application. So if you are interested in joining this working group, please let me know, or there is a process that you can follow in the ISSR website that you can reach out to working group leads. Cool, so let's go ahead. We have actually really great, two great speakers today. So we're going to start with our first speaker, Dr. Mark Liebold, professor at University of Missouri, United States. He's going to talk about defining cell types and cell states. Stage is yours, Mark.
Thank you very much, Amir. I'm going to share my screen, hoping that first you can hear me well.
We hear you fine, yeah.
Perfect. And okay, oh, here it is. So it should come smoothly, hopefully. What I'm seeing, I should be able to share my screen. Am I right?
Yes, we can see your screen. Just if you put it in presenter mode, it should be fine. Yeah, perfect.
Yeah, awesome.
Okay. Thank you so much. So yes, I'm going to talk to you about the work we have been doing during the last 5 years, roughly, in the field of plant single-cell biology. And today, the focus of my talk will be really on the soybean plant, and more specifically, of course, for the purpose of this workshop, on the root system. And so here, what you can see as a first picture is basically some new technologies that we have been using, named spatial transcriptomics, as a way to map the activity of genes on cross-section of an organ, in this case, the soybean nodule. And I will work extensively during my presentation, provide you information about this technology and the work we have been doing. And as part of my talk, I really want to share 4 major postulates as a way to really root the information that I will be sharing. And the first one is, I would say, quite an obvious one, which is basically that the cellular complexity of root organs is a bottleneck for system biology. And that's, I would say, the idea is really that in this case, the root system or the nodules, as you can see here on the right side with the medicago truncatula nodule or the soybean nodule, they're going to be composed by different cell types with different functions that altogether really contribute to the overall biological activity of the organ, the root system or the nodule. And so we can make, I would say, the postulate that each of those cell types will have a very different way to use their DNA, as we do, you know, between muscular cells, nerve cells, very different cell types, same DNA material, but very different way to use the genes. And so if we want to implement successful system biological understanding of root and other root organs, we need to understand how each cell type is going to use the DNA and the genome. And because my talk will be mostly on the nodulation process, I want to remind everybody about this biological process. And so nodulation in legumes is a product of a symbiotic mutualistic interaction between cell bacteria, rhizobia in this case, which are nitrogen-fixing bacteria. And so upon a chemical recognition between the bacteria and the host plant, you will see the formation in the outer cell of the infection thread, allowing the bacteria to invade, infect deeper the dividing cortical cells of the root system. And so these divisions will lead ultimately to the development of a new organ that you can see at the bottom right, where you will see those nodules infected by the bacteria, where nitrogen fixations will occur for the benefit of the plant. And in exchange, the plant will provide photosynthase, carbon sources to the symbiont. So altogether, the two partners will find, I would say, a positive outcome. And so as part of the work we have been doing in the lab, we implemented single-cell solutions as a way to explore the unique activity of genes across different cell types. And I want to show here a very short movie explaining the foundation of this system. So in single-cell biology, you always start with, of course, your plant. And it could be any organ, any species. And the idea is to decompose this plant in individual biological entities. In our case, we work extensively on nuclei isolation, which gives us a lot of flexibility. And so those nuclei could be isolated from any plant organ and collected, purified, cleaned, before we can use them as an input into a macrofluidic system. In this case, the 10x genomic system, the chromium system. And so in this macrofluidic system, you will see each nuclei paired with potentially a bead, which is covered with unique DNA sequences. And those kind of interactions between beads and cells will be embedded into all droplets, which will isolate them one versus another. And now we can move towards the reverse transcription, because each bead has a unique tag, as highlighted with those X's here. You will have a unique way to label the transcript, all of them from one single cell type with a unique tag. So you can, with the DNA sequence of the tag, reallocate a specific transcript to a specific cell. And so at the end, you have, I will say, a collection of gene expression analysis at the single cell resolution. That's basically the process of single cell biology. And so we use this high throughput droplet system to basically develop the biosynthesis of thousands of independent RNA-seq libraries, one for each cell we are analyzing from a specific organ. And so if we focus on the root system, here you can see basically the outcome of two of those experiments. On the left from the soybean root, on the right from the soybean nodule. And so what you can see here is a two-dimensional representation of the distribution of the nuclei, and each dot on those two different graphs will be one nucleus. A distribution of those nuclei according to their transcriptomic profile. And so those, what we call UMAP, highlight basically population of nuclei that share the same transcriptome associated together into the same cluster, which is highlighted here by different colors. Whenever you see two nuclei with a very different transcriptome, they will have, of course, a larger distance and will be allocated to different clusters. So that's basically the first outcome of single-cell transcriptomic experiments, is an assessment of the cellular diversity from a gene expression point of view of all the nuclei composing a specific organ, in this case, the soybean root or the soybean nodule. But now you have, of course, one caveat. When we perform single cell, we isolate the nuclei, we lose the overall organization of the cells, we lose the identity of the cells in the organ. And so for that, we have implemented with a company in Germany named Resolve Biosciences. We have implemented, I would say, high throughput and also very high resolution fish experiment as a way to explore the expression of genes on a cross section of an organ by using a very high precision collection of crop sets. And so here, for instance, from the UMAP of the root of the soybean plant, we can select a collection of multiple genes, 4 to 5 of genes, which are going to be expressed in one specific cluster highlighted here in red, or another cluster here in red as well. And those collection of prop sets can be used to question on a cross-section of the root the activity of those genes. And you can see here how well those different prop sets, highlighted in yellow, red, green, will all map against the phloem of the soybeans roots. Where for the second collection of prop sets, for specific of these second clusters, they will all map very well against the xylem tissues of the soybean root. And so we can expand basically collection and design of prop set for each cluster, each group of nuclei highlighted with a very unique transcriptome here composing the soybean root. And that's what you can see here with collection of genes expressed specifically in purple in the cortical cells or in the epidermal cells. You will have, of course, here in this case, some under the epidermal cells, yes, endodermal cells here, and you're going to see pericycle, cambium, and the vasculature. And that's helped us basically with those type of technology, mapping the activity of marker genes to name, allocate a specific function of each cluster, in this case, composing the soybean root. So we can really bring back now an identity of cell type for each cluster composing the root. Now we can apply exactly the same strategy, and here is another example on the soybean nodule, where you are going to see, for instance, in blue, a population of marker genes specific of those clusters A and B, which allow us to label those two clusters as the inner and outer cortical cells of the nodule. And so we can continue like that forward. For instance, highlighting the genes, marker genes expressed specifically in the vascular bundle of the nodule. Or, and that was very interesting to us, we tested also prop sets that can detect expression of the symbiont genes. And here you have a collection of prop cells that will highlight in the center of the soybean nodule all those cells infected by the bacteria. And so that means this technology, molecular cartography by Resolve Bioscience, allows us to explore at the same time the expression of genes from the host, but also from the microbes. What was very interesting to us is when we start to explore the activity of genes in red here, of marker genes here for those red clusters. And you can see that those 4 genes are going to be all expressed in the infected cells of the nodule. You have a perfect overlap between those red labeled transcript and the yellow one. And for instance, as you can see here, when we use marker genes for the bacteria, you have a zone which is depleted, which is uninfected cells in the infection zone of the nodule. It's pretty well defined. And you can see our red markers show exactly the same kind of depletion in the same location. So this is, I would say, a beautiful way for us to have now some very interesting marker genes of the infected cells of the nodule for 3 clusters, F, G, and H, there, all 3. Oppositely, we have also a very nice detection of marker genes, 2 of them, specifically in the cells not infected by the bacteria, but that will still belong to the infection zone of the nodule, and that's the one in green. So we have, I will say, a very high resolution, but also it's a very clean set of information that we can access from this technology, molecular cartography. Now I really want to focus on those 3 clusters. This is very interesting to us because that suggests that potentially we can capture at the single cell level cells that are all infected by the bacteria, as Resolve Bioscience tell us, but with different transcriptome, suggesting potentially different states of cells infected by the bacteria. And so because we cannot omit the possibilities that those cells were identified or collected from different nodules, we co-explore the possibilities that those cells are coexisting in the same nodule. So first, what we did, we performed a comprehensive analysis of the unique transcriptomic feature of those different clusters. And when we compare the cluster F and G in terms of their transcriptomic profile, they don't show so much differences. So what we did, we focused mostly comparing FG together versus H. And that's where we can see some very interesting information. In the cluster FNG, you're going to see a lot of genes associated with RNA processing, transport, and chromatin modification, suggesting that they are very much involved in the biosynthesis of RNAs. Where in the cluster H, you're going to see an enrichment of genes involved in the nodulation process per se, and more specifically in the organonitrogen compound metabolomics and biosynthetic process. So that means here we have a population of cells which are active in fixing, assimilating the nitrogen with the symbionts. FNG doesn't seem to be involved in this process of nitrogen fixation yet. And as a way to further support that, we explore the activity of some very interesting marker genes. CCS52 in legumes is a gene which is an inhibitor of mitosis. Basically, when it's expressed, like in this case in the FNG cluster, it's involved in what we call endoreduplication. Basically, you multiply the number of copies of DNA as a way to boost transcription and its metabolism. And you can see those two genes are going to be almost specifically expressed in FNG, like if those cell types are really dedicated in this process of endoreduplication. Oppositely, in the cluster H, we are going to see a massive activity of the leg hemoglobin gene which is very important to maximize nitrogen fixation by the microbial nitrogenase. And so this is, I would say, a very good demonstration that the cells in the H cluster are really involved in active nitrogen fixation with their symbionts. So it seems to have really two different populations of cell types, all of them infected by the bacteria, but having very different kind of biological properties. And so what we did next, we explored if those two different cell types can coexist in the same nodule. And for that, we went back to our molecular cartography experiment. So you have in red here genes that are in red you have genes that are specifically expressed in the infected cells of the nodule, the cluster F, G, and H. And you can see that here on this dot plot, where you can see a very large dot with more colors, of course, for F and G for this specific gene. But what starts to be interesting is we have another marker which is specifically expressed in the H cluster, not in the F and G. It's also expressed to some extent into the vascular bundle and the cortical cells, and despite I don't show this information here, we can confirm this activity. But what was very interesting is this activity specific to the H from molecular cartography is restricted to a few cells infected by the bacteria, suggesting that those cells where you have both green and red labeling belong to the H cluster, where those where you have only the red labeling will be likely the F and G cluster. So that tells us that those different subtype or state of cells infected by the bacteria really coexist in the same nodule. And so that leads us to now move towards a model where in the soybean nodule you will see different population of cell types infected by the bacteria. And those cell types are going to have, I would say, a similar activity compared to those defined in the indeterminate nodule, like the medicago nodule, where you have zone 2, which is a collection of cells infected by the bacteria and moving towards endoreduplication, and zone 3 in those indeterminate nodules, which are population of cells infected by the bacteria and really involved in nitrogen fixation. So those two cell types seems to be present also in the soybean nodule, with, of course, in this case, a very different tissue organization in the determinate nodule versus the indeterminate nodule. So now what I want to finish is really to move towards two other postulates. Number 3 is really that to understand the plant cells as biological systems, it's very important to bring contextual information. And for that, we expanded our single cell approach to multiple soybean organs. And so we have now with this, what we call tabular glycine max, the overall picture at the single cell level of the activity of the genes. And so we have, as part of this atlas, 10 different organs, and the total number of clusters is 156, and we have over 110,000 nuclear neurons. And so we have different ways to approach now and analyze this information. First, what we did, we look at some genes that has been preferentially expressed in the soybean root, 352 of them, based on the previous publication. And we reconsider their activity. And so, yes, it's true that all those genes are going to be mostly expressed in the root system, but also to some extent to the nodule. But you can see with those red dots some very punctual high activity of those genes in specific cell type from other soybean organs, suggesting they are not highly specific to the root and the nodule cell types. They can have also some activity somewhere else. And here you can have another conclusion, which is the differential activity of those marker genes, 352, across all the different cell types composing the soybean root. And you can see how they can fluctuate based on this heat map between cell types, saying that some might be very active in the epidermis, some other more in the vasculature. And so that's something also important if you want to fully understand their function. I want to highlight a different perspective also from the atlas, which is here a Z-score heat map of co-expression of transcription factor genes. And that was very surprising to us because what we can notice here with this kind of very highly intense blue color is set of transcription factor genes which are co-expressed together in a specific cell type, which are highlighted here on the y-axis, 156 different clusters. And so that tells us that each cell type has now a very unique way to trigger the co-expression of transcription factors, very likely to drive their biological activity. Sometimes we can see a pretty nice overlap of co-expressed transcription factors between two different clusters. And in this case, for two different leaves, that was a guar cell. We have also a very similar set of co-expressed transcription factors between the companion cells of the leaves. What was very interesting is here you have a population of transcription factors which are co-expressed together in the nodule vascular bundle, but also in the pericycle of the root, suggesting some kind of potentially recycling of transcription factor function between different cell types from two different organs. And that basically triggers interesting questions about gene regulation. And so here I want to again to highlight those infected cells of the nodule, F, G, and H cluster that are all infected by the bacteria, and the F and G show a very similar hotspot of co-expressed genes. I want to finish my talk by moving towards postulate number 4. A transcriptome is of course informative, but it does not reveal always the entire complexity of a cell type, specifically because we do not assess post-transcriptional regulations. And in this case, I want to finish my talk by showing how molecular cartography and the high resolution here with multiple zoom in can really benefit our work. So what we did, we analyzed here the positioning of each transcript that you can see with those green dots in the X, Y, and Z axis. So we have a three-dimensional analysis of transcript positioning in the infected cells of the nodule. And we did that because we noticed, depending the population of transcript from gene 1 here or from gene 2 there, we noticed very different pattern of distribution of the transcripts. It could be everywhere in the cell on the left, or very much distributed at the periphery on the right, suggesting that even if you find a gene expressed, transcript allocation can change drastically even in the same cells. The cells can basically drive localization of transcript in very unique ways. And so what we did, we developed a collaboration with Erika Mesquita at the University of Missouri to transform the segmentation of the cells, the definition of the cells here with cell wall staining, chalcofluor white in this case, the localization of the nuclei with DAPI, and the XYZ coordinate of over 1,500,000 transcripts, and analyze the distribution of those transcripts in the context of the cells. And so you have basically a transformation of this distribution here on those two examples for two different genes. The red genes show a very specific and unique peripheral localization of the transcripts, whereas the blue genes will be a much more homogeneous distribution. And so we can transform this information into a heat map and then perform PCA analysis to try to understand the heterogeneity of those patterns. In red, here, you can see the high heterogeneity towards PC1 and PC2 of the localization of those transcripts, where for the blue gene, it's much more homogeneous. Looking at very closely the distribution of the transcripts, we can define that PC1 is mostly associated with a distance existing between those hotspots, where PC2 is more a reflection of the intensity of the hotspots. Now, for me, the big question is, how can we interpret this information? So we move back to the cross-section of the nodule from molecular cartography and spot each cell based on their PC1 and PC2 value for the two genes of interest. And you can see that for PC1, it might not be, I will say, very obvious, but for PC2, we were able to draw some interesting coefficient of correlations with pretty interesting R-squares associated to the concept of eccentricity, basically from the inner to the outer part of the nodule. So it seems that the distribution of the transcript follows this concept of eccentricity, which is important in nodulation because it's also associated with the senescence process of the infected cells. It's always start at the center of the nodule and always finish at the periphery. And so that suggests that potentially we have here a molecular reflection of the fitness state of the soybean rhizobia infected cells in the nodule. So I want to finish my talk by sharing four different conclusions, four different postulates. First, cellular complexity is really a bottleneck for systems biology. And for that, postulate number 2 tells us that the single cell resolutions can help us to better map molecular mechanisms. Postulate number 3 will be that it's important to always bring back molecular information in a larger context to really extract what is unique to a specific cell type or organ. And finally, let's acknowledge that the transcriptome is just, I would say, one part of the equations, and other regulatory mechanisms could be important players. And I want to thank the members of my lab who have been working extensively on those projects, specifically Dr. Yawali, Sutton Tennant, who worked extensively on the molecular cartography experiments, Sandra Tibivillier, who did a lot of work developing and implementing single-cell solutions, and Erika Mesquita Moataya, who has been a good collaborator to develop those analyses of subcellular localization of transcripts. And I will be more than happy to answer any questions. Thank you.
Wonderful. That great talk, Mark. So, yeah, if there's any question in the chat, I just put one question there, and probably don't have much time for too many questions. Maybe, yeah, just one question I just put there. So what do you think would be the main challenges in using single-cell omics in coming years in root rhizosphere biology? For example, do you see challenges in availability of cell type markers specifically, or cell type or tissue-specific promoters, data analysis tools, or high-throughput genome editing tools to use the large set of data that are producing using these approaches for functional analysis? Would like to see your envision.
Yeah. I mean, I think for data analysis, we are going to see likely revolution, and it starts to be, I will say, more advanced, of course, in animal science with the use of artificial intelligence to support analytics. So it's going to take a little bit of time so we can really digest all this information, but hopefully it will come. I think for really, the plant rhizosphere interaction, to me, there's two bottlenecks. First, also capturing the microbial transcriptome. What I show today is a very, a narrow picture of gene expression from bacteria using molecular cartography. It does not reflect the overall transcriptome of the bacteria. And expand, of course, this kind of analysis to understand the physical interactions between plant and microbe, thinking about heterogeneity in the microbiota, depending, the distance from the host, depending also the depth in the soil, depending environmental conditions. It's going to be a very heterogeneous and dynamic system that will require likely a lot of investigation. But first, we need, I think, to find good solutions to capture the microbial transcriptome.
Wonderful. Thank you so much. There's another question in the chat, but I think to save time, we need to proceed with the next talk. But Mark, if you can please look at that. It's a great question by Joseph. If you can just reply to that question in the chat box would be great. Thank you again. Okay, so let's move forward with the next talk. So our next presenter is Kevin Daniel from University of Freiburg, Germany, and he's going to talk about the from root death to repatterning in Arabidopsis under waterlogging stress. Kevin, stage is yours.
Yes, hello.
Hi.
Are we good on the setup? Great. Okay, I'm going to start sharing my screen. Okay.
Yeah, we see your screen, but it's not on presentation mode. We see your node.
I think now we're good.
Yeah.
Well, thank you for having me, and thank you for sticking around. I feel probably you're like me, your head is full of information, but I'm glad you made it all this way. So, Yeah, I'm working in the Hartmann lab. We are in a lab that is mainly focused on flooding biology, especially what kind of signals are involved in the flooding biology. But now in my project, we are also integrating root biology. And this is the reason why I am here. And surely you know that abiotic stress define root system architecture. So you know all about that, right? It's the shape of a root system, and it changes depending on what kind of abiotic stress it is subjected to. And the thing is, if we think about what does the root system architecture look like for a plant which has been waterlogged. So this means that the water level goes up to the soil, but the shoot is still in the air. Maybe you don't really have an idea what's going on. And we are really trying to answer this question of what does the root system architecture of waterlogged root systems look like. You know all of the root stuff, so I'm going to talk a bit about the flooding stuff. And what is important, I really love this analysis from a few years ago, where they looked over 100 studies, and they really tried to see how much is the impact of waterlogging on the roots or on the plant in general. And if we imagine that the area of all of the countries in the world is 100%, and we now know that 12% of arable land is affected by waterlogging, then we could say that this is respective of an area of the size of Europe, China, India, and Venezuela together. And here we also had a look at traits which are associated with the plant fitness. And here we saw that we have a crop yield decrease of almost a third. So yeah, waterlogging does a lot to the plant health, and we can also start to ask the question why it does that. So this is our plant right now. It's still happy. In this plant, it's in the soil. We have a certain oxygen gradient, and then there's a lot of things happening, but many of them revolve around gas diffusion, right? So for photosynthesis, we have carbon dioxide coming in, oxygen coming out, but also in the soil, the roots take up oxygen from their environment through the process of diffusion. And on the other hand, it produces a gaseous phytohormone, which is called ethylene. And this hormone then leaves the root and dissipates into its environment. And of course, we also have a whole soil composition with different bacterial communities and chemical compounds. And all of this changes now if we have a root or a plant which is subjected to waterlogging. So first of all, we will have a much deeper oxygen gradient. And because in the water, gas diffusion is slowed by a rate of 10,000, we will have a decrease in photosynthetic rate. And also in the root, we have a new scenario. So on one hand, the oxygen cannot enter the root. This leads to the lack of oxygen, which we call hypoxia. On the other hand, this gaseous phytohormone, it can now not leave the close proximity of the root. And so it starts accumulating. And taking together this lack of oxygen and this accumulation of ethylene are two signals which are very important for the plant in understanding that it is actually under the water at this very moment. Finally, we have a whole shift in the bacterial communities from aerobic to anaerobic, and as a consequence of that, also a change in the chemical composition of the soil. So a lot is happening, right? Okay. Now, when we started this project 3 years ago, we're still interested in root shape under waterlogging. We had a look in the literature, what we can find, and we also wrote a review about this. We already had some insights. So first of all, we can see that, that the insights that we gain is really centered around the newly introduced ethylene or hypoxia. And we can see that it affects the root growth, the lateral root formation, and the root bending. But we also saw that many of these insights were gained from agar plates, in which we just apply hypoxia or ethylene or its precursor. And of course, you know that's vastly different from the situation in the field. And now to address the question of root shape under more realistic conditions, we decided to use riser boxes. So riser boxes is probably known by most of you here, right? They allow us to see the root system in a kind of 2D structure. And our model organism is Arabidopsis here because it offers us a lot of genetic tools in the signaling research. And we actually we were quite happy with what we could see until we actually started to waterlog our plants. And I'm going to show you why. So on one hand, this is a plant which is not treated, and I think we can see the root quite nicely, so this is very great. But then directly after we actually apply the water, we run into a problem that we don't really see the root, right? So maybe you can see here that there is the root trip, right? But everything in the middle kind of gets lost. And it is really that the visibility of this very thin Arabidopsis root is decreased. So this is something to keep in mind when working with, I would say, very fine root systems, and if you're interested in waterlogging things. And this is the reason, or one of the reasons, why we had to adapt our imaging pipeline. So instead of working from the riser box, we excavate our roots, we put them on these colorful agar plates, showing their whole architecture, and then we skeletonize, we annotate using SmartRoot, and then finally we do the data analysis. Okay, now what have we found out? There's two parts to this. First of all, we ask, okay, what is this waterlogging root phenotype? And this is our standard setup. So we grow our plants until they reach a certain size. Usually that's 12 days. We waterlog them for 3 days, and then we wait another 3 days for a recovery period. This recovery period is actually quite important for us because it has been shown that when you take away the water, there is a huge amount of reoxygenation stress, which also has been shown to kill roots and root tips. But if we now do this, this is basically what we get at the end of our treatment. So we have our plants on the left, which are in the control conditions, and the plants on the right, which are in the waterlog conditions. And if we have a look at them, I think we can see some differences right from the get-go. I think most prominent is probably that we have a decreased primary root length. And we can also see this when we quantify this. And from other experiments, we know that this is the case because the primary roots actually die during the waterlogging, so the hypoxic conditions become too much and the primary root dies. Okay. We also had a look at different traits other than primary root length. For example, the lateral root length, but we really didn't see too much difference here. But if you look at the pictures, here they are again, you can probably agree that something is going on, right? So in the waterlogging condition, all of these roots, they really seem to hug the soil surface, while in the control conditions, everything shifts more towards the innards of the soil. And in trying to put this into numbers, we thought, okay, what if we just do the following? For each single lateral root, we, on one hand, try to look at the positioning, so the distance from the root base, but also its individual length. And combining these two things into one number, we now talk about a phenomenon which we call this lateral root length distribution. So what does it mean? Basically, if we waterlog our root system, we see a redistribution of where the lateral roots are in the soil. And this is a combination of two things. So on one hand, we have that the fact that we have more lateral roots near the soil surface, but also the roots are longer. So here are two different things are happening. And we were interested in, well, how does this really develop over time? So we did a time series where we varied the duration of our waterlogging stress. And this is the quantification for the primary root length. So I'm going to guide you through this a little bit. We have on the bottom are different time points, which range from 4 hours of waterlogging to 1 week of waterlogging. And then here on the very left, we just dip the riser boxes in water for a brief amount. And this basically serves as our control. And the first thing that we can see is that the primary root length between our treatment group and our control group does not seem to differ too much, but only if we waterlog for a duration of 24 hours or maybe 72 hours and more, we start to see this effect happening. And if we imagine that we put what is happening on a timeline, which is shown right here, then probably we would say that, okay, after 24 hours, we have this phenomenon that the primary root starts dying. Now, shifting to the lateral root length distribution, we can see a little bit of different pattern. So here, again, the quantification. And already from the duration of 4 hours of waterlogging, we can see that there seems to be some kind of shift happening towards the soil surface. And this phenotype becomes stronger the longer we waterlog our plants, right? So this difference right here. If we put this on our time scale, then we see that this actually happens at a different time than the primary root depth. And this was very valuable information for us because this way we could really detangle these two phenomena from each other, because one is happening before the other, and therefore we don't think that this the redistribution of the lateral root length depends on the primary root depth. Okay, finally, we also wanted to look at the position, right, and the length of the roots in trying to assess when do the roots at the top surface become longer. So here we see the relative position of the lateral roots on the y-axis and then their respective length on the x-axis. And what I want you to focus on right here is basically the slopes that we can see between our control and our waterlogged plants. And with the shorter waterlogging duration, these slopes there, it doesn't look like there's much of a relation between the relative position and the length of the lateral root. But again, only after 72 hours or more, we really start seeing the slope and also the redistribution of the points, right? So you can really see here that we have more and longer lateral roots, especially after one whole week of waterlogging. And then to finalize our little time scale, after 72 hours, we see that there is an adjusted growth of the lateral roots. And as a take-home message, basically what we see after 3 days in our waterlogging is a combination of different things which happen at different time points. Okay. As a follow-up, we asked the questions, okay, we have new roots near the soil surface. Where do they come from? And I'm sure you're aware that lateral roots, the development of lateral roots has many steps. And we are limited by the analysis pipeline that we're using at the moment. So one of the smallest root we can measure is maybe 0.5 millimeter. But if you think of an emerging lateral root, it will have the size of roughly 0.1 millimeter. Exactly. So these are like all the different options that this waterlogging in a signaling sense could feed into, right? So maybe we have outgrown roots and the waterlogging feeds into it and makes them grow more or less. This is something that could explain our phenotype. But on the other hand, we could maybe enter, or the waterlogging could signal into a stage of the lateral root development, which is much earlier, like the lateral root initiation phase or even the pre-branch sites. And to address this question, we decided to use an auxin-sensitive gust promoter. Auxin, because it is instrumental in the development of lateral roots on multiple stages. And then we looked at the distribution of the lateral root primordia between our control plants and our waterlogged plants. And the graph which I will show you is a summary of this, of all the information that we got. So on the y-axis again, basically we have the length, the primary root length. And then these little dots represent the average position of the lateral root primordia, while these red stars is the average position between all of them together. And if we look at purely at the length, well, we can see, yeah, after one day, we don't have this difference in the primary root length, but then this gets stronger, especially the longer we waterlog the whole thing, as we've seen before. And then if we start looking at these red stars, well, we see, okay, there's maybe a little bit of a shift going on in the average lateral root position, but not too much actually, right? So here's a little summary of this. But if you look at these points for long enough, I think maybe you can agree that definitely there's something going on. And to get a better understanding of what is going on, we decided, what if we just divide the root into two zones? So in the first 10 millimeters and the rest of the root, and then we count the lateral root primordia. And what we can see is actually quite interesting, because we have, opposite things happening depending on the treatment, depending on the zone. So if we are close to the soil surface, we can see that starting from 2 days, we have more lateral root primordia. But if we look at the other part of the root, we can see the opposite. So we have more lateral root primordia in the control condition. And I think the take-home message here is really that maybe taking the averages in these root traits is not always helpful, but instead, if we increase the resolution and also look at the position of individual lateral roots, then we can gain some new insights. Okay, now for the second part, I just want to briefly talk about how are these lateral roots formed, right? Because we are a signaling lab, this is interesting to us. Quick reminder, hypoxia and ethylene are both very important for the plant to acclimatize to the waterlogging stress. And then we just wanted to see, do we have this signaling going on in our Rhizobox system? And here again, we used a gas reporter system, in this case with a hypoxia sensitive promoter, HRPE gas. And here is the general quantification where we can see, okay, indeed, if we waterlog our plants, we do see an increase in the activity of the promoter, hinting hypoxia signaling. And on the other hand, we can then increase our resolution and then again distribute these data points depending on the position of the lateral roots relative to each other. And then what we can see is that the roots which are closest to the soil surface, they exhibit the weakest signal, while when we increase the depth, basically, this signal gets stronger. And it becomes really interesting if we go and look at the ethylene, because, and we use here the same system as before, just with an ethylene sensitive promoter. We have an increase in the signaling, but it shows the opposite effect. So here we have more signaling going on, and this means that now two important flooding signaling show different gradients between each other. And we think, wow, this is great. So this could explain why one thing is happening close to the soil surface, and then another thing is happening if we go further down into the soil. And we also checked signaling mutants which are related to ethylene and hypoxia. Here is the surprise that we had. None of these show, basically a restoration of the phenotype. All of them behave like wild type. And now in the interest of time, go directly to the summary. So what is happening to the roots if we waterlog them? We have the depth of the primary root, and we have a redistribution of the lateral roots, and this is caused by the initiation of new lateral roots near the soil surface. And if someone asks you, well, how does this root system now look like? I would probably think it would be something like this. And surprisingly, this was independent of ethylene hypoxia signaling. And with this, I'm done. I want to thank you for your attention. I want to thank you, my wonderful lab, and of course, all the collaborators for material and advice, and of course, all the funding agencies for making this possible. And with this, I'm very happy to take your questions.
Wonderful. Great talk, Kevin. Thank you so much. Yeah, I do see a couple of questions. First one is, do you have results on lignin-rich roots response under waterlogging?
I do not.
Okay. And second question, Joseph, thank you. Have you estimated lateral root initiation index under normal and waterlogging conditions? My point is that the initiation should not be affected, only lateral root emergence.
So this is a good point, right? And something that I was also trying to find out during the talk is, yeah, we don't really know where, at which stage this is affected at. And although I think it is quite early, we plan to do more experiments here to really confirm what is happening. In our mind, the one key experiment here would be to basically mark lateral root primordia, which are already there before the stress, and then to follow these over time after the stress to really discriminate between what is there before we start and what is there after we start. And then we can also elaborate a little bit more of, yeah, is this only lateral root emergence or initiation?
Great. Yeah, I don't see more questions. Okay, I want to thank our two speakers under this session again, Mark and Kevin. Both really did a great job. Awesome presentations. And I think, yeah, Tim, if you want to say something, if you want to adjourn this.
Yeah, no worries. Thanks again for to the speakers in that last session, and we've made it to the end. That's great. I mean, we've still got over 100 people here, so that's really gratifying to see. I mean, it's been a long day, but it's been a really interesting day, and I'd like to thank all the speakers. We've showed a real breadth of research, different approaches, different techniques, a wide range of plant species we've been looking at, and representation from across the globe, which is really great to see. I'd also really like to thank Hannah Schneider and the working group leads who've really put this together. So they worked hard to come up with this really engaging program and day. So thanks ever so much for that. And I hope that anyone who's joined today has enjoyed it as well. And if you have, then please get involved. Join the society, join a working group, and tell us about your news, and we'll publicize it to the wider community. Basically, it just leaves me to say, I look forward to seeing you at future ISRR events. We're hoping to have some activity between now and the next conference. And if not, then we'll see you at the next conference in Beijing. So thanks for joining, and it's been a really great day.